ado, I would request Professor Shopon Kumar Dotto, who is the President of the Alumni Association of the Postgraduate Department of Botany, University of Calcutta. He is the ex-head Department of Botany, CU, and the Rajbihari Ghosh Chair Professor, and member of all the Academy of Sciences in India and member of the World Academy of Sciences. I request Sir to bring on stage Professor Parthopratim Majumdar, who is our guest of honor, Dr. Himanshu Pathak, who is our chief guest, and can I also request Professor Rita Kundu, our head, Department of Botany, to come on stage as well. At the onset, I would request them to offer floral tribute to Professor A.K. Sharma.
Hope you also may join.
Beginning with the inaugural session, can I now request Professor Shopon Kumar Dotto to come here and deliver the welcome address.
Good morning to everyone for this participation in this event in this Professor Archana Sharma Hall, along with 2 PG rooms and also online, including Professor Rajiv Varsne from Australia, student scholars from IAV and Kraja Barakpur. We are privileged and very happy to see that the dignitaries we have today with us. First of all, I would like to mention Professor Himanshu Pathak, Director General, Indian Council of Agriculture Research, Secretary DR, Minister of Agriculture, Farmers Welfare, Government of India, eminent scientist of our time, with one of the highest citations and made significant change in the improvement of education, science, and administrative culture of ICR. We are grateful for his time. Thank you, sir. Professor Partho Majumdar, he has a lot of other sort of names with his name, but I would simply say he's the National Science Chair, Government of India, one of the highest position that he held today. Also former President of Indian Academy of Science, Bangalore. Thank you for accepting our invitation, ready to be with us to pay tribute to Professor A.K. Sharma. Professor Amit Ghosh is supposed to be here, but because of his family reasons, he couldn't join us today, but he also paid his tribute to Professor A.K. Sharma. Professor Rajiv Varsne, FRS, Director, Western Australia State Agriculture Biotechnology Center, and International Chair in Agriculture, Food Security, and etc., from Mardo University, Australia. Welcome, Professor Rajiv, that you joined with us today in this event. Honorable Vice Chancellor and Registrar are supposed to be here because of certain other reasons. They could not join, but they paid their tribute and wishes welcome to all of you for this event. Dr. Anindya Bandyopadhyay, currently Principal Scientist and Director Biotechnology Division of CIMET, Mexico. Although he came from Mexico, thank you, Dr. Anindya. Please stand and let students see you. He's the Genomics Center head in China at one time. He held many positions. He would be introduced later on before his lecture. So you would hear from Dr. Anindya Bandyopadhyay today. Dr. Sujay Rakshit, another eminent scientist, Director, please, Director ICAR-INDIAN. You would also listen from him. Dr. Gauranga Kaur, Director ICAR-INDIAN, Barrackpur. And there are many other eminent scientists that came from ICAR institutions. They are with us today. And we would have their interactions with you during the panel discussions. Professor Rita Kundu, the dynamic and always smiling personality, head of Botany Department and co-convener of the symposium, Professor Rita Kundu. Dr. Ila Chaudhuri, sitting here, the Vice President of the Alumni Association, and as well as instrumental to keeping the alumni alive and active. So Dr. Ila Chaudhuri. We're missing Professor Rajat Chaudhuri because of certain illness, who was the former president of the alumni and also very active advisor to the alumni. All teachers of Botany Department of Calcutta University, senior colleagues and members of Alumni Association of Botany Department, all scholars and staff of many other universities and institutions who are with us today, and fellow friends. Please accept our warm greetings and welcome for coming and participating in this unique event celebrating birth centenary of Professor Arun Kumar Sharma, who served this department and more than 50 years in an active way. And it is a privilege for us that we are all here today to pay tribute for his 100 years of birth centenary. And I would elaborate a bit more about Professor Sharma. And there is entire detail of Professor Sharma biography or his activities as a brochure is given in the folder. So please have a look when you have time. So Professor Sharma, born in Kolkata, 31st December 1924, BSc from Ashutosh College, MSc and DSc from Calcutta University in 1955. During his DSc, he supervised several PhD students. His initial research on cytogenetics started with Professor P.N. Bhaduri, another eminent cytogeneticist of India. In 1948, Professor Bhaduri left to join IARI. That's interesting. So we have a very much well connected with the ICR and IRI. And then Professor Sharma joined the department on 1952 as a lecturer and eventually became Raspberry Ghosh Chair Professor in 1970. And continued till 1988 and remained in the alma mater as an honorary professor till his last day. Students, including me, many of you may remember his teaching style. Without using slide, notes, or duster, but a mesmerizing, mellifluous diction would remain in our minds for some time, even after he left the class. We used to sit in the class remembering and thinking about Professor Sharma's lecture. That was the style that he had. I will still remember his voice after 30, 40 years. Elected fellow of major academia such as FNA, FASC, FNSC, FNAS, World Academy of Science from Italy, etc. He holds many national and international positions, and that all awards, including Paul Broll Medal from Asiatic Society in 1972, National Professor 1972, awarded Bhatnagar in 1976, Indian Science Congress Association President in 1981, INSA President 1983-84, NASHI President 2011-12, INSA Golden Jubilee Professor 1985-90, India government conferred Padma Bhushan in 1983, GM Modi Award in Biotechnology in 1994, Rathindra Award, also connected with Shantiniketan Vishwabharati, award in 2008 by Vishwabharati. Lifetime Achievement Award in 2010 by Indian Botanical Society. With his own work and research on plant chromosome, AGC recognized the Advanced Center in Cell and Chromosome Research in 1972. And today, the entire Department of Botany of Calcutta University is recognized as Center of Advanced Study due to his and his wife, Professor Archana Sharma's enormous contributions in the subject. A few noteworthy research contributions of Professor A.K. Sharma. Just a few of them I would mention. Number one, detailed systematic studies of chromosome number and structure with microevolutionary changes in karyotypes. Number two, novel and innovative techniques to elucidate physical and chemical structure of plant chromosomes. The techniques of Orsin banding to locate the specific DNA repeats. Number three, chromosomes behavior in differentiated cells resulted amplified additional DNA to respond the demand of cells, which was referred as dynamic DNA for either various functions in differentiated tissues. And many more on cytotaxonomy, developmental and evolutionary biology. Number four, launched the International Journal Nucleus. There is a poster outside. Please have a look of that genesis and development of the nucleus. In 1958, at the age of only 33 years. Now it is attached with Springer Nature. P.P. Kaufman, M.P. Riley, H. Kihara, A. Love, C.D. Darlington, and many other eminent cytogeneticists at that time were attached with his journal Nucleus. And Professor Randall from Cornell University referred his lab the largest chromosome family of the world. As we know today, 80 PhD students and 10 DSc were awarded associated with his supervision, with 500 publications and 6 of them in Nature. Number 5, Chromosome Techniques, Theory and Practice, published by Butterworth in 1983 with 3 editions. That's all a few things that I mentioned, the details. Please look at the brochure that's given in your folder. Sir laid the platform of cytogenetics, which led today's modern genomic research, genome editing, location of genes, characterization of genes, linkage, shears, and phage staining today help us to assign specific genes attached to traits. Example, PIB, big genes that leaf blast of rice, etc. Many Japanese scientists now are following those staining and locating those genes that are either QTLs, that is referred today in very much in the plant breeding. It is not possible to portray Sir with any writer narrations, and so that he can be completely portrayed to you, but you can always imagine what a big picture that he portrayed to us. On behalf of Alumni Association of Botany Department and on my behalf, taking the pride once again welcoming all of you to join us to remember Sir and enjoy celebrating 100 years of Professor Arun Kumar Sharma, to whom we are much grateful as we represent today as a plant scientist in our relative capacity to pay tribute to Sir, and hope all the cooperation we advance the cell and chromosome research and the overall the plant science in the greater dimension of advancement in plant biology. As founder president of the alumni, and during his entire time, he was the key person of inspiration, love, and respect for all of us. And we would remain grateful, remembering his advice, vision, and guidance. A pride for us. Thank you for joining today to make the successful event of birth centenary celebration of Professor A.K. Sharma. Thank you very much.
Thank you so much, sir, for the wonderful welcome address and the amazing words that you said about Professor Sharma. Can I now request our head Department of Botany to felicitate the guests on stage? I would request our research scholars, Monisha, Shubhanita, and Shejuti, to help ma'am do the felicitation honors. I would request Professor Dotto to felicitate our HOD, ma'am. Thank you very much. Thank you, Shejuti, Shubhanita, and Monisha for helping us. We have between us our guest of honor, Mr. Partho Pratim Mojumdar, National Science Chair, Government of India. Can I request you, sir, to please say a few words on this occasion?
Thank you very much. I'm actually very honored to be called upon to be here on stage. I'm probably the most odd man out here in this audience. I have no connection with botany. The reason why I'm here is that I've had some connection with Professor Arun Sharma. Although, even though I've said that I have no connection with botany, I would like to recall for myself the fact that the first research paper that I wrote was published in the Journal of the Botanical Society of Bengal. I'm not even going to describe the contents of that paper, but that's something that I remember very fondly. I have known Professor Sharma. I've known actually Professor Archana Sharma much more closely than I've known Professor Arun Sharma, primarily because Professor Archana Sharma at some point of time in her life started doing work on human genetics, and that's what I do as for my professional that's my professional background. I actually got introduced to Professor Arun Sharma in a serendipitous manner. I grew up at the Indian Statistical Institute. My background is in statistics. One day I went to the library and found this journal, The Nucleus, of which Professor Shapandotto just spoke. And what was very impressive is that if you looked at that journal, the pages of the journal were really, really very impressive in the sense that they were printed on the journal. The entire journal was printed on very high class glossy paper. That was very interesting. And it looked like, in the library around that, there were journals that were printed in the United States, United Kingdom, and so on. And this was something that sort of stood out. I opened the pages and found that the editor-in-chief was Professor Arun Sharma. I even read the editorial and was extremely impressed. So that's when I got to know, and this was in the early 1970s or mid 1970s, when I got got to know Professor Sharma. Subsequently, one day I got a call from Professor Sharma. He was trying to set up a department of genetics here. And he said, would I be able to sort of help him establish the syllabus or frame the syllabus? That's when I actually met Professor Sharma for the first time, after which we interacted for multiple times, discussing the syllabus, etc. And then the remembrance that I have of Professor Sharma is a very touching remembrance, which is that I was giving a talk in 2017 in the Indian Institute of Chemical Biology, and Professor Sharma came to that lecture in a wheelchair and sat in the front, before the front row, listened to my lecture very intently, and essentially left the lecture after listening to me. And that was extremely touching, because that was the first lecture that I delivered, and the whole day was going to be full of other lectures and so on. But he left immediately after. So it was a revelation for me that he essentially came to listen to me, and he passed away a few months after then, and that came as a shock. I was also very, very privileged, very affectionate of the Department of Botany, of the people who have organized the Archana Sharma Foundation, to have asked me to deliver the first Archana and Arun Sharma Memorial Oration, which I was very pleased to deliver. And likewise, I'm extremely happy to be here, standing in front of you and occupying the stage when we are celebrating the centenary of Professor Arun Sharma. So it is with deep respect that I'm here, even though I'm the oddest man out here. But it is really a pleasure for me to be here and to be in the Department of Botany to pay my great respects to Professor Arun Sharma. Thank you very much.
Thank you very much, Professor Majumdar, for your kind words. Can I now request our chief guest, Professor Himanshu Pathak, to please come here and say a few words.
Am I visible? Good morning, everyone. Dr. Shapan Datta. Of course, you know him, I think, much more than me, but he's one of the mentors of many of us here, including myself. So, sir, thank you very much for your kind invitation. Of course, Dr. Majumdar, Madam Kundu, Aninda, and many very distinguished, very, very distinguished scientists, faculty members of this great university. My colleagues from ICAR, Dr. Gaurangakar, Dr. Pradeep De, Dr. Sujay Rakshit, Dr. Rahman, and many others. And more importantly, what I find, large number of students. I think for them, this celebration is much more important than many others. So thank you very much for kindly inviting me and to be a part of this great event. It is indeed a great honor for me, also privilege, to stand before you on this very auspicious and very important when we are celebrating the birth centenary of one of the doyens of Indian science. Not only botany, not only cytogenetics, but whole science of India. And really, this is the most befitting way of giving the tribute to that great man. I have met Professor Sharma only once. Maybe in 2009 or 2010, when sir, you organized one conference here in this department, and you again requested me to deliver a talk. Dr. Sharma was there in that particular talk, and then after that talk, I met him in his chamber. And that was my only association with him. But I met his students, many. And in Sanskrit, they say that phalena parichayate vriksham. A tree is known by its fruits. So whose students are so great, one can imagine what the greatness of the teacher could be. And that one sentence probably can summarize large persona of Professor Sharma. As Dr. Datta was talking, more than 80 PhDs and more than 15 or 16 DSc, and n number of students who have contributed and who are still contributing in a very big way, not only in the field of cytogenetics, biotechnology, but also in many other areas. I think that is the contribution of Professor Sharma. Friends, with this, with the celebration of birth centenary of Professor Sharma, we are also celebrating science, which is really, really important. And of course, this city, though more known as for many things, City of Joy and others, but then I feel this is the city of science. This is the city of science and city of scientists. And probably Dr. Sharma is one of the towering names in the whole list of so many, starting from Dr. Jagadish Chandra Bose, Dr. Meghnad Saha, Yashan Bose, basically, and of course the list is quite long. And this university is again has been one of the sites of all these great men who have contributed so much. Celebrating science nowadays is very important. Celebrating science and performing science has never been easy. As again, they say that science is always in minority. Idea comes only to 1 or 2%. It doesn't come to everybody at all. So the starting comes with 1 or 2 persons, and then you have to convince to the others what you are trying to say. But then scientific spirit and cultivating scientific spirit is something which every society really needs to do. And probably for that reason, in our constitution also, it has been written that one of the duties of every citizen is to promote scientific temper, which very often, we forget, we neglect, and even if we understand it, we do not want to pursue. So cultivating science, celebrating the spirit of science is really important. And on that count, I must congratulate and thank the Alumni Association, which is organizing this particular event. I was going through the biodata what Dr. Datta circulated to all of us. And sir, let me tell you, I have read it 2 times. I was really trying to assimilate some of the things, that how at that point of time, with very limited resources, that excellent science can be done. Now, when we run big organizations, and one of the complaints all the time we listen that we do not have facilities. We do not have this equipment, that equipment, and all kinds of things. But then when I was comparing what Dr. Sharma used to have in his time and what we have got in our time, I think in many counts, we have got much more than what Professor Sharma used to have. But the output, all of us know, is not as much as what Professor Sharma did. Because for any good output, what we need is what I said, spirit, that passion, that perseverance, which many of us are lacking. And without that spirit, without that passion, many things can be done, but not science. You cannot dive deep into science until and unless you have got that passion in you, that zeal in you, to know what is not known, to explain what is not explained. Therefore, Dr. Sharma is always a luminary, a lighthouse for all of us to follow, to take inspiration, and of course, motivating ourselves to do better and better. I shall just put before you only 4 or 5 things what probably we can learn from the life of a person like Professor Sharma. Of course, at one point of time, somebody asked Gandhiji that what the things we should learn from you. And he said, I have nothing to say. My life is my message. One can see my life, and then, of course, they can learn whatever they want to. I'm not going to say, okay, you learn these things and you do not learn these things. Probably in a very similar way, Professor Sharma's life is also something that his life was his message. You can learn, you can unlearn, you can do, you may not do, but that's what the life is. And in that life, there are only few things what all of us can really learn is one, of course, what I said, passion and perseverance. These two qualities can take anyone to a very great height. And without these two, in spite of having all the facilities and everything, probably will not be able to achieve it. So from here to Padma Bhushan, and to all the awards what Dr. Datta mentioned before, all of this were the result of this passion and perseverance. This is the first thing what we can learn from his life. Second one is, of course, extraordinary commitment for education. Teaching to students or to anybody who comes. You know, friends, many of us are associated with teaching. We have got our students, we have guided PhD, MSc. Of course, now we do not have DSc in that sense. But then probably the kind of extraordinary excellence what Professor Sharma could deliver is something which we all can. As I said, Phalena Parichayate. And this is for everybody to learn. Again, in Sanskrit, so there is a good slok, which says all of you probably, I don't know whether students, even today, they read Sanskrit or not. It says, Hey Bharati, Bharati is Saraswati. Hey Bharati Kimtava Bhandara Drishyate, Bayoto Vriddhi Mayati, Chaya Mayati, Sanchaya. Hey Saraswati, আপনার ভান্ডারটা কি বিস্ময়কর ভান্ডার, যেখানে যত ব্যয় করবেন তত আপনার সম্পদ বাড়বে, আর যত জমা করে রাখবেন তত কিন্তু ক্ষয় হতে থাকে. It is not bank account. It is not wealth of gold and money and everything that if you go on collecting and collecting, and it will increase. And if you do not spend, it will go on increasing. But the knowledge treasure is something different. If you go on accumulating without distributing, without spending, after just few years, you will forget everything. If you go on creating and accumulating without spending, distributing anything, it will only take 2 to 3 years and then you have forgotten everything. But then if you go on spending, go on distributing, it will go on increasing and increasing and increasing. So that is the basic tenets of a teacher of education. Third one is, of course, student first. In agriculture, we say farmers first. In education, student first, and that was one of the lessons which all of us, not for us, not for the teachers, but for students, for their career, for their benefit, for their knowledge, for their publication. Nowadays, we have to make some rules and regulations that student names should be given as first author and then faculty member, second author, or something like that. But he did all of this of his own. Next one, of course, you can learn is, which is tremendously important, is self-confidence. What Dr. Datta said, that at the age of 33, he started an internationally reputed journal, the UK. That confidence we certainly need to learn from the life of Professor Sharma. And then, of course, if you are a good teacher, you need to learn throughout your life. You cannot stop. If you stop learning today, then tomorrow you are going to be less knowledgeable, and then next day, and then after, as I said, after a few years, you are again back to zero. You do not know anything. So lifelong learning is something which all of us should learn. And then, of course, last one is selflessness and philanthropy. At the end of the day, if we do not contribute in this way, without anything for yourself, everything is for the society. Very difficult to do. Very, very difficult to follow. But then that is probably one which can make you immortal. There are very few scientists whom I have seen or I have heard about who do this, like what Professor Sharma did. All of you know his philanthropy, donated everything. I know another doyen of Indian science, Dr. B.P. Pal, was our first Director General. He had a large property in Delhi, in Simla, in many other places. And he donated each and every inch of his land and properties in the name of Indian Agriculture Research Institute. There could be many other examples, but Professor Sharma is certainly one of those tall and immutable examples. So friends, today indeed, it's a great day when we are celebrating a life which was fully committed to science, a life which was fully committed to education, a life which was fully committed to the students, and of course, a life which was fully committed to the society. That was the life of Professor Sharma. A life dedicated to science. Let us all try to evaluate some of those things. Spirit of science has always been important. Even today it is more important. It is not that one society has been taught about science and the society will continue following it or not. It has to be pursued by every generation. Because without science, society cannot prosper. Whatever the society, whatever country could be, without science, without new technologies, without modern knowledge, you cannot prosper. But many times, as I said, society tries to ignore that. We have to promote this. And that was the message and life of Professor Sharma. With this, I pay my very respectful tribute and homage to this great man in this department who has spent, as Dr. Datta said, more than 50 years or so. And I'm really fortunate to be here on this particular occasion. Thank you very much, all of you, for organizing it, and I wish that the life of Professor Sharma continues. Thank you very much.
Thank you so much, Professor Pathak. We are all mesmerized by your words. And this especially goes out to the students and the research scholars of this department. I hope you understand what kind of legacy. So it's your duty to bear this legacy and carry it forward. So may the spirit of science live and prosper in this department, because our lighthouse is Professor A.K. Sharma. Can I now request our head, Department of Botany, Professor Rita Kundu, to deliver the customary vote of thanks. You know me.
Dear Dr. Pathak and distinguished guests, on behalf of the Department of Botany, University of Calcutta, and Alumni Association, I extend my deepest gratitude to each and every one of you for your invaluable presence and participation in today's seminar. It has been an enlightening and enriching experience for all of us. First and foremost, I would like to express our sincere appreciation to all our esteemed speakers. Thank you, Dr. Himanshu Pathak, DG ICAR, for gracing this occasion. Thank you, sir, for coming. Thank you, Dr. Rajiv Varshney, Director, Western Australian State Agricultural Biotechnology Centre, Dr. Anindra Bandyopadhyay, Principal Scientist, CIMIT, Dr. Shujoy Rukshit, Director, IIAB, and Dr. Gaurango Gaur, Director, ICAR-INDIAN. Despite your busy schedule, you have accepted our invitation and agreed to come to our department to pay homage to our revered sir, Professor A.K. Sharma. Thank you again. Sorry. Your insightful deliberations will illuminate our minds with valuable perspectives on structural and functional genomics in plants. Your expertise and passion will truly make this seminar a resounding success. I also want to thank our guest of honor, Professor Parthaprati Majumdar. Thank you, sir, for coming and sharing your thoughts with us. I should like to express a sincere gratitude to our Honorable Vice Chancellor and Registrar for their wholehearted support to organize this seminar. I would also like to extend our gratitude to the members of the audience. I do hope that your active engagement and enthusiastic participation will enrich the discussions and contribute to the overall success of the event. It is your presence and involvement that have made this seminar truly meaningful. Additionally, I'd like to extend a special word of thanks to ICAR-Indian Institute of Agricultural Biotechnology, RANCHI, for their generous financial assistance and support. A special word of thanks is also due to ICAR-INDIAN for the technical assistance. Your contribution has been instrumental in making this seminar possible. And we are deeply grateful for your commitment to advancing plant science. I would also like to thank Mr. Akash Mondal for the live streaming of this symposium. Last but not the least, I'd like to thank our dedicated team of volunteers, our students, research scholars, and my junior colleagues, Dr. Momita Bandyopadhyay, Dr. Sudipta Roy, and Dr. Shoumitra Bal, who have worked tirelessly behind the scene to ensure the smooth execution of this event. Your hard work and dedication have not gone unnoticed, and we are immensely grateful for your efforts. Thanks are due to our engineering wing and our non-teaching staffs for their active roles in making the event successful. In closing, I'd like to reiterate my heartfelt gratitude to everyone who has played a part in making this seminar a great success. Your support, collaboration, and enthusiasm have truly made a difference, and we look forward to future opportunities to continue our journey of learning and growth together. Thank you once again, and we hope to see you at our future events also. Thank you very much.
Thank you very much, Professor Kundu. That ends our inaugural session. We take a 15 minutes break for tea. If you will please step out of the room, tea is being served there. And we will meet after 15 minutes to continue with the scientific session. Thank you very much.
Presentation.
Soumitra, দেখছি ঠিক আছে আমি
নাকি এক্ষুনি আছে না লাঞ্চের পর না এখন তাহলে পরে একটা লাঞ্চের পরে পরে আসবে
things with him, and he is disseminating the knowledge to all of us. Thank you, Professor Pathak. That's such a opportunity for us to listen to you today in this hall. He's the fellow of all academia, national academia. I think FNA, FAS, FNASC, FNAS, FAVH, FASCT, etc., and many other things. Everybody like to convert him PhD or honorary PhD degree and etc. That's just now, but that I would consider that is not everything what he is. But I would elaborate a few of them today to you. The Secretary, dear. Government of India and Director General of Indian Council of Agricultural Research. The organization today food surplus because of ICAR and State Agriculture University. Today we are in a in a good shape because of significant contribution of ICAR, and he is the Director General of ICAR. Head of 105 or more institutions and many other research organizations. So it is such a privilege to have him with us today. His specialization is soil science, climate change, abiotic stress management. That's is written here. I don't know, but I think his specialization today is everything on agriculture overall. 30 years plus of experience and education. We already know that he did BSc agriculture from Banaras Hindu University. Many of you don't know the Banaras Hindu University has agriculture. They do have, and MSc Indian Agriculture Research Institutions, that is IRI, PhD also IRI in New Delhi. While you know that P.N. Bhaduri went there, and where Shaminathan joined also in the department of named cytogenetics. Later on, it becomes a different department, but cytogenetics. So there is a linkage or linked, I find, in these people's their own career. Postdoctoral experience and training is University of Essex, United Kingdom, 1996-97. Institute of Meteorology and Climate Research, Germany, 2004 and 5. Management Development Program, NORM, Hyderabad. Executive Development Program, etc., etc. Professional history, he was, before joining as a DG, he was Director NRRI, Kotak, also Director, National Institute of Abiotic Stress Management, Baramati, Maharashtra. And international experience that he is a Humboldt Fellow. Some of you know that DAD and Humboldt Fellowship is a very prestigious fellowship that Germany offered, and he is one of the fellows of that. Visiting Scientist, International Rice Research Institute, Philippines, 1999 to 2001. Visiting Scientist, CSIRO, Australia, so Australia, Philippines, Germany. So all the famous institutions he worked and successfully worked, and that gives the evidence of his outstanding publications that he has. He also was cast fellow, University of Essex, UK. Publication, he has more than 200 research publications and he is ranked 365 and number 4 in rank of entire country's plant scientist and agronomy division. Fellowship, as I already mentioned, and national awards and recognition, all the national awards that prestigious national awards available in ICAR, he got all of them. Just a few I mentioned. He got Rafi Ahmed Kidwai Award, Indian Council of Agriculture Research, 2019. Dr. N.S. Nandha Memorial Award, NAS, 2021. Dr. R.P. Tamban Memorial Lecture Award by the Institute of Society of Soil Science, 2021. Platinum Jubilee Award Lecture, Indian Science Congress Association, 2018. He is a member of many academic and bodies and societies and etc., etc. But his, as I mentioned, his H index or citation, if you are familiar with that one of the highest in his subject and in agriculture. So It's not only that he is successful in administration, that he is doing so well. Now everyone would be so happy that they are working in ICAR because of on the top of the leadership is Professor Himanshu Patel. But as a scientist, he has done tremendous contribution and significant publication that he has under his name and in the areas of agriculture. Now the audience waiting for you, Professor Patel. Thank you very much.
Thank you very much, Dr. Datta. All very distinguished faculty members, scientists, my colleagues from ICAR, and of course, probably most importantly today, again, all the students who have joined. Dr. Datta সবকিছুই বললেন, কিন্তু মেন পরিচয়টা মনে হচ্ছে আমাকেই বলতে হবে। সেটা হলো আমার বাড়ি পুরুলিয়া। এই বাংলায় হাই স্কুল 10+2 পর্যন্ত আমি এখানে ওই বাংলা মিডিয়ামেই পড়েছি, তাই ইংরেজি বলতে বলতে কিছু কিছু ভুল হবে এবং বলতে বলতে হয়তো বাংলা বললে আমি ওই পুরুলিয়ার বাংলাতেই হয়তো একাত্রা বলে ফেলবো আর কি। দ্যাটস রাইট, ইয়েস। আপনারা বুঝতে পারবেন কিনা আমি জানি না। এবং যেহেতু বাড়ি পুরুলিয়াতে, ফলে ক্লাইমেটের সাথে আমার ডাইরেক্ট কানেকশন আছে, নট ইন এ গুড সেন্স। পুরুলিয়া হচ্ছে এমন একটা জেলা বা ওই পুরো অঞ্চলটা, পুরুলিয়া, বাঁকুড়া, বীরভূম, মেদিনীপুরের কিছু পাহাড়, যেখানে খরা, drought is almost a regular feature. ফলে ক্লাইমেট বা ক্লাইমেট চেঞ্জ ওই রিজনের লোককে পড়তে হয় না, ওই রিজনের লোককে ভাবতে হয় না, they feel it, they live with this, and that's my connection with this topic. যেহেতু পুরোপুরি এগ্রিকালচার ব্যাকগ্রাউন্ড থেকে, আমার আগে আমার ফ্যামিলিতে কেউই কোনদিন গ্রাম ছেড়ে বাইরে যান, পুরোপুরি এগ্রিকালচার থেকে। ফলে এগ্রিকালচার এবং ক্লাইমেট চেঞ্জ পড়াশোনা করে হয়তো একাত্রা সেন্টেন্স অ্যাড হয়েছে, কিন্তু বেসিক নলেজ ওখান থেকেই আছে। So I'm fortunate that Dr. Datta asked me to talk on this particular subject. And again, agriculture, IARI কথা বললেন উনি, Indian Agriculture Research Institute. আমরা হিন্দিতে বলি, মানে হিন্দিতে ভারতীয় কৃষিকে আধার ICAR, মানে ভারতীয় কৃষির আধার যেটা হলো সেটা হলো কিন্তু ICAR। যারা ইয়াং জেনারেশন তারা হয়তো জানেন না, হয়তো নিঃসন্দেহেই জানেন না আর কি, যে এই দেশ ইভেন আপ টু 1970 used to live, which we call food to ship to mouth. Large amount of food we used to import, most of it donation under PL 480 program. It will all be coming from US, ship after ship after ship. That will be distributed to each and every corner of this country. And then the stomach of these Indians will be filled. But that was up to 1970, or maybe little plus minus 2, 3, 4 years. But today, then let me stand here. Watch. Okay. But today, this country not only provides enough food to all its citizens, it gives food free of cost, free food to 60% of its population. And then again donates food free of cost to at least 10 to 12 countries of the world. করোনার সময় 60 to 70% পপুলেশনকে এই ভারত ফ্রি ফুড দিয়েছে আমাদের দেশে, এবং কমপক্ষে 12টা দেশকে ফ্রি ফুড ডোনেট করেছে। Now all these developments were because of development in agriculture. একবার দত্তই আমাকে বলেছিলেন আমরা এখানে চাল বিক্রি করি 2 টাকা পার কেজি, গম 3 টাকা পার কেজি, মিলেট 1 টাকা পার কেজি। এখানে ইনসিডেন্টলি আমার ভাই বসে আছে, ও ওয়েস্ট বেঙ্গল গভর্নমেন্টের ফুড ডিস্ট্রিবিউশন দেখে। ও ডব্লিউবিসিএস অফিসার, ওরও শোনা দরকার আর কি। উনি আমাকে একদিন বলছিলেন গল্প করতে করতে যে ওই যে 2 টাকা বা 1 টাকা বা 3 টাকা নেই, যে লোকটাকে ওখানে ডিস্ট্রিবিউট করার জন্য রাখা হয় তার স্যালারিও কিন্তু পুরো দিনে ওই ধানের বা গমের দামের থেকে বেশি। এখন তো পুরোটাই ফ্রি হয়ে গেছে। ভেরি গুড, দ্যাটস গুড। Food Bill Act of 2013 and then of course subsequent. এই এগ্রিকালচারটা কিন্তু বটানির সাথে ডাইরেক্টলি কানেক্ট। IRI যার কথা আপনারা সকালেও শুনেছেন, যেখানে ডক্টর PN ভাদুড়ি এখান থেকে IRI গেছিলেন। IRI শুরু হয়েছিল 1905-এ, 1905-এ বিহারের পুসাতে। তাই এখনো দিল্লির ইনস্টিটিউট এখনো পুসা ইনস্টিটিউট নামেই পরিচিত। এবং প্রথমে কিন্তু মাত্র 4টে ডিপার্টমেন্ট ছিল, আজকে ওখানে 24টা ডিপার্টমেন্ট আছে। তার মধ্যে একটা ডিপার্টমেন্ট ছিল ডিপার্টমেন্ট অফ বটানি। So that time agriculture was not a separate subject. It was Department of Botany, যেখান থেকে ওই সমস্ত বড় বড় যাদের নাম আপনারা শুনলেন তারা ওখান থেকে পড়াশোনা করেছেন, ডিগ্রি পেয়েছে। তাদের মধ্যে একজন হচ্ছেন ভারতরত্ন প্রফেসর এম এস স্বামীনাথন। তাই যদি স্টুডেন্টদেরকে বলতে হয় তাহলে বটানি যেটা অ্যাপ্লাইড পার্ট, বটানি রিসার্চ করে যেটা ফার্মারদেরকে বা দেশের জনগণকে কিছু দেওয়া যেতে পারে, সেই পার্টটা কিন্তু এগ্রিকালচার। আর এগ্রিকালচার এই দেশের জন্য কেন, পুরো বিশ্বের জন্য নতুন কিছু নয়। আমরা বলি সিস্টেমেটিক এগ্রিকালচার শুরু হয়েছিল অ্যারাউন্ড 10,000 ইয়ার্স ব্যাক, তার আগেও কিছু ছিল, কিন্তু সেই দিনের এগ্রিকালচার এবং আজকের এগ্রিকালচার ইজ আর নাও ডিফারেন্ট। আজকের অনেক নতুন টেকনোলজি, হেল্ডিং ভ্যারাইটি থেকে শুরু করে নাও লটস অফ থিংস হবে, লটস অফ থিংস, বাট দ্য প্রসেস ইজ স্টিল দিস। এবার এই স্লাইডটা দেখে মনে করবেন এটা তো আমাদের সাবজেক্ট, এটা তো বটনির সাবজেক্ট, এটা তো ফটোসিনথেসিস, যেখানে ওই ক্রপ, অ্যানিমেল, হিউম্যান, ফিশারিজ, ফরেস্ট্রি, এয়ার, ওয়াটার, সয়েল এই সব কিছু মিলে with that unique process of photosynthesis. And that is the only process which we know which can produce food. And all of you are master of that process. And in agriculture, we feel we are the master of that process. I don't know who is master, but that's a common process for all of us. And this is the interconnectedness. This is the interconnectedness. And when we say not one, not soil or water or air alone, not only solar radiation, but this connection, which now we say one health, one family, one earth, one future. আর বাংলায় বলতে গেলে জল, জমি, জলবায়ু, জঙ্গল, জানোয়ার, জীব, জীবন, জনতা, জগৎ, জীবিকা, all of these are interconnected. সব যদি ইচ্ছে করে লেখা আর কি। All of these are interconnected. All of these are connected to agriculture. All of these are connected to botany, because that photosynthesis and this whole plant processes. And climate is so important. Even today, what we say that if climate goes wrong, agriculture cannot be right, because we are so dependent. Even the almost 100% irrigated areas of this country, or else, say Punjab has got almost 99 to 100% irrigated area. Haryana has got around 95%. Here it is still less. But even in those areas, if climate goes wrong, agriculture cannot be right. And if agriculture goes wrong, then for this country, and particularly for this state, nothing can go right. And that's what Professor Swaminathan used to say. That if agriculture goes wrong, nothing can go right. Therefore, the first Prime Minister of this country, Pandit Jawaharlal Nehru, who said that everything else can wait, but not agriculture. Agriculture cannot wait. And this is the connection of climate with agriculture. But the unfortunate part is that climate, which is the major driver of crop production, which is the major determinant of agriculture, is now changing. Changing for worse. Changing adversely. It is widespread. It is intensifying. And we used to call that, okay, 1.5 degrees centigrade temperature should not be crossed till the end of the century. But now it is crossed almost. Rainfall variability and intensity increasing. Total amount of rain might not have changed. Rather, it has increased. And theoretically also it will increase. But then for agriculture, total amount of rain has got almost no meaning. It is the distribution of rain which is most important. And who can know better than me? পুরুলিয়া যেখানে সব থেকে আমাদের খরাপ্রবণ জেলা, সেখানেও কিন্তু বৃষ্টি হয় 1250-1300 মিলিমিটার। পাঞ্জাবে বৃষ্টি হয় 650 থেকে 700 মিলিমিটার। But then Punjab never bothers about rice yield or wheat yield. And in Purulia, Bankura, in every season, you cannot be sure till you harvest the all uncertainties. Sea level rising, cyclones are becoming fiercer and frequent. And again, probably West Bengal থেকে এটা বেশি কেউ জানবে না। Almost every আগে এটা 3 বছর 4 বছর 5 বছর পর পর একবার করে হতো, এখন তো not once. It could be even একটাই আমরা দেখলাম আর কি, তার মানে এই বছর আর আসবে না। Who knows? And the projection is all of this: cyclone, heavy rain, heat wave, flood, drought. All of these are increasing in frequency as well as in intensity. Both number as well as intensity. All of these are increasing. Of course, we are studying climate. And not only agriculture, there are many other departments also studies. So we have tried to understand which district is more vulnerable, high risk, low risk, medium risk, and so on. But in summary, what we can learn few things. Number one is minimum temperature is rising more than the maximum temperature. And for photosynthesis, this is most important. Minimum temperature, night temperature. Temperature rise is more in Rabi season than in Kharif season. এই ওয়েস্ট বেঙ্গলে কিছু জায়গায় উইন্টার সিজন কখন আসে এবং কখন চলে যায় আর হিসেব মনে হয় আপনারা রাখতে পারেন না। দিল্লিতে এখনো কিন্তু যেমন স্যার বেশ ভালো কোট ফোট হেরিফেরি পরেই থাকেন, শীতকাল গ্রীষ্মকালও পরে থাকেন। ওয়েস্ট বেঙ্গলে মুশকিল আছে। ইট ইজ অলমোস্ট গোয়িং। কিছুদিন আগে আমি একবার সুন্দরবন আমাদের একটা প্রজেক্ট চলতো, ওখানে একজন হাই স্কুলের টিচার খুব ভালো একটা কথা বলেছিলেন, সেটা আমি এখনো মনে রাখি, আমার স্টুডেন্টদেরকে বলি। উনি বললেন, আমরা এখনো কিন্তু ক্লাসে গিয়ে পড়াই, এখানে 6টা ঋতু আছে: গ্রীষ্ম, বর্ষা, শরৎ, হেমন্ত, শীত, বসন্ত। বসন্তের গান টান আপনার নিঃসন্দেহে অনেক কিছু, কিন্তু উনি বললেন এখন কিন্তু অ্যাকচুয়ালি 3টে সিজন আমি দেখতে পাই। ওই বসন্ত হেমন্ত কখন আসে কখন যায় তার হিসেব মেট্রোলজিস্টরাও বলতে পারবে না, কোথায় শেষ হয়ে গেছে। এবং উনি বলেন আজ থেকে 10-12 বছর পরে কিন্তু 3টেও থাকবে না, 2টো সিজন থাকবে। And he was very sure about it. বললাম কি সিজন থাকবে? বললেন একটা হচ্ছে গ্রীষ্ম, আরেকটা অতি গ্রীষ্ম। বাকি এই সমস্ত কোথায় কবে আসবে কবে যাবে কেউ তার হিসেব পাবে না খুঁজে, এই 2টো সিজনই থাকবে। এবং I thought very right. Even today This is fantastic. But this year, the whole country is experiencing probably one of the hottest summers. Delhi crossed 50. Here also 45, 46. Many places crossed 52, 53. Now, these are something which are totally unheard of. But now those things are happening. Rainfall, as I said, rising intensity and variability, reducing duration and number of days, the distribution of rainfall, which is most crucial. And then, of course, increasing extreme events, unseasonal rains, drought, floods, and such. All of these have direct impact on agriculture. Again, some of the basic things, because of changing climate, the risk for the whole food system, crop, animal, fishery, and so on, all are increasing. The impact will come from temperature rise, weather hazards like cyclone and so on, sea level rise. Again, Sundarban is one of the most vulnerable areas globally, not only in this country. Of course, drought, flood, I talked about. But these four are almost known for everyone. But abiotic and biotic stresses, which are not so visible all the time, but those are operating almost on a daily basis on crops, on animals, on fisheries. On a daily basis, these are operating. We did some modeling studies that what will happen in future. So what we observed that if it goes like this, then in the year of 2015 is 2040 to 2050, wheat yield will reduce by 9%, irrigated rice by 12%, kharif maize 18%, soybean and other crops some. But then there will be few crops which might be benefited because of increase in temperature. In colder region, it might increase. Milk yield loss will be around 1.8 million tons per year. Horticultural crops, in terms of quality and so on, pest and disease incidents, biotic stresses will increase. But then there will be few positives. Few positives if you can harness the positive aspects. As I said, total amount of rain will be still more. It is not going to be less. But do you have that reservoir to capture or to harvest the excess rain? If you do not have, then you are going to be losers. So there are several other things. Elevated CO2 will increase yield. But can you harvest that? So these are a few of the aspects. Now, all of you know why this global temperature is increasing. This is because of some gases like carbon dioxide, methane, and nitrous oxide. We call them greenhouse gases. I shall not go into details of those, but these some of the numbers which were given by IPCC, Intergovernmental Panel on Climate Change, that CO2 concentration currently is the highest in 2 million years. Sea level rise is fastest in 3,000 years. Arctic sea ice area is lowest in last 1,000 years. Glacier retreat, again unprecedented, in last 2,000 years. So we are really passing through a very, very different kinds of world, very, very different kinds of climate. And these are all because of emission of greenhouse gases. In 2023, last year, the world emitted around 55 billion tons, not million tons, billion tons of carbon dioxide equivalent, including methane, nitrous oxide, and so on. Agriculture contributes around 10 to 12%. But if you include forestry also, agriculture, forestry, and other land use, then it is around 25%. That means 1/4 of the cause is directly or indirectly from agriculture. Greenhouse gas load in the atmosphere is building at a rate of 2.4 parts per million per year. And India contributes only 5% of all these GHG emissions, global GHG emissions. But then there is another aspect of looking at these numbers. Globally, 10% of the richest countries, they contribute 50% of the greenhouse gases. And 50% of the poorest countries, they contribute 10% of the greenhouse gases. So richest 10% contributing 50%, poorest 50% contributing to 10%, but the impact will be more on the poorest people because they do not have adaptive capacity. They do not have resources. They do not have technologies. And they are going to suffer. And of course, all of you know that prediction is countries like Maldives and many others might not be there. Many other coastal regions are also going to be underwater. And almost on a daily basis, we are experiencing those. But threat is not only climate change. These are additional threats. Decreasing water availability, degraded soil, increasing pest and disease, huge loss of food. I mean, whatever we produce, we lost around 1/3 of that food which we produce globally. And then, of course, over and above, intensifying climate change. So all of these are threats to Indian agriculture and also global agriculture. So with this background, now we are venturing into a journey called Viksit Bharat, where the requirement, I shall not read the numbers. You can see what it is today, 2023, and how much do we need in 2047. Now, how this is possible? And again, let me remind you, in this country, in spite of all these developments in other sectors, this country cannot become a developed country without developing its agriculture. That's the prerequisite. When today also 44 or even 50% of population depends on agriculture. Though its contribution to GDP is around 18 to 19%, but still 50% is here. So leaving this 50%, certainly the rest of the 50% cannot be developed. These are the numbers. So the question is, what is our vision then? This is the vision. De-risking agri-food system from climatic risk. Now, how this is possible? Basic point is science and technology. If de-risking is possible, it will be possible only by science. And that's why in the morning I was talking more and more about science, scientific spirit, and so on. Without this, no. कोई magic नहीं हो सकता. It has to be science. And of course, there's some systematic science which can be done for de-risking agriculture. We call it foresee, adapt, respond, mitigate, and build capacity. Farm building, FARM, and then building. This is what we need to. It is possible. আমি হয়তো আগে কিছু ভয় খাওয়ালাম আপনাদেরকে যে কি করে হবে, কেন হবে, হবে না. But then it is possible. And we have seen it is possible. Because we are working on climate change aspects for last around 25-30 years. There are many options by which even the technologies which are existing today, even without any new technology, the yield loss which is expected or which is happening because of climate change can be compensated. For example, if you do not do anything, then of course we are going to lose around 15 to 20% of yield. But if we go on adopting some of these technologies, including new varieties, management, and so on, then not only this 20% yield loss can be compensated, but it can be even increased. But the point is, it will have some cost. That cost will be quite heavy for small and marginal. Who will bear that cost? Is there any mechanism by government? Is there any some kind of philanthropy from big organizations, industries to bear that cost? Or whether global community is responsible and ready to bear this particular cost? And if you can do that, then all these red areas in the left side map can be converted into green areas. So these rates can be shifted to green if we go on adopting the technologies which are even available today without waiting for new technologies to come out. In Indian agriculture, we have observed that. Even 15, 20 years back, if there was any abnormal year in terms of rainfall, then the production would have been drastically reduced. You can see 2001, 2, all these red lines. These are less rainfall years. But then currently you will see that even in these years, the rainfall was less, but total production of food grain in this country was not reduced. It's still either maintained or increased. Because of this, these are because of technologies. This year, wheat production is one of the probably very prominent examples. Currently, in Punjab, Haryana, Western Uttar Pradesh, and some parts of Madhya Pradesh, around 70% varieties of wheat which are being grown are climate resilient varieties. And of course, CIMIT is here. And because of those climate resilient varieties, which are covering around 70% of the area, wheat yield this time was still record, though there was terminal heat and all other kinds of things. But the yield didn't go to the extent which was people were expecting to go down. And that was same for the last year. So this is the strength of science, how new technologies can maintain or increase the productivity and production. And of course, now we are fortunate that there are several new technologies available. One is, of course, genome editing, and I think Dr. Ananda is going to talk about it. This is not my cup of tea, so I'm not going to touch. I think Dr. Bharsne also will be talking about it. But this photograph, which I am showing on the right-hand side, is a real photograph of one of our ICAR institutes. I shall tell a little bit more about that. Then, of course, we have got other tools like AI, GIS, and all the kinds of things, policy, funding support. So there are certain opportunities now available, which will help us developing more and more climate resilient technologies, developing more and more other options. So with this background, climate change is real. It is going to hit hard to agriculture system. We need to increase the production and productivity. There is no way out. So what are the options? So very quickly, I shall suggest 10 action points for making Indian agriculture climate. First one is, of course, climate resilient varieties. And fortunately, over the last few years, ICAR has released around 2,000 varieties of different crops which are climate resilient. They are tolerant to one or more of the stresses, biotic as well as abiotic stresses. And then there are new crops coming. Probably earlier people were not knowing about this dragon fruit and all of this. Millet was ignored, but then more and more emphasis we are trying to give to these crops which are called as hardy crops or climate resilient crops. So either go for new crops or go for varieties which are resilient to climatic stresses. So those kinds of varieties. This is what. So using genome editing tools, already we have got 2 or 3 varieties in pipeline, which will increase the yield by around 20 to 25% compared to the best available variety. The duration could be reduced by another 20 days. And then, of course, several other advantages. Now, this is one of the very prominent research areas now. Not only ICAR, many other institutes. I'm sure this department is also working on some of these areas. Then, of course, precision management of soil, water, energy, and nutrient. And again, there are several tools available. But then, in some places, they're also going for what we call as farms of the future. Now, probably those who come from marginal or or some remote areas, they still might not think that a drone might be flying on a field. Or AI-based tools can be used for management of water, nutrient, and so on. But in many places, these are reality. These are reality. These are happening. And it will happen. It will happen. Of course, we had a big project called NICRA, National Initiative, now Innovation on Climate Resilient Agriculture, where we are developing, which we call as climate resilient villages. And this plan, which now we are going to announce, Government of India, the proposal is in the whole country, around 50,000 villages will be developed as climate resilient villages. ICAR could develop the model, and we have got around 400 or so, but then 250 and then some more added. So we are going to have those kinds of villages. Carbon markets, totally new. How to reduce emission of greenhouse gases, particularly carbon dioxide, and how to make some money out of it. Again, the framework is ready, and in many places, farmers have started working along with some industries, and also ICAR is participating of how to make carbon farming, which is not only to reduce emission of greenhouse gases, but also to improve soil health, reduce water use, and so on. So all these benefits are being there. Healthy and diverse foods. Not all the time rice and rice and rice, so maybe some other kinds of foods, pulses. And many of these foods are not only healthy, but also they are climate friendly. So diversification of our food habit is also very, very important there. And all of you know, 2023, we celebrated International Year of Millet. And then we hope that probably this will continue and more millets will be consumed, grown, exported from this country. And then this will be quite helpful in climate resilience. Reduction in food loss anyway will be very, very crucial. Losing 1/3 of food is something which cannot be accepted. And all of us can contribute in this. Each one of us can contribute. All of us do some kind of. But then not only modern science. Some of the traditional things also we need to inculcate. There are many good traditions. Even for science also, Indian science is also quite old. It's not that we are having science only just in a few years back. So integration of tradition as well as modern technologies, including varieties and all automation and everything, if we can combine them together, we are going to be benefited. But the crux of all of these technologies and to take all those to its logical conclusion, reaching to farmers, will be human resource development, all the students, all our younger friends. That part is very, very crucial. And science education, in spirit of science here, is going to be extremely, extremely important. At the same time, agricultural extension with new tools and technologies. We call it digital agriculture. All of this can also be putting its role. But then it is not going to be the job or responsibility of only ICAR or one or two organizations. It has to be a big partnership with industry, with society, with policymakers, with state, central, and all of us. And that's what is required. Fortunately, over the last few years, maybe 10, 12 years, and I must thank Dr. Datta, it is not because that he is sitting here. When he was Deputy Director General in ICAR, lots of new facilities we could create to study climate change. Facilities included all our PACE, PAGE, phenomics, and all kinds of tools and technologies. I'm not going to describe each and every one, all of you can study, but these are the tools which are so useful, so important for studying the impact of climate change, the benefits of adaptation tools, how to measure greenhouse gas emission, how to reduce emission of greenhouse gases, and so on. So fortunately, we have got a good infrastructure in place in ICAR and some other places. But then it is not only crop alone. As I said in the beginning, Jal, Jeev, Jivan, Jalabau, all of these are all tied in one. So it has to be integration of all of these. starting from crop to animal to water to precision management and all of this. And that's what will bring what we call as climate resilient. That's what is needed. Improved forecasting, contingency plans are again part of this whole process. We are fortunate that in meteorology and forecasting, lots of improvements have happened. Lots of improvement. There are still several things to do, and we are having a quite large project on this particular aspect. But then, today I've got one or two more slides, but this is a slide which I generally show of how Indian agriculture or Indian development should be different from the development trajectory of developed countries. What I say that we should not be following exactly what the developed countries have done. In many cases, we follow that, and there is nothing wrong in that. But probably for Indian agriculture, we really need to follow different. See, all these developed countries of today, starting from US to Europe to Australia, around 30, 40 years back, and particularly after World War II, for sure, their economy was even worse than what our economy today is. Much worse. But then all of this could improve themselves from developing countries to developed countries. All of this. Now they are developed countries. But after becoming developed countries, each one of those feel and experience that their development is certainly not which we call as climate-friendly development, nature-friendly development, or environment-friendly development. Their development is not sustainable development. Because they are also now facing enormous problems. It is not that we are facing, they are also facing. But somehow they have got resources, they have got technology, so they will be able to manage for some time, but not for so long. So after becoming developed country, now they also need to go from this point to become climate friendly or environment friendly or nature friendly developed country. We are still here. So we should not be following like this. Let us go to this point, and from there, we shall go to that. In the very simple logic of geometry, we should be following this path like this. Not go there, let's go like this. Because in geometry, all of us know that the length of this side of the triangle is always less than the sum of the other two sides. So we should be following this trajectory, not the other. And what is this trajectory? This is the trajectory of low carbon, low water, low nitrogen, low energy, and so on. Can we harvest such huge yield with low carbon, low nitrogen, low water? Yes. If you can increase the efficiency, if you can really take care of soil health, if you can really follow some modern tools and technologies, you can produce more or less similar yield with much less of water, much less of nutrient, much less of energy, much less of nitrogen. And we really need to do. We need to do those for ourselves and also the commitments which we have given to international communities. So we have got promises to. What are those promises? These promises we call as Panchamrit. So by 2030, we will be achieving this, and by 2070, we will be attaining which we call as net zero. Net zero means whatever we will be emitting, we will be also accumulating or absorbing the same amount. So the net is going to be zero. That's what we really need to do. Now, how Indian agriculture can contribute to fulfill these commitments? Can Indian agriculture attain net zero emission or carbon neutrality? This is something on which all of us need to. But the examples which I gave, including adaptation, new varieties, precision agriculture, extension, digital agriculture, and so on, including our traditional tools and techniques, we are sure that we will be able to keep these promises and we will be able to produce what is required by 2047. This is possible. And that is the vision. How to do that? Why to do that? That is the vision of climate resilience. So friends, to summarize, intensifying global challenges, not only climate, but many others I showed. We need to transform agri research. Botany is a part of that, as I mentioned in the beginning. Farming the farms of the future, not the way we have been doing in last 20 or other 50 years. We really need to change that. Building capacity, education, skill, training, and so on. And of course, partnership is very, very crucial with all of us, industry, society, scientists, and everyone of us. And that's what we really need. With this, thank you very much for your kind attention and giving me this opportunity. Thank you.
The words are actually not enough to thank Professor Himanshu Pandey for his wonderful, wonderful lecture. I'm really thankful on behalf of our department and Alumni Association and all the audience. We are a little short of time, so without any more ado, I will move on to the next lecture. So the next, we will now listen to Professor Rajiv Varshney. Just a second. So he's been with us since 10 a.m. in the morning, and he's waiting to speak, and we are also waiting to listen to him. I would request Professor Rita Kundu, Head Department of Botany, to introduce Professor Rajiv Varshney.
Can I request to share my screen or allow screen sharing, please?
So, thank you, Momita. Now I'm going to introduce Professor Rajiv Varshney. Professor Varshney, FRS, is an agricultural research scientist specializing in genomics and molecular breeding with more than 20 years of service in international agriculture while working in India, Germany, Australia, Mexico, and several countries in Africa. At present, he is serving Murdoch University, Australia, as a director, Centre for Crop and Food Innovation, director, State Agricultural Biotechnology Centre, and international chair in Agriculture and Food Security with the Four Futures Institute. He is an adjunct professor with more than 10 universities in China, Australia, Africa, and India. Professor Varshney is a globally recognized leader for his work on genome sequencing, cataloging, and utilizing genetic diversity, genomics-assisted breeding, seed systems, and capacity building in developing countries. He has made centrally important contributions towards improving food and nutrition security in India and several countries in Africa and Asia by assembling genomics, developing genomic resources, and integrating genomic technologies in crop improvement programs in many tropical crops and delivering several superior crop varieties to some of the world's poorest farmers. For instance, he had decoded genomes of more than 15 crops and delivered more than 20 legume crop varieties through genome-assisted breeding in many countries. Professor Varshney is a highly prolific author with 23 papers in Nature Journal and Clarivate highly cited researcher for 19 consecutive years in a row since 2014. He is the only agricultural scientist in India and the only translational plant biologist in Australia to achieve an H-index of more than 100 as per Google Scholar. At present, his H-index is 129 and citations are more than 70,000. Professor Varshney is the fourth Indian agricultural research scientist to receive fellowship of the Royal Society. In addition, he is an elected fellow to about 10 science and agricultural academics society in India, Germany, USA, Africa. That is a rare feat for any scientist. His research work has been recognized with more than 30 prestigious awards from USA, Germany, China, India, Nepal, Vietnam, Philippines, UAE, and many other countries in Africa, including the most coveted science award, Shanti Swarup Bhatnagar Prize, and the most prestigious agricultural science award, Rafi Ahmed Kidwai Award from Government of India Crop Science Research Award and International Crop Science Award from USA. Huilu Friendship Award, Science and Technology Partnership Award from China. Professor Varshney has been named as the top 100 scientist from Asia by the Asian Scientist magazine in 2022, among 25 global researchers in biotechnology by Nature Biotechnology in 2021. So this is all about Professor Varshney's field. Now, I would like to request Professor Varshney to deliver his talk. Thank you.
This is our sir. Yes, yes.
Sir, please unmute yourself.
Okay, can you hear me?
Yes, sir.
Okay, okay. Very good. Thank you very much. Very good afternoon, everybody. And let me thank Professor Rita Kundu for very nice introduction. And I would like to avail this opportunity to thank Professor Swapan Datta, who has been my mentor and well-wisher for a long time. I had a privilege to work with him on several projects as well. So thank you very much, sir, for giving me this opportunity. I'm very much grateful to see many stalwarts.
Hello?
Can you see? Yes, can you hear me?
Sir, please close to your mic.
Oh, so you were not able to hear me? Okay, now how about that? No?
Yes, sir.
Can you hear me now or not?
Sir, can you speak louder little bit?
So is it audible or not yet? I'm just sitting on that my laptop, so I don't think that I can go much more closer than this one. It's not possible. Sorry about that. So are you able to hear me? Can somebody say something, please? Or they're trying to fix the problem? Or you cannot hear me at all?
It's all right, sir.
Can you hear me now?
Yes, yes.
Okay, good. And you can see my slide as well? My slides are also visible, sir?
Yes, 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 sir.
Okay, okay, very good. So let me try from beginning again. So good afternoon, everybody. And let me thank Madam Professor Rita Kundu for very nice introduction, and also my sincere thanks to Professor Swapan Datta, who has been my mentor and well-wisher for a long time. I had a privilege to work with him on several research projects about which I will also mention in this presentation. It's also great to see Dr. Himanshu Pathak, Director General of ICAR and Secretary there, and listening his fantastic presentation. And also in morning, Dr. Partha Majumdar, and also here Professor Swapan Datta. So this is great. And I'm aware that there are many other senior colleagues and friends, including Dr. Soja Rakshay, Dr. Nandya, Bandhu Padhyay, and many other colleagues are present in that room. So this is very exciting. Let me offer my apologies that I am not able to come in person, though I was very much looking forward for this presentation and joining you. But I feel really privileged and honored to be any speaker in this international symposium celebrating birth centenary celebration of Professor A.K. Sharma. As other senior colleagues, stalwarts mentioned in earlier sessions, that we all are aware about the great doing of plant science, plant science of, I don't know what is happening here, in India. But though I never had a privilege to meet Professor A.K. Sharma, but when I was doing my PhD with Professor P.K. Gupta at Meerut University, and Professor P.K. Gupta is also cytogeneticist like Professor A.K. Sharma, I'm aware with many stories, including one of the book of Professor A.K. Sharma about the chromosome theory and practices. So I'm very much aware about his name and contribution. I also put this screenshot of one of my presentation from 2022, where I had an honor to deliver Professor Archana Sharma and Professor A.K. Sharma oration lecture, where at that time point I delivered a talk on genomic interventions for crop improvement. So let me pay my respectful tributes and homage to Professor A.K. Sharma, who is the doyen of botany in general and cytogenetics and cytotaxonomy in particular. And we know that his contribution, so I'm not going to highlight his contribution again, but one key thing is that chromosome techniques, theory and practice, the book, which I have put this thing here, and also the nucleus. I think these are the legendary, or they are the legacy of Professor A.K. Sharma, which many of us are continue to advance further. So I would also like to, so he has done great work on those chromosomes. And I think that many of us who are working in the genomic science, we are going much more deeper in chromosomes in that, at that structural level of the DNA, and to understand the genome architecture and functions for crop improvement. But why we are doing this thing? Because as Dr. Pathak also mentioned, many challenges about Indian agriculture, and the challenges what he mentioned about Indian agriculture, more or less they are same for the global agriculture. So we are having similar kind of challenges all around the world. And one of the key thing is overpopulation. In the context of India, we know that we already are 1.4 billion people, and by 2050, the whole world will be reaching about 10 billion. So question is that the food security. At present also, so right now we are having around 7 billion. We are adding, or will be adding another 2 to 3 billion people in by 2050. But even now, we are still having around 800 million people who are going to bed hungry. And there are several countries in the world, especially in sub-Saharan Africa, which are the places for the severe hunger. Country like India, they are doing great. So we don't have the severe issue in those countries, which is very good news. But when we talk about the nutrition, then malnutrition is also very important. Unfortunately, in our country, we still have the issue of the protein deficiency and many other things. So I think overall, we need to address the issue of the nutrition as well. And these are the challenges all around the world. And we will think that, and we all are aware about ongoing conflicts and geopolitical issues around the world. Either this is Russia-Ukraine war, or right now we have seen Israel and Palestine and many other places in the world, that they are also adding more misery towards the food security. Dr. Pathak highlighted the role about climate change, and I was so happy to see that how Indian agriculture is addressing this issue. But if you would like to see that the whole world, we need to be focusing on this particular aspect, that how we can mitigate the adverse impact of climate change on the yield. And I'm very positive, optimistic that we will be able to make it happen. This is not that, well, these are the problems now. Even we are aware about the issues of that 1960s when we had the problem of those famine, etc. And then at that time point, we know that Green Revolution has been a success story from India. And there are many heroes, including our own Professor M.S. Swaminathan, Professor Gurdev Khush, who have contributed a lot in this important area. And we all know that genetics and breeding, they really helped in the form of developing the short height varieties and thanks to RST allele, etc. So Green Revolution has been really wonderful, made wonderful impact on the food security, especially in Asia, South Asia. So as a result of the Green Revolution, the good thing is that we have now, world is producing almost 3 times what we were producing in 1961. We got the improved farming incomes. We got extensive irrigation facilities. But at the same time, we had the liberal use of the chemical inputs. And if you would like to see the impact of the fertilizers, so in 1961, if you would like to see that this started at that time point mainly in the developed worlds, but now this moved to the developing countries. The good news is that, well, the thing is that on one hand, we were able to produce more food, but at the same time, you can see the global fertilizers consumption has increased more than 4 times. And many times people say that why we do not have the Green Revolution in Africa. And not only that, just the fertilizer consumption, of course, the policies, issues, scientific and technological interventions, a lot of things. But if you would like to see from the consumption, fertilizer consumption perspective, Sub-Saharan Africa contributes about 25% arable land, but use only 2.75% of the fertilizer. Number of regions. But at the same time, if you see the whole world, and if you see the agriculture, either this is North America, Europe, or many countries in Asia, wherever you see these green color or yellow color, this is the agriculture production. In those parts of the world, we also having the higher consumption of the fertilizer. Many countries in Sub-Saharan Africa, we don't have the higher yield because we don't have much fertilizer there. So this is the directly correlated or so. This does not mean that if we need to address the issue of the crop production, we need to put more fertilizer, because we also need to think about the environmental sustainability. Dr. Pathak mentioned about the different gases. So not only carbon dioxide, but nitrous oxide is also very important gas, which contributes about 7% for the climate change. So I think we need to address the issues. Dr. Pathak mentioned very nicely that we should not be just focusing on rice or wheat or only cereal. Those cereal crops are very important crops and they will continue to be important. So we should not think that, well, we should get away from these crops. No, not at all. But what is the most important thing is we need to diversify our crops. We need to think about the legume crops. We need to think about the other core cereals, etc. For legumes, we know that the climate smart crops, they take less water and they are having less carbon footprint. So I think legumes can play a very important role in cereal intensive cropping system, including in Australia, where I'm working nowadays. So we are producing here amount of the wheat and barley, etc. But nowadays in Australian agriculture also, we are always focusing that how we need to make it sustainable. In this regard, I will give some of that success stories from our work, what I did at ICAR-INDIAN. Many of you are aware that I was working at ICAR-INDIAN, worked there for about 17 years. And I worked with a large number of ICR organizations, including Indian Institute of Pulses Research, Indian Agriculture Research Institute, Directorate of Groundnut Research, and many state agricultural universities in India with whom we collaborated in India and also around the world. And we made significant contribution in this direction. So these are the key legume crops important in India, not only in India, but in many countries in Africa as well. Chickpea, pigeon pea, and groundnut. But one of the serious issue when I joined ICAR-INDIAN way back in 2005, the crop productivity. This was about less than 1 ton per hectare. Total pulses production at that time point used to be 10 to 12 million ton, now which has reached to around 18 to 20 million ton, etc. So I mean that This is great story, thanks to policy interventions and that varieties and also farmers, a lot of players, they have played very important role. But let me see about the genetic aspects. So when we would like to talk about enhancing the crop productivity, I'm talking 2005, maybe around 20 years ago. And this was a big challenge, especially when you would like to deal these things and when you are having this yield plateau and you would like to use the modern approaches. I worked before joining ICAR-INDIAN, I was working in Germany, where I was working in the area of genomics. And in 2005, we had the rice genome sequenced. So at that time, when genome sequencing was in infancy, or this was just coming up, or there was a big boom. In 2005, together with Mark Sorrells and Andreas Gnaner, we provided a concept that how genomics can be helpful in the breeding program. So we gave a concept, framework, genomics assisted breeding. And that was published, or that was on the invitation of the Trends in Plant Science from the 10th anniversary issue. So that was my background, that well, that we can use the genomics approaches, but then we would like to translate these things in crops like chickpea, pigeon pea, and peanut, which used to be called orphan crops. We did not have much resources. So it was very challenging. So, but we wanted to address this challenge, and we are having very limited use of the genetic diversity. So to address these issues, what we did, we established many international consortium together with the partnership from the ICAR institutes, state agriculture universities, and many other partners in Asia and other countries. So as a collective efforts of these consortium, we were able to deliver the genome sequence. At the time point, genome sequencing was considered to be only remain with that advanced countries like United States or Europe, etc. But when I was in India, we demonstrated that while working in India, you can lead the genome sequencing consortium. But now the good news is that many organizations in India, they are working on this aspect. Anyway, we delivered the genomes of pigeon pea, chickpea, pulse millet. In peanut, we are having the different kind of genomes. So we did the A genome, B genome, and also not only these crops, but many other important crops, including sesame, mung bean, ajoki bean, pea, etc. And we have delivered more than genomes, more than 15 crops. And so now what I wanted to tell that due to the advances in the sequencing technology, coupled with consortium efforts, we were able to deliver the genome sequences in large number of crops. Now, the next thing is the genome sequencing, they can provide you that how many genes we are having in a crop, like in the case of chickpea, 29,000 genes, or in the case of peanut, etc. But do these genes express in all the tissues at all the times? No, they do not. So for instance, when you are having the drought, a particular set of the genes will be expressed in the specific tissues. So we need to understand their location, their time point. And for doing these things, we have developed the atlas, like atlas, as we know in the geography, that which country or which city is present in which country, which continent. The same way, by developing gene expression atlas, we can figure it out which gene will be expressed at what time point in which tissue. And when you get these kind of information, this is very helpful to devise the breeding strategies. So we developed those kind of gene expression atlas in the case of chickpea, pigeon pea, and groundnut. Furthermore, as initially we were using that short sequencing technology or small read sequencing technology. So at that time point, the genome assemblies, they were not great, but we kept on evolving ourselves with the advances in the sequencing technologies, and we kept on developing the improved version of the genome assembly. So like, for instance, in 2022, we published improved legume genomes, and then which was also helpful to understand a specific phenomena of legume biology, which is about the nitrogen fixation, that why some crops like chickpea and pigeon pea, they are having a one kind of set of the nitrogen fixation, which is not present in peanut. In peanut, you are having the different kind of thing. So this was a very exciting thing. Anyway, the point which I want to make, that we kept on evolving ourselves in terms of developing the improved genome assembly. So for instance, in the case of pulse millet, we developed platinum standard reference genome assembly. So now it is a possibility. Earlier, we used to have the scaffolds and context. Now we can have one scaffold to the whole chromosome, and we call them chromosome label genome assemblies. as we were talking about Professor A.K. Sharma's contribution in terms of the chromosome, earlier this was not possible, but nowadays this is possible to even identify the telomeres. And we are saying telomere to telomere genome assembly, T to T genome assembly. We are able to identify now centromeres as well, based on the sequencing data. So Professor Sharma, when he was doing this cytology and cytogenetics research in terms of measuring the number of chromosomes, identifying chromosomes, etc. But now, through the genome sequences, we are having the possibility to identify the telomeres, centromeres, and develop the chromosome level genome assemblies. And this was one of our paper in soybean published last year, where we have developed the gapless chromosomes and together with all those centromere and telomere assemblies, etc. So this is about the genome assemblies. And the next thing is about that whether one genome alone is good enough to understand each and everything. Probably not, because if you sequence the cultivar 1, not cultivar 2, sometimes you may have the unique genes present in cultivar 2, which are not present in cultivar 1, you may miss it. The same thing in the cultivar 3. So what we need to do, we do not need to sequence just one genome. We need to have several genomes if we would like to understand the diversity of that particular crop. So this concept called pangenome, we extended this concept from pangenome to superpangenome. And what is superpangenome? So for instance, in the case of chickpea, we have Cicer genus. You are having Erythrem species. So many times people are doing the pangenome in Cicer erythrem. We went one step further. We said, well, hold on. In Cicer, I'm having 8 different species. Can we understand the pangenome across all these different species at the genus level? And we gave a concept of the superpangenome based in 2020, way back in 2020. And during last 3-4 years, though from our lab, our superpangenome was published just 2-3 weeks ago. But people all around the world in tomato, in soybean, in rice, they already have published the superpangenome during last 3-4 years inspired by this kind of concept. Why we wanted to do this thing? Because as we know, in terms of agriculture, we have the domestication bottleneck. Then we are having this breeding. So during the domestication and breeding, we have lost several important alleles which are present either in the land races or in the wild species. Now, through the genome sequencing approaches, we have a possibility to identify those important alleles. And with this objective, as I mentioned, that we have worked with Professor Swapan Datta on many of these projects, including pigeon pea, chickpea, etc. So where we have sequenced more than 292 pigeon pea lines, 429 chickpea lines, and we identified the genes associated with different traits, including the climate change related traits, where we identified the genes. We have extended these approaches like genome variation map in the different crops, including peanut here, in collaboration with Guangdong Academy of Agriculture Sciences. And while sequencing more than 300 peanut lines, understand the genome variation, we were also able to map several of the genes associated with the different traits. Similar kind of work we did in the case of pea together with Chinese Academy of Agriculture Sciences, where we decoded the genomes for about 118 exons in Azuki beans with about 322 exons. So what I'm trying to say, our lab has developed an expertise from the genome sequence to the genome variation map. And then furthermore, together with ICR, and of course, all this work was from ICRISAT and collaborative partner from ICR Institute or so. What we did, we went one step further, and here we decoded the genomes for more than 3,000 chickpea lines. And this was the first time that for any crop, we got the whole genome resequencing data available, not for hundreds of thousands, but more than 3,366 lines. And top of that, we also did the phenotyping of these lines. And here I would like to again highlight the contribution of Professor Datta, that at that time point, he was the Deputy Director General of Crop Science in ICR. And when I used to interact with him, I say, sir, we need to do the phenotyping. Well, then breeder organization people say, we have never done the phenotyping. Then Professor Swapan Datta would like to organize the meeting with the breeders, with the different ICR Institute. ICRI said, can we do this kind of work or so? And this is the way that like push or inspiration or encouragement that we have worked together, and we were able to phenotype So not just sequencing, phenotyping more than 3,000 lines at 6 different locations for 2 different years. And once you got the data, we were able to develop the haplotypes for different agronomy related traits in the case of chickpea. And furthermore, now we have completed the sequencing of more than 10,000 lines, which is a huge work. And I think so we will see that when we will be able to publish, because analysis takes lots of time. So far, there is not a single crop where they have done the whole genome sequencing. People have done skim sequencing. I'm talking the whole genome sequencing at the level of pangenome. So this kind of work now, what we have done, that we have targeted more than 10,000 chickpea lines around the world. We have sequenced all of them. We are trying to make the sense from these data. So this is the beauty of this sequencing technology. And I'm sure that next generation of scientists or those young students sitting in this hall should be inspired that now you can sequence anything very fast and very cost-effective manner. Friends, I was talking about the super pangenome and about the sizer. So as I said, like what we did in the sizer species, we developed the de novo genome assembly for 8 different species. Earlier, people were developing just 1 or so. And based on all these 8 genomes, you have done this analysis. So for instance, here, this is the cultivated one, Cicer aritinum. This is Cowley desi, Cowley chickpea, reticulatum, echinuspermum, penetifidum, yamashite, all these things. And now you are having very clear relationship that which genes are missing in which species and how we can transfer the genes from one species to other. So I think this is really very exciting. And this was just published 2-3 weeks ago in Nature Genetics. In the case, if you are interested, please read it and let me know if I can be of any help. Now, we are continuing similar kind of work here in Australia. So like for instance, in Australia, we are working on wheat, we are working on the horticulture crops, but legume crops are also very important. Good thing in Australia is that Australian conditions, like environmental conditions, is much more worst, I will say, than many places in India, because here we are like in Western Australia, where I am. First is that all the agriculture is rain-fed agriculture, and our soil is not a soil, rather sand, and we are having the issue of the drought, heat, etc. So even then, through the breeding technology, they already have the varieties which are having much more higher yield than many other developing countries. But what we would like to do, we would like to push this boundary further. For this, recently, we are developing the pangenome for Australian chickpea varieties, and we already have decoded, or we already have sequenced large number of chickpea varieties from Australia. We are working further in the detailed analysis to understand that what makes Australian varieties much more drought tolerant, heat tolerant, and how we can push this limit further. Similar kind of work we are also doing in wheat. Wheat is also very important crop in Australia. And what I have done, that I have initiated an international initiative on development of wheat pangenome and haplotype catalogs, where we are working with more than around 8 to 9 different countries. And these are the people that are coming together. I'm still very much interested to have the colleagues from India. I have talked many times in ICR and Indian Institute of Wheat and Barley Research. Hopefully, we should be able to bring them on board. But anyway, what I'm trying to do, that while each country like Canada, China, Germany, Italy, so these countries, they are developing the pangenome in their own countries. We are doing in Australia, and then together with IPK and Helmholtz-Munich, we will put all these things together at the international level. And in the case of Australia, while working with the 3 different company, AGT, Intergrain, Longreach, Plant Breed Company, we are doing the de novo genome assemblies of several varieties which are coming from the different states of Australia. So this is the way that we are working, not focusing on one crop, but because we need to think the environmental sustainability, we need to think about the proteins, we need to think about the carbohydrate, etc. So this was some of these our contribution towards this genome assembly, genomics work. Now let's move further, that how we can use these things for the crop improvement. When I was at ICAR-INDIAN, our main focus was how we can integrate the genomics technology in the breeding program. For that, when you talk the breeders had the 2 challenges. One, the cost for use of the sequencing and genotyping was expensive. Second, in many state agriculture university, breeders were not well equipped in terms of the facilities or in terms of the capacity building. So what we did, on one hand, we established that cost-effective genotyping platform. So for instance, the different marker genotyping platform, they're very cost-effective. And while using these platforms, we did a lot of sequencing, genotyping of the breeding population. We put those breeding population in the field and we use the controlled environmental conditions. And then when we put these things data together and based on these things, we mapped 20 to 50 traits because breeders were interested to identify the markers or genes associated with different traits. So we did this thing in the case of chickpea, groundnut, and pigeon pea, etc. The next thing was translate that how we can use this research or genomics research in the breeding program, how we can take these genes to the field. And as I said, so initially we used to have this cost. So we always talk about the cost-effective genotyping platform. For that, I worked with the Bill and Melinda Gates Foundation together with CIMMYT and IRRI and ICAR as a lead organization. And we established a platform called High Throughput Genotyping Platform, HTPG. Through this one, we have a possibility to genotype a single line in less than $1 to $1.5 per sample. Good news is that this platform nowadays is being used by 8 different CG centers, 30 different NARS programs, and many organizations in India, they are using these things. This is being used across 28 countries, and we got the markers for more than 18 crops. So this is a very sustainable model now. Second thing was to train the breeders in use of those sequencing or genotyping technology. And when I was at ICAR said, we trained more than 485 scientists from more than 36 countries around the world. And the main purpose was how breeder community can use the genomics resource in the breeding. And these two factors like low cost genotyping plus training, this was very, very helpful. And as a result, many breeders in India, not only from IRI or IIPR, but also from many state agriculture universities, including PAU, University of Agriculture Sciences, Raichur, or JNKBB, many other universities, they came on board and they used these technology in their breeding program. Once they developed the better lines, then they went to the All India Coordinated Research Project and they had very extensive, the field trial, etc., and the lines which are doing great, then they were released. And I'm so happy to mention that even in the case of chickpea, which used to be called orphan crop, where we did not have the genomic resources, now we got the genome assembly, we got the genomes, gene expression atlas, we got the pen genomes, and some breeders like Bhardwaj from IRI, they have been very proactive to embrace those technologies. And then as a result, now we have developed, delivered IRI, IIPR, they have delivered several varieties, drought-tolerant chickpea varieties with the yield more than 2 tons per hectare. And they are not just on the paper, they are these varieties that are already grown by the farmers. Similar is the case in the Fusion wilt resistant, where people like Shailesh Tripathi and also here the picture of Dr. Mannoor. So there are many people, they have been involved in the breeding program from the different places in the country, and they have delivered several of these varieties. And we feel very happy that the genomics research, we did not just publish that or shared with the public community, but we also utilize them in the breeding program. This is the example of Ascochyta blight-resistant chickpea lines in the case of Punjab state. And not only in India, our research has also helped the breeding program and farmers in Africa. And here, these are the success stories of 2 varieties, chickpea, Jelly2 and Grar, which are giving higher yield more than 15% through the genomics assisted breeding. Similar is the case in the case of Kenya, where we have delivered several lines coming from our genomics assisted breeding program. And not only Kenya, but in Tanzania also. So now this has been, so basically it took some time for us to establish the pipeline. But now even when I'm in Australia, I keep on receiving the calls or mails from my friends and colleagues. Hey, there another line has been released now. Another line has been released. And this gives me lots of satisfaction, happiness that that research what we generated has also been put into the use and reaching to the farmer. So this is the case of chickpea, same thing in the case of groundnut. We are having high oleic groundnut varieties. We are also having late leaf spot resistant variety. And not only in India, but also many other places in Ghana, etc. So I think a lot of success stories in the case of peanut and also in the case of pigeon pea. So I am not giving all these same slides again and again. So, but I feel so happy that we already have delivered more than 20 varieties of chickpea, groundnut, and pigeon pea coming through the genomics assisted breeding in India, Ethiopia, Ghana, Nigeria, and Tanzania, Kenya, and many, many countries. So I think these are some of the success stories that how you can enhance the crop production even of these legume varieties or so. Now going one step further. So we talked about some work about the legumes. We already know about the importance of the cereal crops like rice or wheat or barley. In Australia, wheat is very important. My lab is working on wheat. But another thing is the horticulture crops. When I landed in Australia, though I never worked earlier on the horticulture crops, but Hort Innovation, which is one of the leading agency for the horticulture crops, they contacted us and they say, Rajiv, we would like you to establish a advanced genomics platform for Australian horticulture. I say, well, I never worked in Australian horticulture. They say, well, we are having some crops which are not genomic resources rich crop. You need to develop a platform. And we established a proposal, and this is an initiative about $13.6 million. And in this project, initially, we are working on 5 important crops, including banana, pineapple, papaya, custard apple, and passion fruit. And we brought the partnership from different organizations here in Australia, some partners from internationally. And what we are doing, we are basically assembling the genomes of these crops. Our plan is to sequence entire germplasm collection, mining of the candidate genes, development of the cost-effective genotyping platform, and put all these resources in the database. And here in Australia, this is also important that we are establishing our relationship with the industry partners, international community, knowledge dissemination. In the case, if you are working on some of these horticulture crops, we will be more than happy to work with you. And very short frame of time, together with different partners, what we have done that we have developed a genome assembly in Williams Cavendish together with our partners from coffee and banana. This is very big genome, and we are having 33 chromosomes. There were some assemblies published earlier, but now our genome assemblies provides out of 33 chromosomes. So all the 33 out of 33. So you should have 66 telomere. We have identified 60 out of 66 telomeres. We have identified about 30 centromeres out of 33. So I think, and then some of these published literature, and we compared our genomes and we found that our genome assembly is much more bigger than that one. Second, now with this possibility of these new assemblies, we have a possibility, we were able to develop the haplotype resolved assembly. This is the case of pineapple. So in pineapple, now we got the haplotype 1 assembly, haplotype 2 assembly, and you can see that we got all centromeres, we got more centromeres in the different assembly or so. So I think this is the way we have developed now the assemblies. In the case of pineapple, we have developed this assembly in the papaya. Papaya is also, we are now trying to identify the sex chromosome specific genes, etc. So now in the case of papaya also, our colleagues here at Murdoch University, they have developed the genome assembly, the same thing in the case of passion fruit. So what I'm trying to say, we have been working on all these different kind of assemblies. The same thing in the horticulture crops now. Our lab also started to work on many other horticulture crops. And there's another project which is about $7 million. And here idea is how we can do the genotyping in the faster manner, and we call this RACE project. So this is another horticulture crops. Another one, because of legumes, we are very much interested in legumes. So in Australia, we don't work on pigeon pea and peanut, but now we have got other crops like in pulses or called fava bean and lentil. And now I'm leading this initiative. And this initiative is called the genetic gains for yield. Here I'm again working with large number of partners here in Australia and also globally. And what we are doing more or less similar kind of thing, having the diversity set, phenotype them, sequence them, put them in the platform, and so that we can utilize the genetic diversity. So this is also a very exciting project. Finally, unfortunately, I don't have time, but one other key thing was that on one hand, we were talking about the nitrogen fertilizers. But what is happening? Several crops, including wheat. So when we are having this excessive use of the nitrogen fertilizers, these crops, they are not making the best use of these nitrogen use or this nitrogen fertilizer. And what they do, they're having lots of time nitrogen leaching, nitrous oxide. This is adding to the climate change gases also. So what we are trying to do in this project, through the genomics and gene editing approaches, we would like to develop the wheat lines which can have the higher nitrogen use efficiency, so that farmers should be able to apply, spray limited amount of the nitrogen fertilizers, and we should be contributing less nitrogen pollution in the soil. So this is also going in the area of environmental sustainability perspective. Last project which I'm leading, and this is about the heat tolerance. And in the heat tolerance, again, Dr. Pathak mentioned about the great story about the climate change. And now in context of Australia, this temperature is increasing regularly, and our plan is that how we can provide that new germplasm to our breeding programs in Australia so that they can tolerate much more temperature. And again, so what we start from the germplasm, we do the field trials, we do the heat chamber trial, sequencing, putting these things together and provide this thing to germplasm. So I think, so this is the way that we have been working on wheat as well. So in summary, my lab is continuously working on the legume crops, chickpea, fava bean, lentil, etc. Now we are also working on wheat, making wheat is the drought tolerant, heat tolerant, and better in the nitrogen use efficiency. And on top of that, we are also working on the horticultural crops. And idea is also making these horticulture crops climate proofing horticulture crops through developing the advanced genomics platform. So I think this was in summary that we have been working. So in my opinion, the take-home message from my presentation is crop diversification is essential for sustainable agriculture. In terms of the technical aspects, genomics, pangenomics, GWAS, and haplotype analysis can identify precise genes and genetic selection tools for genomic breeding. Fast-forward breeding processes, they can create and incorporate superior alleles and haplotypes and crop improvement. One area I did not discuss in my presentation, but we are working very heavily now, and this is the use of artificial intelligence and machine learning, especially with the large scale of the sequencing data. As I was talking, for instance, in the case of chickpea, we got more than 10,000 lines sequenced. We are doing the simulations, so we are on the different aspect. Finally, I would like to also mention that partnership is very important, and this partnership should be international, national, and also the government and funding agencies supportive so that we can have sustainable agriculture for the food security. And in the end, I would like to thank large number of partners around the world from India, including ICR, IIPR, IRI, DGR, many state agriculture universities, and now many partners here in Australia with whom we are working. So this ends China and then many other global organizations and large, huge partnership or funding from the different organizations, including ICR, Department of Agriculture and Farmers Welfare, Gates Foundation, CGIR nowadays in that GRDC, Hot Innovations. So lots of organizations, they have contributed, several people from my lab. They are working on these different aspects, postdocs, PhD student, technical assistant, etc. Thank you very much. And finally, I would like to leave the slide here about the International Wheat Congress. So in the case, if you are working on wheat, please do join. We will also have a privilege to have our Director General and Secretary there, Dr. Himanshu Pathak, also in Australia in September. So please spread the word. And with these words, thank you very much once again. And back to you, please. Hope they can hear me, or this is a big silence now, it seems.
No, we can hear you.
Okay,
Thank you so much, Professor Varshney. It's always a pleasure to hear you speak, and especially when you end with the last slide, that proves that you are actually one of the, if not like one of the leading agricultural scientists of our age. Professor Shobhan Datta wants to say a few words to you.
Sure. Thank you. Yeah, thank you, Tanushree.
Hi, Rajiv. Thank you very much for being with us today this afternoon. And your talk, definitely a talk of fascination of modern agriculture from plant science to any beyond of any science. And that's for definitely for human welfare. But one thing that we are missing you to felicitate here, what we thought after being a fellow of Royal Society. So you have to promise us and me that you have to come again to the Department of Botany of Calcutta University to give a talk, and we would have the privilege to felicitate you, right?
Thank you very much, sir. So nice of you. I will do that one. Thank you very much. Yeah.
Okay, thank you very much. So once again, for a fantastic and a great talk. Another big hands to Dr. Rajiv.
Thank you very much, sir. Thanks very much. And thanks everybody for listening. Thank you very much.
Thank you, Rajiv.
So we move on to the next lecture, which will be delivered by Dr. Anindya Bandyopadhyay. I would request Professor Krishnendu Acharya to introduce the speaker to us.
Good afternoon, everybody. It's a great pleasure to introduce Dr. Anindya Bandyopadhyay. Right now, he is a principal scientist, leads genome editing, International Maize and Wheat Development Center, CYMMYT, Department of Biotechnology. His area of specialization is CRISPR platform, synthetic biology, biofuel, novel agribio products. Dr. Bandyopadhyay proved that science has no boundary. He has his graduation and master's from zoology. He started to understand plant biology under the supervision of Professor Dotto and Professor Shormista Roy at IRI and Calcutta University. So after completion of his PhD, he did many postdoctoral work in Medical University of South Carolina, then University of California, Berkeley, then University of Kentucky, probably, yes. After that, he started his career as a group leader and research scientist, crop transformation and promoter discovery, Maharashtra Hybrid Seed Company Limited. Then after that, he worked as a molecular biologist in IRI. Then he joined as a professor, University of Philippines, Department of Biology and Biotechnology, Philippines, where he's working up till now. During his journey as a researcher, he has published 64 interesting papers and published in different journals of international reports. So those who are interested to know about his work, please follow him in ResearchGate. He is a very fantastic ResearchGate profile too. So now Dr. Bandyopadhyay, dais is yours. Thank you.
Before he takes over, we have a small thing to do. Can I request Professor Ashok বিশ্বাস to please felicitate Dr. Bandyopadhyay on our behalf? And can I request Monisha to help Professor বিশ্বাস do the honors?
So it's an honor to be here. And really, I'd like to thank Department of Botany, Alumni Association, and especially Professor Datta for inviting me here to give a talk. যেটা বললো আমি জুওলজি পড়েছি, বিএসসি এমএসসি দুটোতেই এই নিচের ডিপার্টমেন্টে পড়তাম। তারপরে জিন ধ্রুবজ্যোতি বাবুর ল্যাবে কিছুদিন কাজ করছিলাম, জিন ট্রান্সফার শেখার ইচ্ছে হলো, তখন একটা পজিশন বেরিয়েছিল ইরিতে, আমি গেলাম ডক্টর স্বপন দত্তের ল্যাবে, তারপর থেকে বটানিতেই রয়ে গেলাম, আর অন্য কোথাও যাওয়া হলো না। তো একটা কথা, আমি ওটা বোধহয় একটু ভুল হয়ে গেছে, আমি ফিলিপিন্সে ইউনিভার্সিটিতে ছিলাম না, ওটা
ঠিক আছে, আমি অ্যাকচুয়ালি ইন্ডাস্ট্রিতে চলে যাই। আমি ইরিতে ছিলাম, মাঝে একটা সময় ইন্ডাস্ট্রিতে চলে যাই 6-7 বছর। এখন আবার ফিরে এলাম রিসার্চে 1 বছর আগে সিমিটে। তো লেটস স্টার্ট। So I have divided my talk in majorly 3 parts. So first part would be like basic CRISPR, and I kept only a couple of slides, but after talking to Sudipto and talking to Dr. Datta, I realized some students will be here, so it's better to keep some more slides and give some idea on what CIMIT is, what kind of collaboration could be generated, etc. So one part would be CRISPR, second part with an example from rice on plant genome editing, and finally what we do in CIMIT. So these are the 3 major areas of my talk. Okay. So initially, we knew how to develop GM and how to transfer gene in plant genome or any genome, but it used to be random. It can go anywhere. But what is the beauty of CRISPR-Cas9? It can, instead of random insertion of the gene, you can use CRISPR-Cas9 system and you can target a region of genome and you can send your gene there. So this is the uniqueness of CRISPR with respect to genome editing by transgenic development. So 10 years back it started, and immediately multiple things started happening. Number one, I would say a lot of funding started coming, pouring in. You can see from 2010 and from around 12 or 13, from private sector, for government, a lot of funding came up in this area. And then multiple genome editing tools were there, like zinc finger nuclease or other kind of meganucleases. But once CRISPR-Cas9 came up, because its simplicity and it's cheap to use, very quickly use of CRISPR-Cas9 become popular globally. And also you can see a lot of patents started coming in plant science. China is the one, in animal science, US. These are the 2 countries holding most of the patents in today's world. So, and we should know that Jennifer Doudna and the other lady, Charpentier, they got the Nobel Prize on CRISPR. But who basically developed it fast and who basically put it for IP? There was a lot of patent conflict and etc. And that gave us a little bit issues in using CRISPR or like democratization of CRISPR around the globe. But anyway, so this is one of my favorite slides, which I always tell that whoever has eaten yogurt or cheese from any shopping mall or from any reputed company, those people have eaten a lot of CRISPR last 20 years. দই এবং চিজ খেলেই আমরা প্রচুর CRISPR খাই এবং আমরা যুগ যুগ ধরে খেয়ে যাচ্ছি, যুগ যুগ ধরে at least 50 years আমরা খেয়ে যাচ্ছি. So why? Because all these companies started selecting their organism for developing cheese and yogurt. And they found that there are certain bacteria which are basically resistant to fudge viruses. They are normal enemy. So they started selecting those, and later on they found it's nothing but there is a special region in their genome which is called CRISPR array. So those bacteria are having a special region. What happens? When a fudge virus attacks a bacteria, the fudge virus inserts its genome. So bacteria cuts that into small pieces and place it, internalize it into its own genome and keep it as a memory. First time it gets sick, but when second time the same bacteria, same fudge viruses attack, then what happens? They use this memory, and then through CRISPR system, they attack those genomes of newly infecting fudge viruses and they cut them. So it's an adaptive immune-dependent memory of a bacterial system. So this was established, and a lot of publications were there during 60s, 70s, 80s, etc. But later on, what happened, these scientists who got Nobel Prize and the other person I showed from Broad Institute of MIT and Harvard, they showed that this system could be hacked or this system could be modified to do genome editing. So what is that? A simple protein, which is known as Cas protein or any nuclease, which cuts the genome. And then one gRNA you design it as per your need. That will go and bind to certain region of genome, and it will carry the the nuclease with it, and nuclease go and sit there and cut. So after the cut is made, what could happen? Number one, random mutation may happen. Cell will try to repair that cut region and will bring some mutations. So random mutation will happen. And if it is within a gene, that gene will be knocked off. That will not work anymore because of random mutation. Or we can also do targeted mutation by supplying outside DNA fragment there, which can get incorporated inside of the genome. So after identifying this thing, it became hugely popular because only one small protein is needed and one mRNA is needed, and you can do targeted insertion, targeted knockout, and many other things. Immediately, last 10 years, globally, people have started modifying this nuclease and modifying this system. And every week, you can see one new publication is coming out with some new addition or new development. So, gene activation, gene repression, then gene epigenome editing, DNA imaging, RNA editing, many kind of stuff. People have taken this simple system, they attach some other protein with it, and that protein has done something like activation, repression, etc. So already multiple applications of this simple system now we could see happening in the global biological research area. And for plant, what is the major interesting part we have seen here? And where is the difference between GM technology to this one? So in GM, we send the tDNA. Bialistic, nobody does these days because it gives you multiple copies. There are many other rearrangements, etc. So commercially, it was stopped long time back for the industries. But for research organizations as well, nobody is doing bialistic anymore. So I saw that I gave example of tDNA. So you have agrobacterium tDNA, you have your gene of interest inside of it, you send it to the genome and it stays there. You have a beautiful line, which is resistance of something or with some beneficial characteristics. But for genome editing, what you will do, same process, you will take a tDNA and instead of your gene of interest, you put a CRISPR-Cas9 inside of that. It will go and sit on the genome. Then it will produce CRISPR-Cas9 system. CRISPR-Cas9 will go to a certain region of the genome. You can identify which region it should go. And then it will do some change. And that change will bring a beneficial trait, a beneficial characteristics. And then in the next generation, you can segregate the tDNA out from the system. So there is nothing foreign in it, only few base changes. So you can, although scientifically there is no reason, but globally, GM, there are a lot of controversies and many other things came up with GM. So scientists found a way to do GM or do similar kind of changes in a different way, where you can just segregate out your externally given system. So this is the uniqueness and difference with respect to GM and CRISPR. And multiple applications came up. So I kept 2 slides, like starting from cancer therapy to fertility related different aspects to genetic disease related. All the genetic diseases can be easily targeted and some base changes can be done and reverse from the disease gene could be converted to a non-disease one. And then different infectious disease, virus, bacteria, they insert their genome inside of the their targets And you can basically target their genome by CRISPR-Cas9 and get rid of it. And also organ transplantation, a lot of use is happening. CRISPR-based first diagnosis, now people are doing. We have also done in one of the industries, this is the publications there, where we identified CRISPR-based identification, quick identification of coronavirus. So that we also done, and all of the viruses I have mentioned here, people have developed a quick system by CRISPR. Livestock animals, these nice Dalmatians, they always have kidney stone. They are prone to that. They have some genetic disease there, and people are knocking out genes there and could make Dalmatians kidney stone proof. And then faster horse breed, healthier fish, dehorning of cows, etc. we all know. So these are very simple stuff people have done already, and some or other companies or some or other countries have developed this kind of products, and they went to the regulatory, different stages of regulatory scenario. Now, in case of plant, I personally worked on some of this. So more fruit per stem kind of tomatoes, non-browning agaricus, and then hop-free beer, spicy tomatoes, gluten-free wheat, etc., etc. Many things now are in pipeline. 6 products are in market already and available. Many countries have accepted, including India, accepted it as non-regulated. So it will be treated, any product by genome editing. There are 3 types. The SDN1 type will be treated, and SDN2 type will be treated as regularly bred product. So there will be no extra regulation on those. Similarly, in industrial sector, industrial biotechnology sector, people are using different kind of cells. It could be filamentous fungi, it could be yeast, it could be algae, it could be bacteria, plant cell, etc. And they basically changing the flux of certain product development inside of these cells and making a particular product inside of the cell, like cosmetics precursors and many other things, and then taking it to the large scale in the industrial scale and producing those products. So multiple applications of CRISPR-Cas9, briefly I have mentioned, and definitely many more applications are happening these days. I am not bringing all here. One example I'd like to give, we worked during our days in Erie a few years back, where we basically changed number of stomata in rice. So we had a notion that if we reduce the stomatal number in rice, then the plant will be able to store more water inside of his body. And then it could be drought tolerant, or at least water use efficiency will be higher. So we used both CRISPR-Cas9 and CRISPR-CPF1 or CRISPR-Cas12a, both the types. And we targeted exon 1 of one particular gene. It's called florigen. And what we did, we basically knocked out in 2 different ways, one with one CRISPR system, the other with other CRISPR system. And both the cases, we have done sequencing, etc., etc. We identified where the changes happened, what kind of editing we have done. And then we did sadhanan, etc., to show that CRISPR tool is segregated out. And then finally, if you see the top, these are the stomata, these yellow stars are the stomats in rice plant. And after knocking out, you have only few. So different line has different number of stomats. And so from four times reduction to 8 times reduction, we have taken multiple lines and we have checked where the yield penalty is less. Because reducing stomata will reduce entry channel of carbon dioxide as well. So photosynthesis will be less. So there will be some penalty in the yield. So we tried to see from 2 times to 3 times to 4 times to 8 times, which reduction gave rise your higher water use efficiency, but no change in photosynthesis. So that kind of stage we identified. Then I left Erie and many things. This was we published this one, and then Virginia Tech, they basically highlighted this work, and then University of Sheffield, they continued this work as a collaborator, and they took it forward in other crops as well. So this is one of the examples what we can do very quickly by applying genome editing and make a trait, which is like 3 months or 4 months time. We can have the trait, then we take the plant in the next generation, maybe another 3 months. So within a year, we can develop a trait. And if it is an elite line, then you don't need any further breeding to bring it to elite line, etc. So you take an elite line, which is popular, farmers are using everywhere, and you take that elite line, edit there, and immediately you get your targeted line, and you put them in the breeding pipeline, or you just develop hybrid, develop seed, and start giving the seed to the farmers. So time is just like 1 to 2 years, you can have it. And then later on, what kind of application you want to use, whether you want to make hybrid or you directly give that lines. So it is pretty faster than traditional breeding. So now I'll switch the gear a bit. What we do in CIMMYT. So these are previous work, some background of CRISPR-Cas9, then what we have done in ERI. Now what we are doing in CIMMYT. So before going to that, Dr. Datta told me what is CIMMYT and what is that you should mention. So there is an organization called CGIAR, Consultative Group of International Agriculture Research. It's an international non-profit organization which has 15 different research institutes. ERI is one of them, where Dr. Datta was there. I did my PhD. And then it's in here in Philippines, and there are many other, one in India as well, which is ICAR-INDIAN in Hyderabad. And then now I am in CIMMYT. CIMMYT, this is the largest at this point of time with respect to all CG centers. And here, these are the centers I have enlisted. So under CGIAR umbrella, there are 15 research organizations. We are one of the biggest out of these 15. And then if we see CIMMYT, this is our headquarters in Mexico City. Our next big center is in Kenya. And another one, we have an institute called Borolague Institute of South Asia in Delhi. So these are the 3 major centers I would say CIMMYT has. And it has presence in more than 80 countries. So at least a breeding station or a small lab or an office, we have at least in 80 countries across the globe. So our scientists, our breeders, they go to different countries, they gather the information, what is needed for that country, then we develop those products and give them for free. So we are a non-profit conglomeration under CGIAR. And we also collaborate with universities. We take PhD students, we take postdocs, we take technicians, all kinds of things across the globe. We advertise in our CIMMYT website, as well as we advertise in the CGIAR central website. So, and for genome editing, we have a specific strategy in CIMMYT, where we have this market intelligence team, as I was mentioning, 80 centers. Economists are there, social scientists are there. They go to the field, they try to understand what are needed for that country, what are the main crops, and what are needed, and what we can do. Then a breeding design team sits, they make the whole trait pipeline, and then trait teams sit, they identify which trait should be done by breeding, which trait should be done by some other technology. And then genome editing team sits and we see that out of 10 or 15 traits which are needed for a certain country, which one is amenable to genome editing and which one will be faster to do by genome editing, and then we go for those traits. So 2 major points we mentioned, as I mentioned, that we do all editing in elite line, no model crop, no model plant, no easier varieties. We are not allowed to do. So whatever genome editing we have to do in elite lines. And if a natural variation is there, normal breeding can handle that. We don't touch that. We only touch the difficult ones, which normal breeding cannot handle. So these are the 2 major things. And we take industry CRISPR technology as well as public sector. We take it through our pipeline. And once the seed is produced or developed, then through government, NGOs, nurse partner, we give it for free to the marginal farmers and farmers of Global South, Latin America, Africa, and India. And where do we see genome editing sits? If we consider the whole plant breeding in one slide, we will see that trait design and different kind of molecular breeding, genome editing can play a central role by one-step trait integration very quickly and can give rise your product in a much cheaper way and much faster. So this is why, while all these are helpful to each other, it's not that any technology is replacing any other technology, but if all these technologies are together, based on the trait, based on the availability of natural variations, we decide which way to go. And genome editing definitely plays a central role nowadays in breeding pipeline. So the first project I would like to mention, this is our most advanced project, which we have developed recently, and quite a good number of publications are coming out. This is MLN resistance maize for Africa. So MLN is a combination of two viruses. One is MCMV and another one is SCMV. These are non-affected plant. These are affected plants. And same time they were grown, etc. But it's a devastating disease for Africa. And many Western countries in Africa, they get devastated by this particular viral disease. And huge amount of loss happens of maize crop. So one wild variety from Thailand, after a lot of search, we found that there is one particular variety which are resistant to this particular dual virus disease. But it's a very exotic kind of variety. Nobody uses it, and all other benefits are not there. This is a very poor quality variety, I would say. So we took that and we identified that chromosome 6, there is a QTL, quantitative trait loci, in the genome, which is important. We mapped that. Every 100 base pair we knocked out by genome editing. And after mapping, we found out that there is a particular gene which is responsible for these resistance. And what we did, we knocked that gene off in many kind of elite varieties, our varieties of our interest. And we could see that significant viral resistance we could achieve by knocking out that gene. So this is 3 months back, I have taken this picture in Kenya, Naivasha. Naivasha, this area is prone for this particular viral disease. It's a hot spot. We planted our general lines and we planted our edited lines. And we could see that edited lines, without any yield or any penalty, it's giving almost 100% resistance to these two viruses. We have also identified another one QTL in chromosome 3, which we are fine mapping now to target the gene. And once we get the gene, we'll knock that off as well. And we will then bring two editing together by crossing or whatever maybe the way of multiplexing, etc. And we can get the next round of antidote ready if these viruses could break the barrier. Within coming 5 years, we will have the second round of stuff ready. So this will come out in journal soon. Also, we are working on wheat. We have already developed wheat with LR67, MLO, and PPO. So LR67, you all know, is a hexose transporter. If you knock this off, then you can get rid of a lot of fungal diseases. So that has been shown in many crops. So we have done it in wheat. And MLO is a powdery mildew resistant gene. So if you knock this gene off, these are all susceptible genes of plants. and then you get powdery mildew resistance. And we have done another one, which is PPO. This is basically after you make the dough of wheat, it becomes black. And in some of the popular varieties of Middle East, Pakistan, Afghanistan, India, Nepal, those gets quite dark black very quickly. So we wanted to get rid of these traits. So this is due to PPO. We knocked those off. And you can see here, I have shown in short that these are the normal ones, which become dark. But once we knocked out PPO, they are keeping their color. So it's a visual kind of trait, and which is quite important. Sometimes if it becomes black, then in industry, bread development and etc., they cannot use it. So this is one trait, LR67 and PPO. Yes, so different lines we developed. I'm not going in detail all of this. So we are working here with John Innes Center. They are doing our greenhouse trial, and they will also do field trial. We are also working with Agri-Food Canada. They are also doing field trial of the powdery mildew. These guys are doing field trial for, John Innes is doing field trial for rust. So already we have developed these edited lines. Already battery is gone. Yes, and these are the ones I was talking about, that blackness. So you just polyphenol, you knock out polyphenol oxidase. Now what we are doing multiplexing. 3 gRNAs we have developed in one construct. We'll send that. It will go to 3 places for 3 genes, and it will get rid of MLO, LR67, and PPO, all 3. So we will have a wheat line which will remain brown, which will not get black after exposure to environment, also which will be powdery mildew resistance and which will be rust resistance. So we are doing that multiplexing at this point of time. Also, we are working on groundnuts. So aflatoxin is a very well-known fungal disease. So we have identified certain genes in aflatoxin. Those are published. And then we are doing genome editing to get aflatoxin resistance groundnut. And again, we have taken all the elite lines and we are doing direct editing in the elite lines. So no intermediate breeding is involved to save time. Also, we are working on pearl millet, RANCI. Like pearl millet is a very important millet in Africa. In India also, in the poorer section, this is one of the source of protein. But what happens after production, it gets rancid very fast due to different molecular aspects. And you cannot use it due to foul smell. Multiple polyphenols get produced. So we have taken 2 genes, basically to knock out some of the lipoxygenase. So rancidity is caused by hydrolytic and oxidative rancidity. So we are knocking out both. And we have developed the plant, and this will be tested very soon. So this is one of the project. Again, we are using elite inbred lines. And one more, we are working to witch wheat striga resistance in maize. So this is another one project going on in collaboration with University of Amsterdam. Harold Bormister is a very famous striga person, biochemist. So he basically identified, last year he had a science paper, he identified many genes in striga pathway, which basically helps striga to grow and infect the plant. So these are the plant genes. So we have identified, he is our collaborator, so we have identified 7 genes, and one by one we knock them off, and plants are ready. Whenever we get the seed, we'll send the seed to him to do the biochemical analysis and check what kind of change we have brought by editing. So this is one more. So I just finished some of the examples which we're doing. There are more projects, but maize, wheat, pearl millet, groundnut, and recently we have started PGNP. These are the crops we are working in CIMIT. We are having multiple projects, but our target is not doing basic science. Our target is to develop products and give that products for free to the global south, poor countries. So that is why funding agencies fund us. I thought I will briefly mention about CRISPR, how its IP is complex, but I'm skipping that today. But the good news is Latin America, most of the countries, North America, they already consider genome editing as non-regulated. So it will be treated as any normal product. Europe, UK, this is a little bit old slide. So this red is not there, I would say, it is yellow now. So UK, they have approved. Very recently, last month, EU legislation came up. They are telling it in a different name, but genome editing 1, SDN1 will be treated as normally bred crop. So there will be no need of extra regulation, etc. So that's a good news. India already declared in many African countries, as I was mentioning in old slides. So there are at least 6 countries they have already approved, and there is no. Japan already came up with a product, and GABA tomato is already available in any supermarket of Japan, and people are purchasing in a big way. This is the one thing. Australia, still South Africa and New Zealand. These are the 2 countries they are considering it as GMO. Other than these 2 countries, and one caveat in Europe, they don't want to accept any patent. They don't want to accept any patent before March this year. So whoever discovered these, whoever has developed product, which company, university have developed something and patented those, they don't want to accept those. They want all these for free. I don't know how much that will be feasible, and in today's global trade and etc. But at least they have accepted the technology as an important technology, and they need to take it forward. So that's it. Okay, I'm skipping this one. Just 1 minute I will take for one slide, which I skipped now, but this is something for the students I thought I will talk about briefly. In China, I stayed 2 years in China in one of the private companies. I saw it in my eyes. These small pigs, chotto shuor. So they like it. Women like it. They take it in their vanity bag, and they roam around like Chihuahua, the small breeds of dog. They are developing small breed of these different animals by genome editing. Now government banned it, but there are already companies who has product in the market. Okay, so this is just for fancy. Yes, pet. Yeah, so they're doing it. Number 2, bioterrorism. You all know this can be used. Most of the countries knows how to develop corona type of viruses and make it public for public consumption. So any countries, those who are doing molecular biology last 10 years, 15 years, there are scientists who can do this. So we need to think about that. This is a scientist who become very popular. I forgot his name. He basically changed one gene in embryo of a HIV positive mother, and the father was also HIV positive. By changing that one gene, he could give rise to child who are HIV free. So that was great initially, people thought, but without any regulation or anything, without telling anybody, he did that, and Chinese government took him to jail, and he was in jail for 3 years, and he came out recently, and government told that he can do anything other than science. So that kids are basically absolutely healthy, great, but they have enlarged brain. So it has off-target effects. So until and unless enough research is done, it's not wise to use this technology everywhere in human breeding or etc. Because once your human is produced, we have plant scientists, we have opportunity. If a plant has pleiotropic effect, we can throw that off and we can use the correct one. But for kids, you cannot do that. You cannot throw one of your kids and keep the good ones. So this kind of applications, and there are already multiple traits identified, which can be changed by genome editing in the zygote level, embryonic level in human. So you can make your kid with more muscle, you can make your kid more brainy. So those kind of genes are already identified. So if properly not controlled, any company can come up with something which is not very nice for society. So while doing this kind of technology, while these kind of technologies are extremely important for all aspects of human life, but misuse, chance of misuse is also there. So regulation and all these things should be in place in all of the countries. So I thought I will talk about that as well. So that's it from my side. Thank you very much. These are the funding agencies and collaborators. Thank you very much.
Thank you, Anindo. Just listening him, I realized that one of the very famous saying that even it was mentioned in the morning, that science does not have a boundary. So Anindo started from Calcutta University, then went to International Rice Research Institute, then to US, different very good laboratory in US, then came to China as a head of genome editing group, then came to a private company, big company of India, Reliance Myco, and then of course, later on Reliance, and then now head of biotech cement. So physically, see Dr. Anindo's moving from one center, one corner, one region to the extreme another region. He started even science, basic science of plant photosynthesis, C4 rice, and looking for the water carbon dioxide efficient rice by C4 photosynthesis. He published several papers on that. Now, his later part of the work also looking for the stomatal structure and changes of stomatal structure to get water efficient rice. And later part, much more trait development. So see, an animal researcher coming from completely a different background and came to plant science, and then from plant science, moving around, the science doesn't have any boundary. That is, he himself is the evidence. The science doesn't have a boundary, that you can move around the science as long as science is exciting and gives you certain interest and pleasure, and what Anindo is enjoying. Let's give a big hand to Anindo. We are proud of you, Anindo. Fantastic talk, and I'm sure all of us enjoyed Anindo's talk. Thank you.
Thank you, Dr. Bandyopadhyay, for that wonderful talk. So with this, we come to the end of this scientific session, and we will... Huh? Morning scientific session. Yes, sir.
একবার একটু ব্যাক ক্যামেরাটা দেখুন না, দেখা যাচ্ছে কিনা। এখানে দাঁড়ালে দেখা যাচ্ছে তো? ওকে।
এই বসো, এই পালাচ্ছ কোথায়? বসো, এখানে বসো, এখানে বসো। এটা দেখি
স্ক্রিনটা ফ্লোট করতে, এটা এখানে ফ্লোট করছে তো, হাইড করে দিয়ে।
Very good afternoon to you all. I hope you have all had your lunch. Please settle down because we are ready to begin the afternoon scientific session of the day. We have 2 lectures lined up. The first will be delivered by Dr. Sujoy Rokhit, Director of Indian Institute of Agriculture and Biotechnology. And I would request Professor Shudendu Mondol to please come here and introduce the speaker of the day.
Good afternoon. I'm very happy to be present here and to introduce one of our very bright young scientists who hails from Shantiniketan, early education from Vishwabharati, from there to BHU, then to IARI, then finally to Japan. And coming back, he joined ICAR and working since there from scientist, and presently he reached to the climax of the position of the director in the Indian Council of Agricultural Research. So, as I have said, he had his postdoctoral research work in Japan, that is in IoT Biotechnology Research Center, as a JSPS fellowship from 2003 to 2005. And joining ICAR, he reached 2008 to 17, worked as a principal scientist, plant breeding in the Indian Institute of Millet Research, Hyderabad. Then after 1917, he joined as director, ICAR-INDIAN Institute of Maize Research, Ludhiana, from 17 to 22. And now from 22, he is working now director at the ICAR Institute of Agricultural Biotechnology, RANCHI. I'm very happy to inform you that he has already 26 hybrid cultivars, improved cultivars from the different species, like from the traits of maize, cotton, pea, and chickpea. He developed highly reproducible regeneration and transformation protocols from both mature and immature maize embryos. He has to his credit more than about 200 research papers, 40 genetic stocks, over around 900 DNA sequences registered to his credit. And in 2018, when Indian maize production was challenged by a new invasive pest called RB1, Sapodoptera fraguita, he led the fight as the director of the National Maize Program, and he played a crucial role in establishment of the ICAR-INDIAN Institute of Maize Research at Ludhiana. He is, for his scientific achievement, for his work, he has been elected as a fellow of the National Academy of Agricultural Sciences, Indian Society of Genetics and Plant Breeding, and also National Academy of Sciences in India, Allahabad. He has received also a number of awards, like Dr. B.R. Varole Award for Excellence in Plant Genetic Resources, then Agricultural Leadership Award 2021, Dr. A.P. Joshi Award 2020, Kalaya Krishnamurti National Award 2021 from Bangalore, Chaudhary Devilal Outstanding ICAR PROF Award 2019, then Best Annual Report Award 2020, and also you'll be happy to know that he had, apart from JSPS postdoctoral, also he was selected Boy Scout Scholarship, CSIR Travel Grant Scientist, and also Jawaharlal Nehru Award of the ICR, and also UNESCO Fellowship in Biotechnology. Best PhD student of IARI Medal and IARI Merit Medal, Jawaharlal Nehru Memorial Award, and many other awards. He has been the president of Maize Technologies Association of India, New Delhi, and President Agriculture and Forestry Section 106th Indian Science Congress. I am also happy to announce that during the ICR QRT team, I was one of the members for visiting 22 ICR institutes, which I was delegated by Professor Swapan Kumar Datta, and Sujoy worked with me in that team and efficiently ran about 7-8 top-ranking scientists in that team. So with these few words, I'm very happy to introduce our young from Shantiniketan, because I was a former student of Shantiniketan Vishwabharati. Worked there, Professor Datta worked there, starting from his early career from Shantiniketan. So I request Dr. Rathina. Thank you very much.
Thank you, Professor Mondal. I will now request Professor Rita Kundu and Professor Shobhan Datta to do the honors by felicitating Dr. Rokhi. Tanushree, please, will you help them?
Thank you, Dr. Mandal, for your kind words of introduction. I start my talk with my homage to Professor Sharma. I never had a chance to interact with him, but I saw only once during the centenary session of Indian Science Congress. And from distance only I saw him. But I know his work and the kind of contribution he has made. So with this, I'd like to start my talk. But before that, I must confess that I'm not a public speaker, but when somebody's mentor insists that you will have to speak, so I had to speak. So when the Professor Datta asked me that you will have to deliver a talk, I have chosen this. So you may wonder that I am from Indian Institute of Agricultural Biotechnology and why I am talking on maize. I must confess that more than half of my scientific career, I worked on maize. So there is a general human biasness towards this maize. And I always feel that the kind of position maize should have got in Indian agriculture scenario that perhaps it has not got. So in my presentation, there will be 3 parts. First, I'll try to impress upon that why I say that there is a silent revolution in maize, and then how maize can contribute towards the nutrition and energy security of the country. So globally, if you see, maize ranks second in terms of its area, but it tops in terms of production. And if you see the growth rate of maize, both in terms of area as well as production, it is one among the highest among all the cereals. If you see production country-wise, India ranks fourth in terms of area, but seventh in terms of production due to the less productivity of average productivity of maize in India. But sometimes average is mostly used and abused statistical parameter. If you see that average yield of maize in India is around now 3.5 ton per hectare. But if you compare with USA, which is nearly 10 ton per hectare, but you take the duration of maize in USA, it is almost 180 days. But in India, it is only 100 days. So per day productivity of India, maize in India is certainly much higher. Now, if you see the growth trajectory of maize, since 1950, the overall maize production has increased almost 18 times, with almost 3 times increase in area and nearly 6 times increase in productivity. Now, if you see the growth rate, again, within India, both in terms of area, production, or productivity, it is one among the highest. And this is not only true for last 5-6 years, it is true for last almost 3 decades. The growth rate of maize is much higher than what we always talk about rice and wheat. Whenever we talk about green revolution, we always talk about rice. I know here most of the people are working on rice. So it is people may not like, but this is the truth that last 30 years, the maize is progressing much better than any other crop. Now, if you see, almost 70% of the production is coming from Kharif maize, mostly from central and peninsular India and Bihar. But of late, this particular state, West Bengal, has come up in a big way. So what the maize scenario is concerned. Now, if you see the per annum growth rate of maize, and you can find that for last over 30 years, it is 50+ growth rate. And currently, the productivity is increasing by nearly 90 kg per hectare per annum. So this is one among the very significant achievement. Now, if you see the application of technology, first initially we are talking about land races, then we move to double cross hybrids, composites, then mixed of double cross, 3-way cross, different type of composites, and finally to single cross hybrids. 2 important interventions when we talk about maize scenario. One is establishment of AICR on maize in 1957, and then introduction of seed policy in 1988. And after that, when private sector came into play, you can find that there is significant progress. But overall, I'll say that focus on single cross hybrid has contributed very significantly in achieving this phenomenal growth rate in maize, or yield gain in maize scenario is concerned. Now, if you see the growth rate in all the crops, like first it is rice or wheat, it has contributed immensely towards achieving food security, which of the morning our Secretary Dare and DG ICAR was talking about. But of late, it has reached almost a plateau. But maize is continuously achieving more than 50 kg per hectare per annum growth rate. But if you see how we have achieved, rice and wheat had both technology as well as policy support. You know, PDS is a big boom towards maize, and now all politicians are giving 1 rupee or 2 rupee or free rice and wheat. So obviously there is a lot of demand. But in maize, it is mainly everything happened because of the technology. There is little support of any policy in the last several years. So, but in recent past, there is certain renewed emphasis on maize. I'll be talking when I'll be talking about energy scenario. Now, if you see, this is a district-wise scenario. You can see that last 30 years, how the area of maize has increased across, whether it is in Kharif maize or Rabi maize, and the areas with the dark color means higher area or higher yield. That is increasing very significantly. So this is really a very significant part. Now, all this progress which we are observing in maize, that has been mainly propelled by demand of maize in the poultry industry. You can see that this is currently around 50% of the maize is being consumed, which is further going to be increased. And by 2030, our demand is going to be nearly 43 million tons. So you can see that there is a reduction in use of maize, use of maize as food. but certainly indirectly, maize is consumed immensely through poultry and other industrial products. Now, we all know that nutrition plays a very important role. If you see out of 17 SDG goals, 12 directly or indirectly related to nutrition. And stunting as well as wasting is a very common phenomenon, particularly if you see Africa or Asia, and more so in India. Now, if you compare the nutrition quality of maize and wheat and compare with rice, certainly they are much better, but wheat has little bit added advantage, particularly in terms of protein quality. Why it is so? That if you take the normal maize, there are two important amino acids, that is lysine and tryptophan. They are quite low. But there was a mutation that is called OPEC2. This mutation increases the content of albumin, globulin, and glutenin, and reduces the concentration of zein. Zein is actually very low in content in terms of content of lysine and tryptophan. So as a result, the overall content of lysine and tryptophan in when the mutant is present in a homozygous condition, increases significantly. So when this was discovered in late 50s or so, then suddenly there was certain use of this, and there are a lot of composites and hybrids coming into picture. But then there came some markers, molecular markers were identified within. So there were some markers which are identified within the location of OPEC2 gene. And then there are a lot of SSR markers and other markers to go for foreground selection as well as background selection. And deploying that in the first, in 2008, if I remember correctly, that time, just our Professor Datta joined as DDG ICAR in Crop Science Division. So the first mass-derived QPM hybrid was released in 2008, which was named as Vivek QPM9. It is a essentially derived variety. So now, I was talking about only OPEC2, but there is another gene called OPEC16. So when using marker selection, when both OPEC2 and OPEC16 were combined, you can see that there is increase of nearly 152% lysine content and 332% of tryptophan content in this dual mutant. And all these have been achieved through marker selection. Now we come to provitamin A. You know that is again vitamin A is an issue, and again Professor Datta worked a lot on golden rice. But when you look at or you try to use any gene for improvement, first you need to know that what is the metabolic pathway. And in this particular case, this CRT-RB1 has been identified as a key gene. So markers for functional markers for this particular gene has been identified, which has been used for foreground selection, and for background selection we have many markers. So using that, then this in 2017, this dual mutant, which is having provitamin A as well as QPM, was released in the name of PUSA Vivek QPM9 improved version. And you can see it has got a better content of provitamin A, tryptophan, lysine, while its yield level is compared to the normal one. Subsequently, now we also know that vitamin E is also a very important component. So alpha tocopherol, if there is increase in alpha tocopherol content, then there is those lines will be containing higher amount of vitamin E. So when the pathway of this has been derived, it has been found that this VTE4 allele, if it is moved in any other variety, there is a possibility of increase of alpha tocopherol content by over 3-fold. So certainly with that we can improve the vitamin E content. And this triple mutant, where or triple biofortified maize, which is having high QPM, or it is a QPM background, which is provitamin A and provitamin E. It has been identified in 2023 for cultivation in Northwest Plains Zone, Northeastern Plains Zone, Peninsular Zone, and Central Western Zone. And you can, if you compare with the normal maize, you can find that the vitamin E content has increased almost by 2.5 times. Provitamin A content has been increased by more than 3 times. Lysine and tryptophan also tripled in these varieties. So all these are happening using marker selection. Now we also talk about iron and zinc. Now you know that maize contains nearly 30 ppm iron as well as 25 ppm zinc, but only 5% of this iron and 20% of this zinc is available, bioavailable, because of high content of phytate. More than 80 to 90% of the phosphate is stored in the maize grain in the form of phytate. So as a result, there is biostimulation. So there are different mutations, like you can find LPA1, LPA2, and LPA3. These mutants are involved in the phytate pathway, which in the mutant line, there is a disruption of the phytate production. So there is less content of phytate. So in our studies, we have used LPA1 and LPA2. And in 2022, first low phytate maize variety, that is PMH1LP, was released. And subsequently, you can find that low phytate, under low phytate background, we have QPM lines, we have QPM plus provitamin A lines, where nearly 30 to 40% less content of phytic acids are located. So you can see that, as I was mentioning, that AICRP on maize was started in 1957, and first using OPEC2 gene composite, 3 composites were released in 71, then there was a series of QPM composites. Then first QPM hybrid came as Shaktiman 1 in 2001, and then there were some other hybrids. But as I was mentioning, that first mass-derived QPM hybrid, QPM 9, was released in 2008. And after that, there was a series of markerized selection-based biofortified maize hybrid, which is having QPM, provitamin A, QPM plus provitamin A, or QPM plus provitamin A plus low phytate. And first low phytate hybrid was released in 2022. So in this way, till now, there are 29 either composite or biofortified varieties have been released through AICRP on maize. But out of these 28 varieties, 18 are having derived through molecular markers, that is DNA markers or marker selection. Now you can see that how much importance government is giving, that out of these biofortified hybrids, there are 5 hybrids which are dedicated to the nation by our Honorable Prime Minister. So often question comes that why we should grow biofortified varieties, whether if unless and until their yield level is comparable to that of their normal maize. And you can see that when QPM plus provitamin A is compared with its counterpart of QPM, or with the normal, or QPM plus provitamin A, or all these are compared with the normal. And this is based on the AICRP data. That those students who don't know AICRP, it is called All India Coordinated Research Project, where multiple locations in the countries, like under AICRP on maize, there are 30 regular centers and there are around 23 voluntary centers. So nearly 50 plus locations every year trials are conducted, and you can see that how many locations these have been tested, and the data is there. So you can find that they are quite often, or currently, whatever hybrids are being released, they are comparable to their counter normal maize in terms of yield level is concerned, but their nutrient level is certainly much better. Now, as we talk that maize is extensively used in the poultry industry. So what is the effect of these biofortified varieties on birds? Then here you can see that body weight gain, BWG, is much higher in the biofortified maize as compared to the normal maize. At the same time, the FCR, that is food conversion ratio, is much lower. That is the food intake divided ratio of food intake and the biological weight gain is much better when you are feeding these animals with biofortified maize. Similarly, the flesh quality or the yolk color is much better when you are feeding them with biofortified like provitamin A rich maize. Similarly, if you see the low phytate maize I was talking about, you can see that when you are feeding this chicken with low phytate maize, there is higher body weight gain, higher P content in the bones, as well as there is reduced phosphate transporter gene activity in the intestine. So obviously, this biofortified maize can contribute directly or indirectly to the nutritional security of the country. Now we all remember that when Ukraine war started, India took a stand that they will not budge to the pressure of the Western world, and we continue to buy our petrol or petroleum products from Russia. It was just, I personally feel it was not that just a global posturing, but it was our compulsion also. But you can see that currently our requirement of petrol is around $40 billion, and which is, as our economy is changing, it is further going to increase. So this has forced our government to introduce that national policy on biofuel in 2018, and then 2022 it was revised, which targets that 20% blending of ethanol in petrol is compulsory by ethanol supply year 2025-26. So that means by 25-26, our production will have to be, bioethanol production will have to be from up to around 8 billion liter, which is a very big target. So where from we can get? First generation bioethanol is only possible. Second, 2G ethanol or 3G ethanol is not yet that commercially viable. So one source is sugarcane, then damaged food grain, maize, and sweet sorghum. So out of this, if you see sugarcane, certainly per hectare, ethanol recovery is much higher. But per unit time and per unit water input, if you take, then certainly in case of sugarcane, it is much higher. In name of DFG, it is often the rice is used, and you know that rice consume almost 3 times that of the water that maize is consumed. So certainly maize is a very good alternative. Now, if you take 100 kg maize grain, it can give us around 40 liters of bioethanol plus around 30 kg DDGS, that is dried distillery grain soluble. So this contains around 25 to 30% protein and around 15% fat, which can contribute to the requirement of around 25 to 30% in the feed industry. So it is able to produce our energy, as well as it is able to cater the need of the poultry industry as well. Now, if you there is a calculation of the bioethanol plants. So if they use the maize vis-a-vis others, there is a possibility of around 700 rupees per quintal net income they can achieve. But obviously, that all depends on the storage cost, transportation cost, price fluctuation, and how much biomass, that is the maize biomass, you are able to put fire in the process of this whole process of ethanol production. Now, why maize to be preferred on ethanol production? Like, as I was mentioning, that the government initially approved use of damaged food grains. But in name of damaged food grain, mainly it was rice grain was used. And it is not that damaged rice grain was used, it is the whole rice grain was being used for ethanol production. So government has now put a ban that rice cannot be used for ethanol production. So now why maize? Maize has high yield potential. It has scope of enhanced production by increasing its yield as well as acreage. Food security is independent, that is, maize is principally not consumed as food as compared to rice or wheat. So that issue is not coming. Then round the year availability, because maize can be grown almost across the country throughout the year. Multiple cropping is possible. Then reduced transport costs. So local production as well as local consumption is possible. It has got, as I was mentioning, that less water requirement, so less environment footprint, and the byproduct, that is the DDGS, can be used in the feed industry. So if the farmers are growing maize from Annadata, they can become Urjadata also. So now if you see the demand of our E2020, so by next year, our requirement of maize or ethanol would be around 1,016 crore liter. So this, if you see that how much of sugar you can use and how much grain you can use. So government has given a target that 50-50. So 50% should be from sugar and sugarcane, and 50% should be from grains. But currently we are having around 80-20 or 83-17. So it is again a very huge target. So now if you see the projection, so by 2025 we need ethanol from maize around 350 crore liter. So which means another additional demand of around 9.2 million ton of maize will have to be produced. So for that, we need to increase the yield level of maize. Then high starch with better ethanol recovery. That is, waxy maize is very much possible. Then quality or better quality DDGS of the maize, so that there is higher demand or higher price the industry can get from the feed industry. And then certainly climate resilient maize, as our DG was talking about in the morning. So if you see, now maize has not remained a mere food crop, it has become a commercial crop. So now if we need to achieve this increased demand, one is land requirement, another is productivity. So if you see the productivity, current level of productivity of Kharif maize is around 40 quintal per hectare and Rabi is around 65 quintal per hectare. But there is ample scope by increasing the coverage of single cross hybrids, popularization of base management practices, and mechanization. Similarly, there is also possibility of yield-led acreage expansion, and there one is the tapping of rice fallows in this region. There is a lot of rice fallows are there. Then upland rice in eastern India, where a lot of rice is consumed, replacement of Boro rice. Then in the western Indo-Gangetic plain, we can replace rice or crop, can go for crop diversification and then intercropping with other crops. So with this, we can bring around 25 to 30 million hectares. Now, when we, Professor Datta, you may be encouraged to see this slide, that when we talk about crop diversification in western plains zone, that is Punjab and Haryana, to replace rice, that is non-basmati rice, we are not talking about basmati rice. So you can see that quite often say that the yield level achieved is not on par with rice, so that maize will not be able to competitive. But here you can see this is not a result of 1 acre or 2 acre. This is 100 acre we have taken in Punjab and Haryana across various districts, and you can see that average we could achieve up to 73 or 75 quintal per hectare yield level in case of maize, and that situation where the maize field is surrounded by rice crop which is full of water, that water is seeping in, but still it is we could achieve. And this is not 2 years data. Third year, by the third year, I left the maize program. So certainly yield realization is possible, but handholding is needed, and outscaling of best management practices, and then price realization. These two, so that farmers can sell their produce effectively, unlike rice where the government is procuring. So this is very much important. So there are different technologies how we can increase the or we can manipulate the starch content so that there is better recovery. So just before lunch, Dr. Arinder was talking about genome editing. So these are some of the targeted genes which are being used to increase the lignin, change the lignin content, or the ethanol, improve the ethanol recovery, or increase the yield level. So that we have now got a good regeneration and transformation protocol. So similarly, best management practices, I was talking that now there is in agronomy, there are a lot of chemicals which have come, which can be used extensively for managing the weeds. Similarly, mechanization is another area which is, in particular, so far maize is concerned, has not received much attention. So with this, I can conclude that maize production is steadily increasing with steady increase in area and productivity, and it can contribute immensely towards crop diversification, whether it is in East India or Northwestern Plains zone. Though there is reduced use of maize as direct food, but biofortified maize can significantly contribute in nutrition directly when it is used as food or processed food, because now maize is consumed extensively, not as directly food, but the processed food like cornflakes and others, and indirectly through by promoting this poultry health. Then maize can contribute significantly in energy security with little conflict with food security. But in this whole process, the key is best management practices, including popularization of single cross hybrids, weed management, crop production technology, that is mechanization. But policy support, sir, I didn't put that slide particularly. I did an analysis that how many scientists or how many technicals and what is the overall funding to rice or wheat vis-a-vis maize. If you see, it is really, there is mind-boggling difference. So certainly, if we need to harness the full potential of maize, we need to invest or change our policies so that the maize receives its due importance. Thank you.
Thank you very much, Dr. Shuja, for giving again another excellent talk, and everyone is definitely convinced now that how important the maize is and what an industrial crop today, and in future how the ethanol production or bioethanol production would make a big difference of less importing and giving supply to the country. One or two comments or questions from the students, particularly from the students. If you have any question that you want to know or you want to throw some challenges from the students. Anyone? Can I ask a question? If I see that if there is an interest of the others. As you mentioned of the QPM maize, and you know that QPM maize is very important for giving better protein and nutrition maize. And there is variable lines that has different levels of QPM. Now, with the very high levels of QPM in maize, in some lines, do you see any pleiotropic or any yield differences or impact, or impact on taste or other sort of issues?
So recently, we have published a paper in Frontiers of Nutrition, where we have clearly shown that there is no change in the taste of the chapati, quality, and other aspects. Pleiotropic effect as such, not much reported in maize, and as you've seen, that I put one slide where I have compared the yield level of maize with normal maize, QPM, or this biofortified maize with others. So you can find that there is not much difference in terms of yield. But yes, sir, as quite often in the process of release of this, when we talk about this EDVs, we always compare with the original variety. But by the time, suppose a variety was released in 2008 or 2009, and then we have developed EDV, it will be certainly be released. But by that time, there would be many other normal maize hybrids whose yield level would be much higher. So when I was leading the maize program, I made it compulsory that in the trial, we'll have to keep the normal maize also as a check. So it need not to be surpass that normal check, but it should be on par, so that the yield level is sufficient. So as such, we don't find any pleiotropic effect. Thank you.
Thank you, Dr. Lokhi, for that wonderful lecture. We move on to the next lecture of today, which will be delivered by Dr. Gaurango Kaur, Director, ICAR Central Research Institute for Jute and Allied Fibers. And can I request Professor Shapur Bhattacharya to please introduce the lecturer, sorry. Before he does that, I will request Professor Shapon Datta and Professor Rita Kundu to please do the honors and felicitate our speaker.
Felicitate Dr. Kaur.
Okay. Is it necessary to introduce in like a video?
Yes.
Good afternoon. I do have the privilege of introducing Dr. Gaurango Kaur, who is director, ICAR Central Research Institute for Jute and Allied Fibers, Kharagpur, Kolkata. His research areas are quantity and quality improvement in jute and allied fibers, allied fiber crops, land use in cropping, cropping system characterization using remote sensing and GIS, crop growth modeling, water and watershed management, climate change research, mitigation, and adaptation. He had been the principal scientist, ICAR-INDIAN Institute of Water Management at Bhubaneswar, India, from 2010 to 3rd March 2020. He received many awards. He has received many awards. The most prestigious among them is USDA Norman Borlaug International Agricultural Science and Technology Fellowship Award 2008. Other awards are ICAR National Fellowship 2011, Fulbright Senior Research Fellowship USA 2011-2012. He had been the secretary of Indian Meteorological Society, Bhubaneswar chapter from 2012 to 2013. He has received the Rajdhani Samman Award 2015 and received DST Societal Innovation Award of 2020. He is a fellow and member of several societies, among which are Indian Association of Soil and Water Conservationists. He is fellow of that. Association of Agro Meteorologists. He was a fellow in 2012. Fellow of Association of Agro-Meteorologists, FAAN, 2010. Fellow of Indian Society of Soil Sciences, FISSS, 2018. He is fellow of West Bengal Academy of Science and Technology, FWAST, 2017, and he is fellow of currently from 2020. Was elected fellow of Indian Society of Coastal Agriculture. So that's an introduction, brief introduction of Gaurang Kaur, who is going to give us the next lecture. Thank you.
Thank you very much. Good afternoon to all of you. In fact, I was quite acquainted with the scientists of this university a few months back only. And of course, it is through our mentor, through our guide, Professor Sharma Sahab. And we find that today our students also are attending here. And I will say that they are finding second home in this university. So there is a lot of scope. What I mean to say, this is a lecture, but it is not delineating, or this is not the last or the least. I will say it is good beginning. And it is the beginning that I don't know how many of you have visited this Institute of International Etudes, and the potential is enormous. Earlier we used to say the sky is the limit. Now we say that Mahakas, the space is the limit for this institute. And what we find here that we belong to ICAR, Indian Council of Agriculture Research. But this is
very, very pertinent to botany. So what we find here, when there is a application part, today also we are talking about the application of botany, application of plant is agriculture. Whether it is and why I'm pointing out this, we know the 5F, and all botanists should feel proud of that the 5 necessary F for life comes from botany, comes from plant. And when applied, it is agriculture, like food, feed, fiber, flour. And this is the strength of the botany. And that means our sustainable development, growth, everything to some extent depends on these 5 F's. Besides that, we talk about the mitigation and adaptation through climate change and ethical point of view, how the crop is agriculturally important. But these crops is additionally, I don't know how much our botany or any botany syllabus of any university or college or school has been covered by these 7 natural biodegradable species, for which our country is very rich in genetics, technology, and today we have commercialized 3 varieties in front of our DZ, and still, 95% of the world's seed requirement comes fulfilled through our country. You have seen the product. We have actually bought some sample. In which way? We talk about the plastic removal. We talk about the use of natural fiber. Even it has come in the agenda of the G20 declaration. So if we promote, what I am emphasizing that, because we treat this important crops as a marginal crop, non-traditional crop, non-importance crop, but it is a very much cash crop. And we have seen that how that, farming community, the women community, the self-help groups can benefit from this type of research and from the product. So I'll say that to some extent that unemployment problem can be solved. So presently you see that jute, kenaf, roselle, flax, shanef, sisal, and rami. I don't know how much botanical research has been covered and which books has been covered. Maybe it is confined still, but I will request that all you please visit these institutes and study the potential of these crops and take up this as a curriculum also to teach that this is the substitutes of the plastics and synthetics, not only in the country, but also throughout the world. And we have seen that since last 2 years that export has been developed. That means the outside country, particularly USA, UK, France, Australia, they are taking it really serious. So you see that presently we do a lot of 150 to 180 crores of seed business. 5 million farmers are engaged, 3.7 lakh factory employees, and defense of livelihood, exports of jute products also this year 38 crores. So this is, huge amount, because the crop area is very, very less, about 6.5 lakh hectare. Whereas in rice, you see 42 million hectares. So in that case, the export is very high for these products also. But again, this has not only economic value, but also ecosystem service value, as well medicinal and immune system service value. So now, Dr. Datta is here. And in fact, under his guidance and mentorship, we are exploiting each and every aspect, whether it is a fiber or it is ecosystem service values like Dr. Suya Rakit was talking about bioethanol. We are exploring that bioethanol from jute stick also. And it is a carbon footprint is very low, but carbon sequestration is very high. So that case, this is good candidate for your carbon marketing also. 15 ton of carbon dioxide absorbed by 1 ton of jute plant of 120 days crop and releases 10 ton of carbon dioxide. So in that case, there is a lot of scope of research for the student because these are asset in the country. So why we not extensively use and do the research and show the other parts of the world that we are enriched with this type of crops. So, and in fact, it is the proposal from this university also that we are opening one society, Indian Society of Plant Science. So where there is a lot of scope for collaboration and a lot of thinking for the research, in which line we can do that in applied aspects, basic aspects. In that way, we can do that 1 + 1, not 2, but 11. So this is what is the challenges in case of jute now, because so far, to some extent, the research has been covered in the jute. And that's why I'm telling that there is a great challenge because area is going to not increase. Rather, production, high production is harmful for this crop. We astonish. So whenever we are having good seed, good technology, but if the product crosses 70 lakh bale, it will be surplus. So in India, present requirement is 70 lakh bale, but our production within the even diminished area also because productivity is increased. So in the godown, the excess production will be 30 lakh bale in every year. And here the policy intervention is required. And if there is an excess, because it is a high intensive crop, expenditure is very high, farmers cannot get the MSP, then farmers will lose this, cultivation. So ultimately now, our research is confined to the quality. And if we do that, diversified products, like, bags and decorative items, we can do additional lakh bale for jute diversified products. But it is our challenge. Why not? We will have full potential, which is locally available in spite of having all side of technology. So that's why when you talk, why do we can target? Because one target is the breaking yield barrier. So presently we have that existing 40 quintal per hectare. Can we do the 50 quintal per hectare? Because we don't need much area. That's why you can say it is just like a vertical expansion. Then we can bridge the yield gap because there is a difference and improving the fiber quality. And here in 1 and 3, and also in the 4, there is a need for genomic improvement. And that's why today I am humbled and it is my pleasure to attend this lecture, Dr. A.K. Sharma Memorial Lecture on this, because here we can do genetic improvement through the jute and allied fiber crops for what we want. We need the breaking yield barrier, we need the improving fiber quality, and also the herbicide tolerance and climate resilient agriculture. So that's why we know that the quality, if we improve the quality, then we can have market in the outside the country, in different parts of the world. So you see that now we are working on this 5 research program. So the students, you just see that, and every program is now important in the context of today, whether it is a climate change, whether it is ecosystem service value, whether it is a diversification, whether it is a profiling, immunity boosting, and nutritive compounds we have found from the jute leaves, rosal juice, even flax seeds. There is a lot of opportunity to utilize this byproduct also. So in this way, if we link the national program also, it is very much fitting. Then where is the lacuna? Why you are not going ahead with these crops? So also besides this, we need the pre-maturing flowering tolerant jute variety, because this is important. Because in the jute, the sowing window is very, very narrow. And if it does the prematuring flowering, it is harmful for fiber production. So how that now our genomics improvements, our crop improvement scientists, they are working day and night. And how genomics enable improvements can do our target, which we wanted, and what is their possible application. So there are different approaches, basically starting from genome sequence, the annotation, the maps, the transcriptome, the gene expression data, genomic markers as we talk about from today. Then we have identified the candidate genes and genetic markers. So this is the base for approaches which we do that integration and utilization of the genetic resources for the improvement of the jute and allied fibers. So if we see that the genome sequencing, what is the progress at CRISP on jute and allied fiber? We have done in the genome sequencing of the CRISP SONA, that is microbial rating accelerators, to improve the quality of the fiber. Genome sequencing of JRO524, because this is the basic or backbone for any type of genomic improvement research. Also, we did the sequencing of the jute mitochondrial genome, of course, in partnership with the ICAR and IPB New Delhi. Our team has worked on this. And also the transcriptomics at CRISP, what we did here, the bust transcriptome of white jute, and also the hypocotyl type transcript of the white jute, flower transcriptome of Tosa jute, and also that, they developed the SSR marker around 10,000, essence is identified around 5,000, and TFs characterized more than 2,000. So basically what we did here, what we need, the lignin is the one of the candidates. If we can reduce to some extent reasonable amount, your rating duration will be reduced and the fiber quality will be. Similarly, the pectin biosynthesis pathway, beta-galactosidase pathway, and aquaporins and heat stress, HSF and NX factors, which is for flax. So this is basically for quality improvement, and to some extent that abiotic stress, we can minimize that. So ultimately, we talk about the application aspects, because how we can do that, the basic research can be linked to the application. It may be the low lignin jute fiber, and it is faster rating. And I have talked about that. Now we have to improve our yield through vertical improvement, means breaking the yield barrier, because that those areas can be utilized for other crops, for other varieties. But what are the requirements for a year we can do that, breaking yield barrier through faster growth and stress tolerant varieties. So this is the genome sequencing, the report of this. This is the first whole genome sequence, as you know that, for Capsulas eritorius. And in this university, under the leadership of Dr. Datta, has also done a lot of research on these aspects. And of course, that, this is the results on, we have published it, genomics data, the draft genome of Calcutta eritorius. And this is the genomic syntax relationship of Calcutta eritorius with cocoa and rimoni. So this is the, in fact, we have also done that comparative genome assembly statistics for sequenced jute genomes, whether it is an Indian initiative and Bangladesh initiative and Chinese initiative. So what I mean to say here that we have done some basics, but as for the need, we have to progress and target the specific trait, in which way we can, improve the quality and improve the, fiber to some extent the apparel quality. And now 250 number of items have been prepared from jute and allied fibers. And that is, huge demand, which can be substitutes the plastics and synthetics. So those are the students here. They should take place that we will not use the plastics and synthetics, where we are very rich in the natural fiber. We are having natural fiber institutes, natural fiber technology, and farming community, land and industry. Only if we need the policy. And nowadays we talk about the startup. Many students now, even the IIT students, they talk about the startup. And this is the one area which can start the startup. So this is the first report of the jute microbial genome pasteurization. I have already told that. Then also the RAD and SMP-based population structure of African Indian jute, also we have published. And then this is important works we did that because, the jute rating is a microbial process. So we have identified the 3-4 bacteria and their nature, their characteristics, because it should be the pectinolytic bacteria. It should not affect the cellulose. So that's why it is important study that genome comparison, which have identified the different Bacillus species in a biphosphoratic bacterial consortium. And with this, around 15 lakh farmers so far have used this product. And they have got the benefit that improvement of the 2 grades fiber quality and the reduction from 24 days to 10 days. So this is again published in the Springer Nature in the scientific report. And one of the biotechnological tools and sequencing we have important. And again, that broadening the genetic base, as you know that the jute and the genetic base is a very, very narrow, and also cell impoundability. And that's why that we have tried to the broadening sheet base of the jute and allied fibers. And for that, we have genotype diversity and the population structures of jute and SANEP. The SANEP is another fiber crop, and the magic populations also developed. The mutant line also developed from creating additional variability and the jute. Then also we have tried the interspecific hybridization of the cultivated and wild species and the pre-breeding program, because already that a lot of money is spent, particularly in agriculture, for controlling the biotic stress based on diseases. And in that aspects, we have started the research on pre-breeding so that we can, through back crossing, we can take that characters into developing the varieties. And in that case, we have now started a lot of pre-breeding program in the institutes. And the first fertile sexual hybrid also developed in our institutes. And this is the results. Now we told it is a Capoli, also interspecific hybridization. And we have given to the All India, ABT trial this year, hopeful that it will, some lines will improve the yield by, 30 to 40%, 50% yield. And this is the major candidate to improve, to break the yield barrier, in the jute and allied fibers. So this I have told that, the pre-breeding material they are developing. We are directly utilizing the wild accessories. Also, we are crossing with the cultivated varieties with the wild varieties, and particularly the prematuring flowering intolerant. This is a super problem. So what I mean to say is, we have identified the problem. While we can do that, we are addressing to the traditional farming systems, but now when the genome editing tools and genomic improvement ambience has come into the picture, we can develop this now, we can utilize through genomic material improvement, and it has direct use of that, like, particularly the stem rot resistance disease and hairy cattle resistance disease. So this is, again, I have told that prematuring foreign intolerant genotypes. And today we have one variety commercialized JROIB3, and that is to some extent the prematuring foreign intolerant. And last year, this variety has saved that I have already told that sowing window is very, very narrow. If farmers sow early, there is a prematuring fire. So this is one of the undesirable characteristics in the jute cultivation. So to some extent, that variety is solving that problem. So this is the some study of the hypocotyl transcriptome decoded. We have done it and it has been published also. Then here that provides the first clue that how the beta-galactosates influence hypocotyl development in a fast-growing plant. And it also helps in designing faster-growing jute varieties. But again, we have to go miles to go before we slip to utilize this variety. And recently, we have started the research in flax. In fact, we have got one project on genome editing because flax is such a crop which don't require the rating and very easygoing crops. But one thing is that we cannot, so far, it was actually really, it needs some cool climate. So heat shock factors we're identifying, and we can grow the flax in everywhere and anywhere. It will reduce the import. Presently, we are totally importing. And there was no farms for commercialization. And today, on first non-jute variety, that is flax, we have commercialized today. And he has taken huge amount of seeds, winter seed from us, which will reduce half of the import. So this is one good news that few days back only in the meeting Delhi QRT Info, I told that is that we are talking about the dissemination of the non-jute variety or allied fibers. And today only we are commercialized, and one big seed company, he has come forward and he's telling that it is my duty now to stop the import. Presently, we are importing from Belgium the linen. We make the linen salt from this. And also now the flaxseed, every diabetic patient is taking the flaxseed. So there is enormous opportunity for the crops and for the institute itself. So this is the new initiative. I think we are in the under genome editing projects. We have tried to produce double high-protein breed lines within just 2 generations, and the heat shock induced transcriptome analysis in the flax. And this is the details of the crops to be, we are targeting these traits, the early flowering traits, reduce lignin good fibers, and herbicide tolerance, because this is the traits we are targeting in jute through genome editing projects, and also the flux traits, the Floyd inducing lines for rapid varietal development and terminal heat stress. So ultimately, again, till date, the CRISPR-Cas9 genome editing tool has been not applied for jute genome modification. Dr. Anand was presenting for different crops. So here there is again challenge to our scientists. We have got that project, and ultimately, if we can success that particularly low lignin jute variety and resistance to biotic stress, tolerant to abiotic stress, heated water stress, herbicide tolerance, because this is one problem. Because in jute cultivation, farmers actually a lot of expenditure on the weed control. Then multi-trait edited lines, and ultimately, what we need at this juncture, the improved quality and improved rating. And I am it is to be informed that India and Bangladesh still dominates in the world market in jute, 97%. But Bangladesh depends on us. On 95% of the seeds we export every year. And so this is the magic of the jute and allied fiber crops. But we are having another 6 crops which are equally important and a lot of potential in different sectors, like whether it is automobiles, like CISL. Now people are walking the earth-prone world, and a lot of business is coming in the real estate. So these are the things I don't know. Revamping the policy and attention to this type of crops where we have locally actually strong. So vocal for local, I mean to say that. And now these are the already people have studied in outside the country, the lignin content in the jute fibers, it has been published. So our road ahead. So this is, we are targeting now. So that's why, like herbicide tolerance, the strategy, transgenicity of editing, low fiber lignin content, reduce pectin content, the historical fiber content yield, stem resistance. So again, we are talking about the water use efficiency, but scope is there to utilize that for the seashell as well. So these are the some key questions for herbicide tolerance. That what are the ideal targets for conformation and or biochemical changes? So these are the some of the issues for the researchers as well as the students. But my urge is that most researchers, at least it should get this crop should place in the bottom. And when we apply this, it can link to the plant science and agriculture. So I think, thank you very much for your kind listening. So this I can link. And I am the man of the soil physics, but I have got fascination in botany and agriculture. In fact, when I read the civil service, I took the botany as optional and got the maximum marks so that I had appeared in the interview based on botany marks. So thank you very much. Source of interest, inspiration, encouragement. And I think 4 years I am here. We have reshaped our institutes also, and we have developed one museum, and where you can enjoy. Thank you very much, sir, for your invitation. And I acknowledge, in fact, that for inviting us for this type of important event, though it is a very small, but small is always effective for future implementation and for sweet remembrance or short connection. Thank you very much.
Thank you. Don't go away. Just wait here. Okay. Take one chair. Thank you very much, Dr. Gauranga, for a very fascinating and very important seminar or talk that he has just delivered. And with his talk, you realize that it's not the structural genomics, the functional genomics or improvement of traits and ultimately how it helps in industry, how the botany or the plant science, the different varieties of jute and allied fibers he showed, all the plants are basically of botanical science. And then its application, that in the morning we heard that the application of plant science is agriculture. And this is the similar thing. I think I'm glad that all the speakers, they put their presentation in such a manner that the basic understanding of science as well as its application and towards the industry and economics benefit. Food, feed, at the same time, fiber. All of them are covered in our presentation today. So now the final, last exciting sort of discussion that we are going to make, and that's a panel discussions. And this panel discussion, we'd like to have it very open type. So I would encourage the students to raise questions, any questions you would like to ask, or you have in mind that you would like to clarify. You can challenge the speaker, you can challenge yourself and challenge to the podium, to everyone. So in that context, we have selected a few people, but it's open to everyone. Even you are not in the dais, but you are also part of this panel discussions. So Dr. Kaur is with us. Then I ask Dr. Sujay Rakshit to join, Dr. Anindya Bandyopadhyay to join, Dr. Professor Sudhendu Mondal to join, Professor Sharma Das. Professor Sharma Das, please join. Hey, kata chair di debe, kata chair bari do, 8ta chair lagbe. Professor Sharmila Chattopadhyay, is she? Sharmila to join. And Bardhaman Rajiv. Rajiv, তোমার টাইটেল আমি ভুলে গেছি, কি টাইটেল? ও বন্দ্যোপাধ্যায়, এত বন্দ্যোপাধ্যায় না চারিদিকে, বন্দ্যোপাধ্যায় রাজীব চলে এসো বর্ধমান ইউনিভার্সিটির পার্সপেক্টিভে। কজন হলো? Who else? We are missing? অনিন্দ চলে এসো। প্রফেসর কৃষ্ণেন্দু, এই অর্গানিক বলবে না কিন্তু, ইনঅর্গানিক বলবে। চলে এসো। শর্মিলা তো আসবে, হ্যাঁ সবাইকে বলেছি। শর্মিলা চলে এসো। ও ইয়াম শুভদীপ মজুমদার, আরে ভুবনেশ্বরের ফ্লেভার কিছু একটা দাও এখানে। ভুবনেশ্বরে কি করছো, মানে স্টুডেন্টসরা কি চাইছে, সেখানে কি রিসার্চ করছো, একটু কিছু বলো, সবাই দেখুক। তুমি ওখানে বসতে চাইছো, বসো, চেয়ার হয়ে যাবে। আমি তো বসবো না, আমি তো এখানে দাঁড়াবো, আমি তো হুইপ চালাবো। দেখো কটা চেয়ার হলো, এখানে একটা চেয়ার আছে, এখানে বসো। না আমি বসবো না, এখানে বসো। কোন সামনে? এই সামনে? হ্যাঁ তাও দিতে পারো, কয়েকটা চেয়ার সামনে দিয়ে দিতে পারো।
ওই দিক থেকে এদিকে বসে আপনি দেখে নেবেন, বেশ ওই দিক থেকে বসিস, বেশ ভালো হবে। ওই এরকম ভাবে বসলেই হবে। 500 টাকা ঝুলি
এই দিকটা কোথায় আছে? Or I give you open challenge. Anyone want to be part of this side? Also raise your hands. You are welcome also to join voluntarily. Anyone, student, teachers, scientists. এই সবাই নিচেই বসলো? I would not see anyone. একটা হ্যান্ড ব্যাংক আছে? কাজ করে? Fine, no, that is all right.
ব্যাক ক্যামেরাই করে দিয়েছি। ব্যাক ক্যামেরা
Okay, the next that what you like to do, what's the issue that you would like to discuss? First part of the discussion, we would like to have prospects of botany and science. What we are doing it, what we should do it, and how that should really help us in advancing our science and understanding of the subject. Prospects of botany and plant science, or plant science or botany to the prevention of other areas of science, including agriculture. What I have heard today, the DG said himself, plant science or botany actually basically its application is agriculture. Finally, the moving towards any development, that's agriculture. And Professor Shaminathan, even PM Bhadri, they all are basically Shaminathan's degree also basic science and then moved to agriculture and joined even cytogenetics. So that's a very interesting thing that we could learn, we could see today. Now, I think who should start saying a few things? Okay. Okay. I think this part has a large number of scientists, teachers, and professors are here, and this part is. Don't expect that you are only listening. You also interact. It will be more fun and have interaction and challenging anything you want to. I think one of the important component is tomorrow there is election results. So election results will be very interesting, fascinating, unpredictable or predictable, irrespective of outcome of tomorrow's result. Science doesn't have a boundary, neither science has any limitations. It would prevail the truth only truth. What Swami Vivekananda always said, his version. Now the truth is based on hard work and experimental and evidences and based on facts. It might change. The truth in science also might change. One of the things that I remembered when I was young lecturer at Vishwabharati, was the two papers of our time published in 2 consecutive years. And the second, both papers published in Nature. I don't know if Shangila remember it not. I used to tell it to the students at the Vishwabharati. And the second paper, the first sentence of the abstract says, I was wrong in my first paper published in Nature, published in other 1 year earlier. And what was wrong, he explained again what is right. And it was on sieve elements, transportation of water through sieve elements. That is science. It also can change, but you have to be honest and to be true. And truthfully, you have to explain that what it is. And that's the beauty of science. So science does not need to be afraid of telling that I was wrong or I did the wrong thing or this experiment was wrong because of that. Interpretation is important. Why it was wrong, why it didn't work, that has to be interpreted. And today, I think about the scientists, particularly students, in the respect of students, most of the students are average students. 10% or 50% students or 20% students are good students, or a few percentage would be brilliant students. But we have to deal with the more, majority of them are general average students. Now the average students or general students all over the world, including India and Bengal, so at the moment they have a real struggle because the struggle of new opportunity of jobs, struggle for looking for new dimension or something which is missing, it is entrepreneurship. Today's from many talks, particularly Goranga Kaur and others, they have made a lot of issues of the entrepreneurship. DG's talk also made a lot of things like that. But now, in this context, our students are struggling to look for their future career, to look for their future development. What we should be doing or what we should be telling to the students that how and what we should be looking for, and in the context of this present uncertainty, that how your studies, how your research, how your mind could be built up for looking future aspect. I think I would like to see some of the members to highlight that issues so the students can be motivated and encouraged that what they should be looking for, irrespective of outcome of tomorrow's election result, or irrespective of what is the political scenario in Bengal and in India. But if I believe it, other day also in the Ramakrishna Mission seminar, some of the teachers took me to interact with some students to give them some advice. I told them only one thing, just study. Do study, study, and study. And then that would help you not only understanding your work and science, what your commitment should be, but at the same time, that would lead you what is the opportunity and what else you should do. Do not look for any jhanda. I said it very categorically. Do not look for any jhanda. Do only concentrate yourself in study. When things are bad, this is an opportunity for the smart student or smart people to concentrate on work and concentrate on only think what you can do the best. As a student, you have the best judgment of your study, best judgment of your work, and commit yourself with that one and look forward. You would find an opportunity what is best for you to come. So with that, I stop and I ask anyone first time. Dr. Kaur, okay.
Thank you very much. I think here I think a lot of students, and as Dr. pointed out that the condition of the job, the education versus job, or correlation between job and education may not be after 10 years, 20 years, like this, what we doing today, or 10 years back. So, before that, I think that every country, every student, whatever may be the stream or your background, you should know the country's resource, country's challenge. And in that case, I don't know how many of the students will know that. But India takes about the burden of 17% population of the world. Whereas we have only 2% of the land share of the world and 4% of the water share of the world. So you see that how much tremendous pressure of our natural resources, particularly the soil, land, and water. In one way, our population is increasing. We cannot have any type of technology to adopt or mitigate that population. So your denominator is always, will increase. The numerator is soil, water, land, productivity. So whatever the scope is to increase your numerator. Otherwise, your fraction will be the less. So this is a great challenge, how to maintain that fraction. And not only in the quantity, but also in the quality. The quality of soil has been declined. I am just giving perspective in terms of plant science or agricultural science. The water quality has been declined. Total soil productivity has been declined. Tomorrow, that 10 years back or 5 years back, by utilizing 200 kg fertilizers, what productivity we see today or after 10 years, we will see half of that. So this is called total soil productivity has been declined. So this is a great challenge. In one way, our population has been increased. In another way, our natural resources have been declined, and both quantity and quality. Our food quality has been declined. We are changing our diet. We are changing from vegetarian to non-vegetarian. Water footprint is increasing. Carbon footprint is increasing. Energy footprint is increasing. Have you analyzed, even from your curiosity, any student, what is your water footprint in day-to-day life? So this type of thinking now is very much required. So that if you want to save your resources of the country, your education level, your thinking level will be application oriented. And if you think that application oriented to save the resources of the country or to improve the productivity of the country, to reach your technology to the masses, in that case, you will, build some sort of mind in the startup. Now concentrate of, every aspects of the potential of the technology. Today, the medicinal plant, many hormones, many alkaloids, these are the source of the medicine. Ultimately, the startup will be the solution in the future. But in which way? That's why now our education policy has been changed. So every aspect, every page, every line, every word of your education should have some linkage with the application. And it should improve the productivity and ultimately, if we stand the plant science with the service sector, it is our great challenge. Because today our Director General has already told that climate change is again another dimension. Otherwise, one of the resources have been declined, population has been increased, increasing and your climate change in another dimension. So I request that your thinking in the curriculum, your research, your thesis topic should be, suppose you are taking one project, beforehand you have to think of what is the utility of this project, which research paper will come from this project, and what is the utility of a research project from practical point of view. In this previous point, I think we have to go ahead because the job opportunity, education, there should be not much correlation in the group. Thank you very much.
Dr. I think one of the aspects that we are dealing with industrial interaction, as you are very much involved nowadays with many industry. If some sort of thoughts that how this area of science or whatever plant science and botanists are doing, how they can interact or move toward the industrial side.
So, thank you, sir. So this is again another solution. Because ultimately, if you, the student of botany, you will see that people say 5 F's. And within this 5 F's, food, feed, fodder, 12, 5 F's. Many F's are industrial crop. And presently, you can say that we underestimate the industrial potential of this 5 F's. We concentrate, of course, the food to some extent fodder, but other F's is say what is neglected.
I make it more challenging. I'm putting a question now. As in the developed countries or industrial countries, the industry or industry entrepreneurs they put a lot of money for R&D research in all aspects, including plant. Why in India the industry doesn't come or doesn't pay at all any money or investment in plant science or in general of the science?
Yeah, so it is very much true. If you see that, what is the need in apple cultivation? What is the research over is to the apple grower will sponsor big projects to the university. But this is not true in India, whether it is grapes, whether it is fruit, whether it is apple. Because sir, at top level, the policy level, the marketing and linkage to the consumers, it is there is a missing link. Now, in our country, there is a limited requirement of everything. And one thing is that if there is a shortage, in spite of improving our infrastructure, in spite of improving our industry and industrial outputs, in spite of modernization of the manpower, training, and modernization of the instruments and equipment of the industry, we are ready to import the things. Like I told that the linen fibers we are importing from the different European countries. Instead of we are improving the infrastructure in our country, in spite of our technology, in spite of our scientific manpower, in spite of good technology. So this is again that industrial requirement, because if you produce more in the market, the marketing is again the problem. So this is we find that this is again led to that. Because industry, I have found in the jute sector, they don't need even good research because they are telling if you see good technology, good variety, your productivity will be increased. And who will take? No, in jute sector, I have told that same question.
single person. Scientists working in the lab should come forward to show their results so that the industry, new industry, they will be connected. If we know the plant extract, medicine, you know, the cheapest medicine for heart attack or something like that. We take aspirin or aspirin. What product is produced? It has come out from the bark of willow tree. When it is known chemically, they synthesized organic chemicals from the plant extract. They did it. Okay, and now in the lab, they have synthetic chemistry originated how? Only the synthesis of these plants going from the plant. Today we are celebrating, I'm telling you for the students, we are celebrating the birth centenary of Professor Sharma, who worked in this department for more than 50 years in the field of chromosome study, that is psychology. He was born in 1924. If you go back to 100 years, you will get the person Gregor Mendel, 1822. He worked on the pea plant. You know, in the cultivated field, how many plants he cultivated? Getting the trait, the factors. 28,000 plants he cultivated. Simple graduate, a school teacher, no degree in botany or plant sciences. Then he used to get that trait responsible for dominance and the resistance. Cloud color, pot color, light. But his results are not approved by the even scientists after 1900. And that factors, which are nowadays known as the gene sequence node, why it is located in the chromosome. While they are 1888, first time coined the term chromosome. We used to know it. What is a chromosome? Chromosome is a linked structure consisting of a linear arrangement of factors, which are nowadays known as genes controlling the various individuals. After this, Mendel's work on the genetics, all of the scientists came forward. In Indian scenario, if you see the two plants, that is leguminous plant, Pisum, and then selection of the plant. Professor M.S. Swaminathan, why he is so famous? He has 3 daughters, and he selected 3 interesting plants which are needed every family, every day, all over the world. Rice, wheat, and potato. Every family, every house of the world. So selection of the material. That is the role of a scientist or your own thinking. As a student, you have to think it, what material I do. I have to keep a target. Yes, I will go for research. I will not take any technological job. I will try my thinking, taking out some findings in the lab so that I can go abroad to have vision with all of the living science. This is the way. You see the Bengal scenario of the scientists. One generation. Now this force popularized, make them then was that which is that group is over. Young generation, Eke Sharma, Ashima Chatterjee, Archana Sharma, another group of scientists. That group is over. Now the next phase, Dotto and others, young group, they are also going to over. Young should come up. For survival of the human being, plant science are needed. You know, globally about 350,000 plant species. And in India, we have 55,000 plant species. How many are known on the basis of genome analysis? Suddenly, we got Arabidopsis. Small plant, 6 weeks duration, easy to handle in the lab. Scientists, molecular biology, because genetic engineering came in 1970. That's not immunology. Biotechnology word came in 1980. So gradually, you see from 1820 to 2022, 200 years. Indian talents, I'm telling you, not only the botanists, geologists, agriculturists, India is self-sufficient with agricultural production. India is having the technological, we are member of the space club, we are member of the nuclear club, we are member of the Arctic club. So Indian students, our teachers, the way teaching is done in India. So for the students, one science, studying plant, you can reach up to the climate. If you have a chill and if you have your commitment, because in chemical industry, there are many waters. The factory, other places, always there. CSR, ICAR, having many waters there. So that we should be aware, because genome analysis for all these important crop plants, another 5,000, what is needed, it should be done by India. If organizations are there, they are helping, so we should have to think the way we must. Thank you. একটা লাইন আমি একটু অ্যাড করে দিতে পারি। I would like for the students. Today is 3rd. 4th June is significant for 100 years of the letter written by Satyendra Bose to Einstein. First research paper Satyendra Bose has written to Einstein because তার আগেও দুটো পেপার ক্যান্সেল হয়ে গেছে। যে জার্নালে দিয়েছিল ইজ গুড ফর নাথিং। কিন্তু ওনার ওই চিঠিতে আইনস্টাইন নিজে বুঝেছে 100 ইয়ার্স মানুষ এমনি এমনি বড় হয় না, ধৈর্য রাখতে হয়। প্রত্যেকে Satyendra Bose 1 বছর জব করেছিল, যেমন মাধ্যমিক পরীক্ষার সুস্পষ্ট এই সবাই।
Converted the hunter-gatherers to settled agriculturists in Calcutta. Bengali converted the hunter-gatherers to settled agriculturists. Calcutta. You know the next soil holding started in India. Butter from India, then sugar, chocolate, this soil something. But also in Calcutta, something.
She is also from Shantiniketan. আপনার নামটা বলে দিন।
Natural resource থেকে wild থেকে কালেকশন করা ওটা হচ্ছে অসম্ভব টাকা হবে, ঠিক আছে? এগ্রিকালচার হচ্ছে সবচেয়ে এই বলে উনি সাঁওতালদের এগ্রিকালচার স্টার্ট করেন। তাহলে ঈশ্বরচন্দ্র বিদ্যাসাগর ব্যাখ্যা আরেকটা সবাইকে আমার জিজ্ঞাসা যে আমরা ইনসিনারেশন, এই যে আমরা ফসলের গোড়া ঘোরাচ্ছি, এটা পুরুলিয়াতে আমি থাকি, ওখানে একটা সাঁওতালি স্কুলে পড়াই, সেখানে সবাই ফরেস্টে গিয়ে একটা বিড়ি তুলে মারছে ছেলেগুলো পেশা করে, এটাকে আগুন লাগাচ্ছে, এটাকে আমরা ফরেস্ট ফায়ার ভাবছি। জানি না উত্তরাখণ্ডের বা ফরেস্ট ফায়ার নাকি ওটা
বিরাট বিষয়, ওই বিষয়ে আমরা আলোচনায় যাবো না, তাহলে ওটা নিয়ে আলোচনা করতে হবে। পলিউশন তৈরি করছে, আলোচনাটা আপাতত যাচ্ছে না।
সকলকে নমস্কার। আমার নাম মীরা মৌলিক। আমি ডক্টর শর্মার প্রথম দিকের ছাত্রী, এখন সেই 55-এ আমি ইউনিভার্সিটির দিকে ছিলাম। যাই হোক, তো আমার এখানে দুধের ব্যাপারে এত ভালো নলেজ সব শুনলাম, সবাই মোটামুটি সামনে আলোচনা করলেন, খুব ভালো লাগলো। কিন্তু একটা প্রশ্ন হচ্ছে যে বর্তমান সমাজে দেখা যাচ্ছে যে আপনাদের পলিথিন এবং প্লাস্টিক এমনভাবে ব্যবহার হচ্ছে, জুটের ব্যবহার সেরকম ভাবে হচ্ছে না। তো হাই কোয়ালিটি করছেন ঠিকই নানারকম ভাবে, কিন্তু পলিথিনকে হারাতে পারবার মতো কিছু কি আপনারা ভাবনা চিন্তা করছেন যে আবার আমাদের জুট ইন্ডাস্ট্রি ভালোভাবে যাতে গড়ে উঠতে পারে, সেই ধরনের কিছু চিন্তা করছেন কিনা আপনাদের কাছে আমি জিজ্ঞাসা করছি। বড় সায়েন্স
আমি যেটা we have told already during my lecture that if we treat the plastics and synthetics, initially when there is no plastics and synthetics, we were using the jute fiber. We are using the jute fiber before synthetics and plastics, but after coming again we are not using. But one good information that many countries in the world now they are taking jute products from it, and that's why I told that during last 2 years the export has been done. But religiously, if we want to stop plastics, at policy level, we want to stop production. That is not. So plastics and natural fiber cannot compete. It is cheaper. Plastic is cheaper. আমরা চিন্তা ভাবনা করি ডাইভার্সিফাইড প্রোডাক্ট।
যেটার নাম সোনালী ব্যাগ, এটা অনেকটা প্লাস্টিকের মতো দেখতে, যেটা দুধ থেকেই তৈরি হয়। যেটা এখন বাংলাদেশ গভর্নমেন্ট ওটাকে হাই প্রায়োরিটি দিয়েছে, যেটা তো এটা জুটের যে পাতাগুলো আমরা ফেলে দিই, মানে আমরা স্টেমটা নিয়ে এই পাতাগুলো ফেলে দিই, তার মধ্যে হাই নিউট্রিয়েন্টস থাকে, এটাকে আমরা আবার কমাতেও চাইছি অন্য পারপাসে। তো উনি ওই ফর্মুলেশনটাকে চেঞ্জ করেছেন, আর ওটা থেকে তিনি বানিয়েছেন, ওর নাম হচ্ছে সোনালি ব্যাগ। সেটা অনেকটা প্লাস্টিকের মতো দেখতে, কিন্তু সেটা প্লাস্টিক রিপ্লেস করছে না, বায়োটেকনোলজি।
তোমার নামটা বলো, আমি কোথায় আসো সেটা বলো।
আচ্ছা,
আর তুমি যে কাজ করছো জুটে সেটা বলো।
তুমি একদম সোনালি ব্যাগ বলতে দেখতে
ইন্ডিয়াতে কবে পাওয়া যায়?
তুমি তোমার নামটা বলো, যে তোমাদের যে বলে দাও সবাই শুনো।
আমার নাম আমি আইলেস ভুবনেশ্বরে কাজ করি। আমি অনার্স এমফিল মান ছিল। আমাদের ফেলোশিপটা আমাদেরকে বিদেশ না যেতে দেওয়ার জন্য প্রতি বছর 20 জনকে সিলেক্ট করা হয় ইন্ডিয়া থেকে, যাতে বিদেশ না চলে, মানে ট্রেন আন্দোলন না হয়। তো আমরা একটা 1 কোটি টাকার ফেলোশিপ পাই, যাতে বিআই কাজ করি, আমি দুটেই কাজ করি। আমার পিএইচডি জুটের উপর, ডিগ্রি জুট হচ্ছে আমার পিএইচডি। আর জুটের সমস্ত অ্যাসপেক্ট গুলো যেগুলো স্যার তুলে ধরবেন, আমি চেষ্টা করি ওগুলোকে কিছুটা কভার করার জন্য। তো এটার বেটার অ্যাপ্লিকেশন, এটাই আমার এটাই আমার কাজ। আর ভালো এগোচ্ছে, আমি গুড অ্যান্ড এক্সিলেন্ট পাচ্ছি এই দিকতে। আর খুব কম লোক কাজ করে, তাই সবাই দিকে কাজ করে, খুব ভালো লাগে। আর আমার খুব ভালো লাগে, আর যেহেতু বাংলাদেশ, মানে আমি যদি বিদেশে যাই কোথায় যাবো, বাংলাদেশ আর চাইনা, তাছাড়া কোথাও যাওয়া যায় না। বাংলাদেশ আর চাইনা অনেক বন্ধু, তাই আমরা সবাই ঠিকঠাক ভাবেই এগোচ্ছি। ভবিষ্যতে আমার ইচ্ছা আছে যে আমার ল্যাবে মেইন কাজ হবে, দিনের উপরই মেইন কাজ হচ্ছে এখন। আর বিদেশের লোকেরা আমার ল্যাবে কাজ করতে হবে।
বাংলাদেশের কথাটা শুনলাম, খুব ভালো লাগলো, কিন্তু আপনি যে প্রশ্নটা করলেন, আসল প্রশ্নটা করছেন এটা চিপার হবে কিনা, অর্থাৎ যেগুলো আমরা প্রতিযোগিতায় নেমেছি সেখানে তাদের হারাতে পারবো কিনা, এটাই তো দেখতে হবে।
Unlike my other two panelists, I will not bombard with you with a lot of challenges and other things. But I always will say that if I could achieve whatever I have achieved and could reach where I am, all of you can achieve. But only one thing I would like to stress upon you is that you do what you like, or you like what you do. পিএইচডি তে কোথাও চান্স পাইনি, সেই জন্য জাস্ট পেয়ে গেলাম চান্স, আর কিছু হচ্ছিল না বলে পিএইচডি ঢুকলাম, পিএইচডি করছি। যদি এই মেন্টালি নিয়ে করো, ওকে ডিগ্রি হয়তো পেয়ে যাও, কিন্তু ইউ উইল নট বি এবল টু অ্যাচিভ এনিথিং। So by chance, suppose if your aim was not to do PhD, but you have entered, so what I say that if you like, you do it. Otherwise, when you are forced to do it, you like it. So, and then you should have a passion. So you follow it, whatever it is. Sir was talking about botany. I say that geography, political science, whatever you see, read. If you do it, if you learn it properly, you can excel. So only thing is that you have to be sincere what you are doing. So the biggest problem with this generation is what I feel, that we try to, like when my son was small, he used to, Papa, what would be my salary? We never thought like that, because that time this materialistic thing was not there. Mobile or big cars, nothing was there. In our life, when we used to see a plane was flying, our aim used to be But now, since your birth, you are having AC and all these things. But thing is that you should have that passion, that is whatever I am doing, I'll try to excel. At the same time, I'll always tell enjoy, because this is the only time in your life we have enjoyed immensely. Hardly we studied. Yes, but when we studied very sincerely. So please don't take it very seriously. If you suppose whatever Dr. Kaur was telling or sir was mentioning, If you are sincere, if you have a passion for whatever, then certainly you will go to depth of it. And we will try to find out okay, what are these challenges and how we can achieve it. Another problem is that now this mobile, I feel it is a source of distraction. There is a lot of information. But what happens, we just give a search, Google to Google, and we get the answer, but we don't read. There is no alternative of reading, not only your subject. I always say that whatever you, even if you crash, you get, you read it. Whatever, if you keep reading, not only your subject, have a horizon. And our problem is that from our childhood, we are trained to get good marks, not to acquire knowledge. So please focus on that. Whatever you are doing, whatever your PhD subject, certainly if you are doing your PhD, you will know about that subject. I think there is not much question. What are the related things? That is very much required. Now coming to innovation, this is a problem in our Indian education system is that যে বলে না ঘোড়ার চোখের পাশে আমরা লাগিয়ে দিই যখন লেখা চালান আমরা আশেপাশে কিছু দেখি না So this is a problem with Indian education system, but certainly everybody has seen mental apple is falling from the tree. Nobody questioned it. So questioning, we stop. If you see a small child, when we have small child, we get fed up with questions after question after question after question. Then what we do, we start scolding. The nature of asking questions stops. So this is the problem with most of the students I find that why you are doing suddenly a topic area that I am doing. Why you are doing that, you need to ask and how we can do it better way. So this is the thing. Now coming to innovation, just one example I give you. When I was director of MES, one boy, he was in PhD fourth year of MPUT Udaipur. He developed some from orange peel extract, some hydrogel. So he wanted to have some break. So I also didn't, I was not convinced, but I could see that passion in his, the way he was talking to me. So we let him to join as a startup in our institute. So we just gave him some ways that how he can move. And incidentally, just other day from the Additional Secretary Agriculture of Jharkhand government, he made a call that 2 persons have come to me. They want to get some, they have some product, they want to sell it in to the government scheme of Jharkhand. So can you certify? Then he came and I saw, he asked me that you are, because that boy didn't come, because now he has so many representatives. So is it that person is a yes? Then I was so happy that that person now he has is having patent, Japan, USA, I think Israel he has already applied. So from orange peel extract he has made some hydrogel. So the thing is that if you have some ideas and if you have the passion and you have that dream or zeal to follow it, and you should dream. I just tell, I'm not again boasting myself. শান্তিনিকেতন থেকে যখন আমি আসতাম কলকাতায় ছোটবেলায় কয়লার ট্রেন হতো জানলায় শীতও থাকতো না তো যখন এই থানা স্টেশনে সাহেবগঞ্জ গ্রুপ মেন লাইনের সঙ্গে আসতো বাবাকে জিজ্ঞেস করেছিলাম বাবা ওদিকে কি আছে ছোট ছিলাম উনি বলেছিলেন যে দিল্লি ওই ছোট মানুষ মানে মনের মধ্যে যে কি ছিল যে দিল্লি ইজ দ্য পাওয়ার হাউস I always used to dream that I have to go there. And at that time I was not knowing that what I have to do. But when I came to agriculture, I came to know that Indian Agriculture Research Institute is there. First step was when I went to BHU, that I told myself one step towards it. Certainly, if we think something, certainly immediately we will not be able to achieve, but step by step. Next PhD, I went to Delhi. And I could follow it. I am not telling that I have achieved a big thing, but thing is that now also, suppose whatever I achieve, people say that you will get this one also. I always tell them that whatever has been given to me, if I do it sincerely and if I can excel in that, something will automatically come. I tell you, all of you, that whatever you do, I not say that you do research, you do follow some government jobs, teaching job and anything, but follow whatever subject, whatever your passion, there is.
I think I'd like to take a good 3 questions from the students. The students raised these questions. I don't know who. It's interesting questions. Mostly on climate change. The question is like that. I'm not reading it, rather I'm summarizing the question. It is quite clear. One question is that if climate is wrong, everything will be agriculture, everything will be wrong, and vice versa. So what should we think about for the common people or ordinary people that this adverse effect can be removed or can be changed? It's a bit of appeal that what can be done in terms of so and so. Related question is even more interesting. US from Manshu's our PG slide, it shows that carbon dioxide emission is per capita is highest. And the question is, how could it be a developed country when a developed country like the US cannot control their high carbon dioxide emission? And at the same time, so what should we, again, the question is, what measure or what the measures should be done to address that issue? In this context, 2023, climate, the Y meeting, G20 meeting, what was that meeting? So more than 180 countries attended, even our Prime Minister attended that meeting, PROF meeting. And then in that meeting, Bill Gates gave an interview. Bill Gates made a very nice comment in the interview. I heard that interview too. He said that every PROF meeting, all the Prime Minister, President, all the important people come and they discuss on the climate change and they take a decision for the future meeting. And all the discussions are good, all the measurements and things should be done also good. Concerns are right. The solution they sort of suggest that needs enormous funding, but they do not commit any funding. So the problems is known, solution also being addressed with all the eminent scientists, policymakers, bureaucrats, everybody was there. Who is not there? Everybody is there. Even surprisingly, my son was also there. And the problem is that they are not funding to make it effective solution. Now, these are the, I think, some of the really good questions raised by the student. Who are the students that raised this question? I appreciate the questions very much. Good. And some other questions on carbon trading that Dr. Kaur has addressed. Some other question, nutritional security, and one question also. And another question is 2050. Dr. Borlak had a talk feeding 10 billion population. If you haven't read that review, I would suggest you write down Norman Borlak 2050 will come. The PDF will come and you can read it. But anyway, but the question is here that 2050 the population will be dramatically more. What would be our sort of strategy to develop that, how we can address that question? I think Dr. Pathak talk has given some directions, some sort of things, and everyone said that it's a bit of only solution is they have to continuously doing new science, innovative science. Productivity needs to be improved, and with the limited resources that all are identified. The population definitely would be is under control now, even when number one, but it would remain number one, I think, for a long time. I don't think anyone will beat us, but our production and other things has to be improved. But one of the things that might come, that is future food might have traced some energy rules. So we have to eat rather less, with eating less, but with less food, with the more energy. So we don't need to produce so much of millions of tons of rice, wheat, and maize. Rather, we will be combining with some food that gives more energy. I think this has something. What I'm saying, there's no paper on that. It's just I'm giving my just thought. A question from the students. Anyone still, if you have a question, please raise your hands and just talk. It's very important to ask questions because this is an opportunity. You can ask any question to anyone or a common question, not to anyone. Please, I think it will be good to listen to some questions. Please.
Due to this genetic modification, some crop plants are extinct nowadays.
Some crop plants are extinct because of genetic modification. Never happened. It never. When?
Generation পরে যে একদম ফার্স্ট জেনারেশন হোয়াইট জেনারেশন ফাইভ ভ্যারাইটিটা সেটাকে আমাদের ইকোনমিক্যাল সোসাইটিতে করতে পারছি
I think you are 40 students. Hello, the department. I'm requesting you to allow me someday to give a lecture to them. Right? I will give a lecture on this particular aspect, biodiversity, studying on the genetic modifications, and then after effect, post effect, or how it happened that reduced biodiversity, etc. But we don't need to answer this question. We raise any other. Yeah, please, I'll give a talk. I'll talk on this particular aspect. দু মিনিটে বলা যাবে না এটা কোনো আনসার হবে
wide variety গুলো neglected হয়ে যাচ্ছে এবং আমরা করা হচ্ছে না সেগুলো এক্সপেরিয়েন্স হয়ে যাচ্ছে তা নয় কারণ সেগুলো স্টোর হাউসের জন্য গভর্নমেন্টের যথেষ্ট এখানে সমস্ত ল্যান্ড স্টোর করার ব্যবস্থা তাছাড়াও ফার্মাররাও প্রত্যেকটা পকেটে সেগুলো স্টোর করে রাখা সেগুলো খোঁজ খবর রাখা তাহলে বুঝতে হয় আমি পরে স্যার বড় বলতে আচ্ছা আজকে আমরা Today we have gathered here to celebrate the centenary of our beloved sir, Sharma. And I believe, and I sincerely feel that he was the join of plant science of his country. And he started his research with the staining technique, hydrological staining technique. He started his research. And then gradually he is a man of vision. So he gradually understood and then changed his techniques, and finally at the last stage of our research he was the chairman of our board, and he was always advocating about to search for the diverse collection of the diverse plants and exploring the diversity of plants, because India is a vast country and its geographic location is very unique. In the north, there is Himalaya, and the western, there is Western Ghats, and the three sides, there is ocean. Huge coastal lines. So there is, India is a hotspot of biodiversity. So we have to look for diverse plant sources. There are enormous potentiality among these sources. There are so much of medicinal values and pharmaceutical values. You heard from that maize has some potential pharmaceutical tocopherol and all these pharmaceutical compounds are there, medicinal values are there. So we have to look for that. আমার স্টুডেন্টদের কাছে বলার আছে, স্টুডেন্টরা এখন ভাবছে তোমরা হয়তো অনেকেই মনে একটু লিমিটেশন আছে যে প্লান্ট সায়েন্স নিয়ে রিসার্চ করে কি হবে। প্লান্ট সায়েন্স নিয়ে রিসার্চ করার প্রচুর আমাদের সকালবেলা ডিজি স্যার বলেছেন India is exporting now. Different food crops are India is exporting. যখন আমরা স্বাধীনতা আমাদের দেখে এলো আজকে 70 বছর আগে, তখন we were in food scarce country. Now we are food exporter of many food. So এটা তো একদিনে হয়নি, অনেক রিসার্চ করার ফলে এবং এটার অনেক স্কোপ এখনো আছে। তোমরা প্লান্ট সায়েন্স যারা করছো তারা এগুলো অনেক ভালো। So India is improving in many aspects, not only in the agriculture, but also in medicinal, in medical science, in space science. সবাই তোমরা এখন জানলে যে চন্দ্রযান হচ্ছে, মঙ্গলযান হচ্ছে, and NASA has already put space station. India is also planning to place space station in 2030, 2035. একটা মজার জিনিস হচ্ছে এই স্পেস স্টেশন গুলোতে যারা ক্রু যারা করবেন বা যে সমস্ত প্যাসেঞ্জার যারা যাবে তাদের মানে এনভায়রনমেন্ট নেই। Space station is environmentless, and their gravity is not even zero, but microgravity. So in that microgravity, you don't need so much of structure, so much of bone structure, so much of protein you don't need. So you don't need so much of calorie food. So you need some special category of food. And that food category we have to modify, develop. এইগুলো রিসার্চ করে তোমাকে ডেভেলপ করতে হবে যে ওই মাইক্রোগ্রাভিটিতে যারা ক্রুরা থাকবে মাসের পর মাস, বছরের পর বছর, তারা কি খাবে হবে না। তাহলে এই ধরনের রিসার্চ করার প্রচুর স্কোপ আছে। বললাম মেডিসিনাল, এত মেডিসিনাল প্লান্ট আছে, হয়নি, অনেক কিছুই এক্সপ্লোর করা হয়নি। আর উই আর দ্য জাংচার অফ ইনফরমেশন, লস অফ ইনফরমেশন আমাদের কাছে আছে। And also the advanced personal lecture তোমরা শুনেছো genome sequence করে কত ইনফরমেশন গ্যাদার করছে, এবং এখন উনি বলছেন যে সিঙ্গেল জিন সিকোয়েন্স করে কিছু হবে না, প্যানজেনোম সিকোয়েন্স সমস্ত ওয়াইড একটা প্লান্ট ভ্যারাইটি আন্ডারে যত ওয়াইড স্পিসিস আছে সবার সিকোয়েন্স করতে হবে। এটা অনেক কিছু রিসার্চ করার স্কোপ আছে। যারা প্লান্ট সায়েন্স করছো তাদের ভয়ের ভাবনার কিচ্ছু নেই, এত কাজ করার চোখটাকে খোলা রাখো, মাথাটাকে চিন্তাটাকে তাহলে অনেক আইডিয়াস মাথায় আসবে, রিসার্চটা এই জন্য কোনো চিন্তা নেই। আর গভর্নমেন্ট অনেক নতুন নতুন ফেলোশিপ চালু করছেন, অনেক কিছু করছেন, ভয়ের কিচ্ছু নেই, পড়াশোনা করে যাও দেখবে।
সমস্ত নমস্কার এবং আমি একটা কথা বলছি, কি করতে হবে। আমি তো চৈতন্য কলেজে বোটানি অনার্স করলাম, আমার প্ল্যানিং সাইডটা খুব ভালো লাগবে, কিন্তু আমি প্রেসিডেন্সিতে বোটানিতে ভর্তি হলাম। প্রফেসর শর্মার কাছে গেলাম, স্যার আমি তো প্ল্যানিংটা ভালোবাসি, এখন তো সব সাবজেক্ট পড়তে হবে। বলে তুমি কি করে আলাদা বোটানি সাবজেক্ট মিটিং নেই, তুমি এগ্রিকালচারে জয়েন করো। আমি এগ্রিকালচারে জয়েন করলাম। এগ্রিকালচার 2 বছর পড়ার পরে আমি এই ডিপার্টমেন্টের এগ্রিকালচার মানে ইউনিভার্সিটি অফ ক্যালকাটা ইউনিভার্সিটি, হ্যাঁ এগ্রিকালচার পাস করার পরে তখন আবার স্যারের কাছে যাও, ঠিক আছে তুমি আমার কথা রেখেছো, তুমি ব্ল্যাক মিটিংটাকে ভালোবাসো, তুমি প্রেসিডেন্সি কলেজ ছেড়ে তুমি বোটানিতে যে এগ্রিকালচার দিয়ে ব্ল্যাক মিটিংটা করেছো, তার নিজ সাবজেক্টে ভালোবাসো। তখন স্যার আর দিদি একজন আমাকে দেখে ইন্টারভিউ নিলেন। দেখো এখানে তখন প্রফেসর ব্যানার্জি কি বললো যে স্যার আপনি কোনো ফেলোশিপ আছে, বললেন না ফেলোশিপ নেই। তখন স্যার দিদি বললেন ঠিক আছে তুমি একটা প্রজেক্ট বানাও, আর তোমাকে আমি ইন্টারভিউ দেবো 6 মাস পরে, তুমি প্রজেক্টের অ্যাবস্ট্রাক্ট তুমিই বলবে। তখন আমি কি করলাম, জুট রিসার্চ ইনস্টিটিউট ব্যারাকপুরে চলে গেলাম, আমেরিকান লাইব্রেরিতে গেলাম, গোলস ইনস্টিটিউটে গেলাম, এই কেমিক্যাল বায়োলজি সব ল্যাবরেটরি খুঁজে খুঁজে, তারপর আর্কিও দপ্তর ছিল দ্বীপে, ওনার কাছে গেলাম, বলে তুমি দেখো যে জিনিসটা কাজ করেনি সেইটা তুমি খুঁজতে থাকো, খুঁজে তারপরে জিনিসটা মধ্যে একটা অ্যাবস্ট্রাক্ট লিখলাম। ঠিক আছে, ছাত্রী কাছে যাও। এখন স্যার আমি তো সমাজ ধরে আমার কথা মেনে চলছি যে আমি প্ল্যান মিটিংটাকে ভালোবাসি, তবে এটা দেখে উনি খুব এগ্রি হলেন। তখন উনি আমাকে প্রফেসর অনুবংশী চ্যাটার্জির কাছে নিয়ে গেলেন। বলে দেখো এই ছানাটাকে তোমার কাছে দিলাম, তুমি এই ছানাটাকে দেখো কিভাবে কি করা যায়। তারপর স্যার আমাকে নিলেন। তখন আমি অলরেডি অলরেডি সময় মতো কাজ করে ফেলেছি, করতে আর ওই
রিভিউ স্টাডি করতে হতো, শুধু রিভিউ স্টাডি করতাম। কেমিক্যাল লাইব্রেরি বা ইনস্টিটিউট আইসার আমাদের ব্যারাকপুর যে তখন একটা বড় রেস্টের মতন হয়ে গেল লিখতে লিখতে। তখন স্যার বললেন যে তাহলে কি করবে স্যার জেআরএফ দিলেন একটা স্যার আরেকটু বলছি স্যার জেআরএফ দিলাম ফর্মুলে করে জেআরএফ হাবয় কালচার বলে তুমি এখন হ্যাঁ কটন 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 থাকবে পড়াশোনা শুনতে কটন স্যার আমি হচ্ছে চ্যাটার্জি নাম লাগে জেআরএফ তখন সিএসআই স্কিম তখন স্যার বললেন যে তুমি তো ঝুটের হাফ করে ফেলেছো তাহলে সংসার তুমি তো দেখেছি না স্যার ঝুটের হাফ করে ফেলেছো তাহলে ঝুটটাকে তুমি ছেড়ো না তোমার সমাজ কাজ হয়ে গেছে তুমি ঝুটেও কিছু করতে পারো তোমার প্রজেক্ট চলুন আন্তর্কালচার সিএসআই এটা তখন আমি দুটো একসাথে চ্যালেঞ্জ করলাম এবং ছাত্ররা বললেন দেখো তুমি ঠিকই পারবে। আমি ভগবানে আসি পরে 3-4 মাস পরে আমি টেস্টিং প্ল্যান করে ফেললাম মানে জুটের সিট থেকে ওটা পরে তাদের হ্যাঁ জুটের সিট থেকে না হ্যাঁ সিট থেকে জার্মিনেশন করলাম টিমের মধ্যে জার্মিনেশন করে হ্যাঁ জার্মিনেশন করে তখন তারপরে সাধারণ ঠিক আছে তাহলে তুমি এটা তুমি সাকসেস করবে তাহলে তখন আমাদের সবাই সেন্টার সিল করবে সেন্টার সিল করবে আমি বিবিসি মেসেজ দিছিলাম সেন্টার সিল করবে স্কিম আসলো যে অনেক ধরনের সায়েন্স দিয়ে হবে অনেক বাট একদম আছে সাধারণ আমাদের বলতেন যে আচ্ছা আমি বলছি আচ্ছা তখন আমি ওটাই বলছি সাধারণ আপনি এখন দেখো এই বছরে 182 জন আমরা সায়েন্স দিয়ে সেন্টার সিল করব ওরা ডাইরেক্ট ইনক্লুডিং করে ডাইরেক্ট ইনক্লুডিং না তিনি ডাইরেক্ট ইনক্লুড করতে পারছে ইনক্লুড তো পাস করলে ওরা সিলেক্ট হয়ে যায় তাহলে এই যে ধরুন তারপর কি গেল দেখেন তাহলে জুট করলাম কটন হলো ওদিকে মালবাড়ি ক্লাস সিট করে এই চারটে সাবজেক্টে মানে কখন করে পড়ছে আমার মেন অবজেক্টিভ বলার উদ্দেশ্য হচ্ছে এখানে আমরা বোটানি পড়ছে তো বোটানি করতে করতে অনেক সময় হাত দিয়ে যায় সব লেবুলাইতে চোখ লাগে না এক মাস খানি স্যার আমি 24 মাসের মধ্যে 1 মাস খানি এবিএসের কাছে রাখা উচিত তাই কারণ সবাইকে একটা আগ্রহ বাড়বে তখন যখন বোটানি যাবো ফরেস্ট ওটাও যখন ট্র্যাকিং করতে যাবে আমার সাথে তখন তাদের একটা আগ্রহ বাড়বে যে না আমরা এই লাইনেও যেতে পারি শুধু এতটাই তো বেশি লাগে ট্র্যাকিং হবে প্রচুর গেছি আমাদের সময় বড় ইনপুট ছিল হচ্ছে সেরিকালচার এবং জুটের রাইট ফাইবার আগে এগ্রিকালচারে অনেকগুলো ছিল কিন্তু এখন পলিটিক্যাল
মাইক্রোফোনটার কাছে
আমাদের অ্যাপ্লাইড সায়েন্সে কাজ করবার সুযোগটা দিনে দিনে আসছে এবং করা উচিত। আমরা যে সারাদিন ধরে সেটাই করেছি সায়েন্সের পরে বেসিক সায়েন্সের নলেজটা কিভাবে আমরা অ্যাপ্লাইড সায়েন্সে ব্যবহার করে নিজের দেশের দশের উন্নতি করতে পারি। তো আমার মেসেজ টু দ্য স্টুডেন্ট যে অনেক সিনসিয়ার ডিপ পার্সোনালিটি থাকা দরকার, থাকা দরকার, সিনসিয়ারিটি দরকার, এগুলো তো আমরা ছোটবেলা থেকেই শিখে এসেছি। কিন্তু অ্যাপ্লাইড সায়েন্সটাকে ভালোবাসতে হবে যে একটা কাজ করছি তার ফিউচার পার্সপেক্টিভ কি আছে। সিএসআই সিস্টেমে এটা প্রচন্ড রকম ভাবে করা হয়, বেসিক সায়েন্সের থেকে সিএসআই সিস্টেমে আইসিআর সিস্টেমেও তাই করা হয়। সেই জায়গাটাকে ধরতে হবে। আমরা যখন রিসার্চে আসবো তখন আমাদের একটা টপিক স্যার দিয়ে দিলে যেটা বললাম এই পার্সপেক্টিভ একজন আগে বললেন সেই পার্সপেক্টিভ একটা টপিক দেবে কিন্তু সেটা কেন করছি তার ফিউচার কি সেটা আমরা ভবিষ্যতে কিভাবে ইউটিলাইজ করবো সেগুলো তোমাদের চিন্তা করে বার করে
আমার নিজের পরিচয়টা একটু দাও। পিএইচডি করতে চাই প্রফেসর না কোথায় আছো সেটা শুনো।
শর্মিলা আমাদের কাছে বিশ্বভারতীতে ডক্টরেট করেছে প্রথম থেকে
বিশ্বভারতী ইউনিভার্সিটি years back I think I did my PhD in 86. তো অনেক দিন আগে তারপরে আমি পোস্ট অফ করেছি, তারপরে আমি বৈশিষ্ট্য ফেলেছিলাম, দেন আমি CSIR সিস্টেমে সায়েন্টিস্ট হিসেবে জয়েন করেছিলাম, সেখান থেকে
You will be happy to know I was one of the experts for a selection at IICB Calcutta. Expert member.
আমি ওখানে সায়েন্টিস্ট হিসেবে জয়েন করেছিলাম। আমার কয়েকজন স্টুডেন্ট এখনো পেছনে বসে আছে যারা
তোমরা কোথায় আসো বলো, তোমরা কোথায় আসো বলো, তোমরা বলো, তিনজনই বলো কে কোথায় আছো, কোথায় কাজ করছো, নাম। আপনার একটা পোস্টার ছিল দেখেছি বর্তমানে, বলো তোমাদের নামগুলো বলো।
কলেজ পুরুলিয়া,
তুমি বলো। হিংসা অ্যাওয়ার্ড সেটা বললে না,
এটা তো অপরিবেশন, যেটা ফোকাস করো তাহলে কি করে জানবে কি আমরা
মানে তোমাদের মতো একজন স্টুডেন্টদের কিরকম তার অ্যাচিভমেন্ট গুলো দেখো,
আমার জাস্ট একটা এক্সপিরিয়েন্স বলবো, আর আমার বিশেষ কিছু বলার নেই। একমাত্র মানে এন্ড অফ দ্য ডে আমরা যারা কিছু না কিছু অ্যাচিভ করেছি, কাজ করি, চাকরি করি, সিনিয়র হয়েছি, আমরা একটা বক্তব্য, আমরা অনেক সময় ফিলোসফিক্যাল বড় বড় থট অনেক কিছু বলবো, সবাইকে রিসার্চ করতে বলবো, সবাইকে বড় সায়েন্টিস্ট হতে বলবো, এটা স্বাভাবিক। কিন্তু আবার উল্টো দিকের পক্ষ, যারা চাকরি পায়নি তাদের এখন চাকরি দরকার, সেটা হচ্ছে আরেকটা খুব প্র্যাকটিক্যাল অ্যাসপেক্ট। সবাইকে রিসার্চ করতে হবে না, যারা রিয়েলি প্যাশনেট ফিল করবে তারা রিসার্চে যাবে, এটা 1 নম্বর। অনেক সময় লাগবে, রোজই নয় পাঁচটা, পড়ে থাকতে হবে, লেগে থাকতে হবে, আলটিমেটলি ভালো হবে, লেগে থাকবে। কিন্তু সবার এটা নাও হতে পারে, সময় না থাকতে পারে, পরিস্থিতি না থাকতে পারে। প্রাইভেট সেক্টরে প্রচুর কাজ পাওয়া যায়। তো ইন্ডিয়াতে অন্তত প্রায় 150 থেকে 200 খানা প্রাইভেট কোম্পানি আছে যারা ইয়াং ফেসেস খোঁজে। আমি ঘটনাচক্রে 2টি বড় প্রাইভেট কোম্পানিতে কাজ করেছি ইন্ডিয়াতে। আমরা বসে থাকতাম বটানিস্ট খোঁজার জন্য বা প্ল্যান্ট সায়েন্টিস্ট খোঁজার জন্য, ওয়েস্ট বেঙ্গল থেকে একটাও অ্যাপ্লিকেশন যেত।
আর বিশদভাবে বলো, এই জিনিসটা আমি চাইছিলাম আলোচনা আসলো, কেন এটা হচ্ছে ওয়েস্ট বেঙ্গলে?
আমরা বাঙালিরা একটু বোধহয় আদর্শবাদী, এবং আমরা একটু আমরা চেষ্টা করি যে সবাই রিসার্চে আসুক, সবাই বড় বিজ্ঞানী হোক, পড়া, এটা ঠিক আছে, এটা একটা গুড আইডিয়া। কিন্তু এর বাইরেও একটা বিশাল বড় জগৎ আছে, যে জগৎটা আমাদের ওয়েস্ট বেঙ্গলে আমরা ওটা জানতে পারি। আমি যখন ছাত্র ছিলাম আমিও জানতাম না, যে একটা জুওলজি বা বটানি পড়ে অন্তত 150 কোম্পানিতে অ্যাপ্লাই করা যায়, গিয়ে প্রাথমিক চাকরিটা করে অনেক কিছু শেখা। আর বাইরের জগতে অ্যাপ্লিকেশন অফ সায়েন্স যেটা হয় সেটা শুধু সায়েন্স নয়, তার সাথে রেগুলেটরি আছে, তার সাথে আইপি আছে, তার সাথে কোয়ালিটি কন্ট্রোল আছে, তার সাথে অজস্র ডিপার্টমেন্ট তার সাথে জড়িত আছে। এগুলো সবকটা স্পেশালাইজ, ইন্ডাস্ট্রিতে না গেলে এগুলো শেখা যায় না। আর শুধু সবাই সায়েন্সে রিসার্চ করবে এটা আমার খুব একটা প্র্যাকটিক্যাল মনে হয় না। অত চাকরি নেই, অত কাজ নেই আমাদের দেশে। বিদেশ যেতে হবে, এখানে ওখানে দৌড়াদৌড়ি করে করতে হবে। যার ফলে আমার ওই অভিজ্ঞতাটাই আমি শেয়ার করছি যে আমরা প্রায় 750 ক্যান্ডিডেট ইন্ডিয়াতে দেখেছি। ইউনিভার্সিটি, বম্বে ইউনিভার্সিটি, ইন্ডিয়ান ইনস্টিটিউট অফ সায়েন্স, বটনি, প্লান্ট সায়েন্স এর ছেলেমেয়েরা অ্যাপ্লাই করছে ওয়েস্ট বেঙ্গল থেকে, নট সিম্পল অ্যাপ্লিকেশন। আর আমরা বোর্ডে বসে আছি যারা, তার মধ্যে 7 জনের মধ্যে 5 জন বাঙালি। তো যাই হোক, পরের কথাগুলো আমি বললাম না, তো উই ফেল্ট ভেরি স্যাড অ্যাকচুয়ালি। কিন্তু এই বিষয়গুলো আমাদের এখানে অতটা চর্চা হয় না। ট্র্যাডিশনাল এই সাবজেক্ট গুলোতে আমাদের মোর থিওরিটিক্যাল, মোর সায়েন্স ওরিয়েন্টেড, মোর রিসার্চ ওরিয়েন্টেড পড়াশোনাটাই বেশি হয়, তার ফলে এগুলো আমরা জানি না। আমি বলবো 150টা না পারো 10টা, 15টা, 20টা কোম্পানির ওয়েবসাইটে ঢুকে নিজের সিভিটা ওখানে দেওয়ার ব্যবস্থা আছে। এইটা 1 নম্বর। আর আরেকটা কথা বলবো যে বায়োলজিক্যাল সায়েন্স, আমি পৃথিবীর যতটুকু ঘুরেছি, এখনো ঘুরছি, বায়োলজিক্যাল সায়েন্সে যে কোনো বায়োলজিক্যাল সায়েন্স থেকে যে কোনো বায়োলজিক্যাল সায়েন্সে যাওয়া। জুওলজি পড়ে বটানি যেরকম আমি করছি, সেরকম বটানি পড়ে জুওলজি। আমার প্রিয় বন্ধু এখান থেকে পাস করে এখন লিভার ইনস্টিটিউটের সিনিয়র ফ্যাকাল্টি, হেড অফ দ্য ডিপার্টমেন্ট। আমরা দুজন ক্লোজ ফ্রেন্ড ছিলাম। সে বটানি থেকে গিয়ে লিভারে কাজ করছে, আর আমি জুওলজি থেকে এসে প্ল্যান্টে কাজ করছি। তো এনিথিং ক্যান বি ডান। বায়োফিজিক্স, বায়োকেমিস্ট্রি, বটানি পড়লে যে অন্য কিছু করা যায় না এমনটা নয়। রিসার্চে থাকলে সেগুলো করা যায়। আর ইনস্টিটিউট গুলোতে গেলে নানা রকম ক্রস ফাংশনাল, বায়োলজি এখন ক্রস ফাংশনাল, এখন এগুলো বটানি, হার্ডকোর জুওলজি এরকম কিছু। তার ফলে চোখ কান খোলা রেখে সর্বত্র অ্যাপ্লাই করো। ইন্টারন্যাশনাল, ন্যাশনাল ফান্ডিং যেগুলো পিএইচডি স্কলারশিপ বা অন্যান্য স্কলারশিপ আছে সেগুলোতে অ্যাপ্লাই করো। আর পৃথিবী জুড়ে বহু ইন্ডাস্ট্রি আছে যারা ইয়াং ফেসেস খোঁজে, সেইগুলো সম্পর্কে একটু ওয়েবসাইটে ঢুকে ইউটিউব কম দেখে একটু এগুলো বেশি দেখতে হবে, দেখে এগুলোতে অ্যাপ্লাই করতে হবে। তাহলে
আমারই তো মানে আমাদেরই স্টুডেন্ট রয়েছে, আমার মোটামুটি প্রতিদিনের কথাগুলো তো বলি এই ধরনের কাজগুলো করতে। তো আজকে আমি এই মুহূর্তে যেটা বলবো সেটা হচ্ছে একটু অন্যরকম। সেটা হচ্ছে আজকের যা বক্তব্য হলো তার উপরে যদি আমরা একটু সামাপ করি। 1 নম্বর হচ্ছে দেয়ার উইল বি নো কালচার উইদাউট এগ্রিকালচার। এগ্রিকালচার ছাড়া কোনো কালচার থাকে না। তো এটার উপরে আজকে একটু ফোকাস করে কথা হবে। ওকে, কিন্তু এই ফোকাস করতে গিয়ে প্রথম ডক্টর পাঠক যেটা দিয়ে শুরু করেছিলেন ক্লাইমেট চেঞ্জ আর হচ্ছে কার্বন ফুটপ্রিন্ট, এম আই রাইট? তো তোমরাই বলো ক্লাইমেট চেঞ্জে হোয়াট ইউ ক্যান ডু। কেউ বলতে পারবে যে আমরা এই ক্লাইমেট চেঞ্জ কন্ডিশনে কি করতে পারি আমাদের ছোট ক্ষমতার মধ্য দিয়ে? কে কি করতে হবে? গাছ লাগাতে, রাইট? তো স্টার্ট ফ্রম টুডে। ওকে, আজকের এই মেসেজটা নিয়ে যাই যে আমরা এই ক্লাইমেট চেঞ্জের যে ভয়ঙ্কর যুদ্ধে আমরা সেই সেতুবন্ধের অনুমান হয়ে বড় বড় পাথর না তুলতে পারি, কাঠবিড়ালিটুকু হতে পারি। প্রতি বছর যদি টার্গেট নিই 10টা করে গাছ লাগাবো এবং 10 জনকে কনভিন্স করবো, তাহলে একদিন কিন্তু উই ক্যান ক্রিয়েট দ্য চেঞ্জ। ওকে, দুঃখের বিষয় ক্লাইমেট চেঞ্জ নিয়ে যত মিটিং হয় পলিসি মেকাররা করে, রাইট? পলিসি মেকাররা ক্লাইমেট চেঞ্জে মিটিং করেন কিন্তু এসি রুমে বসে। খুব ইন্টারেস্টিং, দে টক অ্যাবাউট কার্বন ফুটপ্রিন্ট সিটিং ইন দ্য এসি রুম। ওদের ডিনার বা লাঞ্চে কিন্তু থাকে হচ্ছে মাংস, যেটা হচ্ছে কার্বন ফুটপ্রিন্টের কত পার্সেন্ট রোল। সো ডোন্ট থিঙ্ক অ্যাবাউট দিস পলিসি মেকার, দে ক্যান গো দেয়ার ওন ওয়ে, বাট উই ক্যান গো ইন আওয়ার ওন ওয়ে বিকজ দিস গ্রিন প্ল্যানেট ইজ ফর আস। তোমাদের জন্য আমরা বুড়ো হচ্ছি, আমরা কিছুদিন বাদে চলে যাবো। So we this green planet in the hand of you, তোমরা হচ্ছে ক্যান্ডেল বিয়ার। তো তোমরা কি করবে তোমাদের কথা আমরা এছাড়াও আমরা করতে পারি কি কার্বন ফুটপ্রিন্ট কমানোর জন্য আমরা কলকাতা বুকে কি করতে পারি এনিবডি কলকাতা বুকে আমরা কি করতে পারি উই ক্যান ক্রিয়েট অ্যালভিওলি কে দেখেছিস অ্যালভিওলি উই হ্যাভ ক্রিয়েটেড আই হ্যাভ ক্রিয়েটেড 8 অ্যালভিওলি যেখানে হচ্ছে মিয়াওয়াকি ফরেস্ট এট কলকাতা সো ডোন্ট ট্রাই ফর কার্বন ফুটপ্রিন্ট তো আমরা এখানে প্রায় মোর দ্যান লাখস ল্যান্ড লাগিয়েছি in 100 স্কয়ার মিটার ল্যান্ডে কার্বন ফুটপ্রিন্ট এবারে নিশ্চয়ই ক্যালকুলেশন করা হয়েছে তো আমরা দেখেছি কতটা কার্বন ডাই অক্সাইড সিকুয়েস্টার করতে পারি অলসো দোজ হু আর ইন্টারেস্টেড যদি কারো দেখতে ইচ্ছে করে যে 100 স্কয়ার মিটারে ইউ ক্যান অ্যাকোমোডেট 500 প্ল্যান্ট এবং 15 বছরে ইউ ক্যান ক্রিয়েট এ ফরেস্ট অফ 150 ইয়ার্স কাছেই রবীন্দ্র সরোবর আছে রবীন্দ্র সরোবরে উই মেক এ মডেল সো ইউ ক্যান ভিজিট এনি টাইম ইউ লাইক টু তো কলকাতাতে যেহেতু আমাদের ভার্টিক্যালি বাড়ছে তো ভার্টিক্যালি বাড়ছে এবং গাছ কমছে তো আমরা এই পপুলেশনকে কার্বন ডাই অক্সাইড সিকুয়েস্টার করার জন্য আমরা যদি 100 স্কয়ার মিটার ছোট ছোট জায়গা পাই সেখানে আমরা কিন্তু 500 প্ল্যান্ট অ্যাকোমোডেট করতে পারি তো অ্যাজ এ মেসেজ আমি শেষ করে দেবো দেন হচ্ছে না আমাকে এবার তাড়া দেবে রেগুলার কথায় ফলে যারা আমার সঙ্গে রেগুলার দেখা হয় না তাদের জন্য খবরটা যদি কারো ইন্টারেস্ট থাকে করতে হবে সেকেন্ড হচ্ছে ফুড সিকিউরিটি ওকে আমরা ফুড সিকিউরিটি আমাদের 10,000 বছরের ট্রেডিশন ছিল হচ্ছে আমাদের এগ্রিকালচার ইটস এ ট্র্যাডিশনাল সিস্টেমকে আমরা করেছি কিন্তু আমরা ইন দ্য নেম অফ ফুড সিকিউরিটি আমরা কতটা ফুডটাকে উদর ফুডটি মানে ফুড সিকিউরিটি না স্যার যেটা বলছিলেন এনার্জি রিচ ফুড বা এখন মডার্ন টেকনোলজির সঙ্গে সঙ্গে তুমিও যেটা বলেছো যে আমাদের অনেক ট্র্যাডিশনাল ভ্যারাইটি হারিয়ে যাচ্ছে দ্যাটস ট্রু আমাদের অনেক ট্র্যাডিশনাল ভ্যারাইটি হারিয়ে যাচ্ছে দ্যাটস বিকজ অফ আওয়ার ইগনোরেন্স বা আমরা মেনটেন করছি না বা অন্য হাই হেলদিং আসছে বলে ওটা ইগনোর হতে হতে নষ্ট হতে পারে তো সেটাও একটা আমাদের চিন্তার বিষয় কারণ বায়োডাইভার্সিটিকে আমাদের মেনটেন করতে হবে অ্যাজ মানে ওদেরও এভরি রাইট আছে এই জগতে বাঁচা আর ছিল হচ্ছে ওয়াটার ফুটপ্রিন্ট নিয়ে তুমি বললাম ওয়াটার ফুটপ্রিন্টে আমরা কিভাবে কন্ট্রিবিউট করতে পারি তুমি কি করতে পারো আমাদের কথা হচ্ছে আমরা কি করতে পারি পলিসি লেভেলে বিরাট কিছু কিন্তু আমরা কি করতে পারি আমরা করতে পারি ওয়াটার ফুটপ্রিন্টে এভরিডে যদি বাড়িতে 5 বার টয়লেট ইউজ করো ফ্লাশ ইউজ করো ফ্লাশের মধ্যে এই যে স্যার জলের বোতল দিলেন একটা 1 লিটারের জলের বোতল ফ্লাশের মধ্যে দিয়ে রেখে দেবে তো এভরি ফ্লাশে 1 লিটার করে ইউ ক্যান জল কন্ট্রোল করতে আমরা এখনই দেখুন 5 বার যদি বাড়িতে ফ্লাশ হয় আমি 5 লিটার করে কিন্তু ওয়াটারকে আমি কম ইউজ করছি এটা ভেরি সিম্পল টেকনিক তো দেয়ার আর সো মেনি টেকনিক আই ক্যান টক অ্যাবাউট ডিফারেন্ট টেকনিক বাট এটা হচ্ছে আমাদের সিঙ্গল ইউনিট লেভেলে সারা বিশ্বে যত মানুষ আছে সিঙ্গল ইউনিট দেখুন তার মধ্যে সবচেয়ে হার্মফুল এলিমেন্ট অর্গানিজম হচ্ছে হিউম্যান বিং আর কোনো অর্গানিজম এত ডেঞ্জারাস অর্গানিজম তৈরি আছে নেক্সট আমাদের ছিল অন্টারপ্রিনারশিপের ক্ষেত্রে বটনি we are lucky enough we have selected a subject like botany from where we can create millions of entrepreneurs so as I said in class also দাদা আমি আমার এই লকডাউনের সময় থেকে এখন অবধি 1650 মানুষের আমি পেটের ভাতের ব্যবস্থা করেছি আই মেড 5 ইন্ডাস্ট্রি পিপল যারা ইন্ডাস্ট্রি করছে এবং ফ্রম কুমারগ্রাম টু কাকদ্বীপ 28 ইউনিট আমরা তৈরি করেছি হয়তো অনেকে দেখেছো বা শুনেছো যে কিভাবে আমরা আমাদের সাবজেক্ট থেকে অন্টারপ্রিনার তৈরি করতে পারি যেটা সবাই বলছে জব ইজ এ ক্রাইসিস সো হোয়াই শুড ইউ থিংক ডিফারেন্টলি টু ক্রিয়েট জব ফর দ্য পিপল আর আমরা এমন একটা সময়ের দিকে যাচ্ছি মেকানে এআই টেকনোলজি আবার আসছে ফলে এই জায়গায় দাঁড়িয়ে জব যে কোন দিকে গেলে আমাদের কারণ হচ্ছে এআই টেকনোলজি আসুক আমাদের যাই আসুক না কেন পেটে দানা ছাড়া কোনো কিছু চলবে না কম্পিউটার খাবো না টাকা খাবো না কারণ হচ্ছে এটা হচ্ছে খুব কমন জায়গা আমাদের ভাবতে হবে পকেটে টাকা থাকবে ধানের ছবি দেখে চালের ছবি দেখে হাওয়া খেতে হবে কারণ হচ্ছে ইফ ইউ ভিজিট এনি ভিলেজ যারা এগ্রিকালচার প্রত্যেকে জানে ইফ ইউ ভিজিট এনি ভিলেজ দেয়ার ইউ উইল গেট এখন 60 থেকে 70 বছর বয়সী লোকেরা মাঠে চাষ করছে কোনো ইয়ং জেনারেশন আসছে না ওই লোকগুলো আগামী 20 বছর বাদে ফিনিশ হয়ে যাবে দেন হু উইল প্রডিউস ফুড ফর ইউ তো এটা আমাদের করতে হবে কারণ হচ্ছে বলেছি দেয়ার উইল বি নো কালচার উইদাউট এগ্রিকালচার এটা কিন্তু আমাদের প্রত্যেকের ডিউটি এই মেসেজটা কিন্তু আজকের মিটিং থেকে সবাই নিয়ে গিয়ে ট্রান্সমিট হ্যাঁ তোমাদের এরিয়ার মধ্যে ট্রান্সলেট করো নিজেরা করো করতে আমাদের হবে তো এখন মেসেজ দিলাম আর বাকি তো অনেক রেগুলার হয় থ্যাংক ইউ
Attest to anyone? তোমার নিজের পরিচয়টা দাও, তোমার একটা ভালো পরিচয় আছে।
My name is Sharma, and right now before all of you, the only identity I have is that all of you are students, and I'm a research scholar of this department, and I'm in my third year of PhD. So right now, what Sir said, I really appreciate what he said. Like I will just take one example. যখন আমি মাস্টার স্টুডেন্ট ছিলাম, একটা সময় আমার কাছে খুব বড় ব্যাপার ছিল আমি নেট কেন পাচ্ছি। তো যখন মানে পিএইচডি করব এই ড্রিমটা ছিল, তখন নেট পাওয়াটা লাইফে একটা সবচেয়ে বড় গোল ছিল, তার বাইরে আমি কিছু ভাবতাম। তো যখন নেটটা পেয়ে গেলাম, এখন যখন কোনো জুনিয়র এসে বলে যে দিদি নেট পাচ্ছি না, বলো না কি করবো, আমি বলি নেট পাচ্ছিস না, তোরা এইচএস পাস করে এসেছিস, 6 মাস পড়বি নেট পেয়ে যাবে, এটা কোনো কঠিন ব্যাপার না, ইটস ভেরি ইজি। এখন পেয়ে গেছি বলে এই কথাটা বলতে খুব সহজ লাগছে, কিন্তু একদিন যখন আমি ওইখানে ছিলাম তখন অতটা সহজ লাগেনি। যেমন আজকে এখানে দাঁড়িয়ে এখন নেক্সট চিন্তা হচ্ছে পিএইচডি করে কি করবো। তো আজকে আমরা যাদের কাছে এই কথাগুলো শুনছি, ইটস ভেরি ইন্সপায়ারিং, বাট আমাদের স্টেটে দাঁড়িয়ে আমরা যারা রিসার্চ স্কলার ফাইনাল ইয়ারের দিকে এগিয়ে যাচ্ছি, সবার মনে হয় একটাই কনসার্ন, হোয়াট নেক্সট। কারণ ইন্ডিয়াতে স্পেশালি ওয়েস্ট বেঙ্গলের একাডেমিক্স এর অবস্থা খুব একটা ভালো না, বিকজ আমি জানি মানে there is severe crisis of jobs. আমি পড়াতে খুব ভালোবাসি, আমি রিসার্চের থেকে বেশিও পড়াতে ভালোবাসি। মানে সৌমিত্র স্যারের সাথে এই নিয়ে আমার অনেকবার কথা হয়েছে, স্যার বলেছে ওয়াই নট রিসার্চ। আমি বলি স্যার আমি ছোটবেলা থেকে বাবা মাকে টিচার হিসেবে দেখেছি, আই ওয়ান্ট টু বিকাম আ টিচার। কিন্তু আনফরচুনেটলি আমি জানি না যে আমার স্বপ্নটা কবে গিয়ে পূরণ হবে। তো রাইট নাও হোয়াট আই এম ফর ইজ getting a job, first a security, and then we can move towards any other security. I don't think without having a, it's my opinion, I'm sorry to contradict. I'm not telling that research করা উচিত না, বাট আমি এটাই বলছি যে আমার পজিশনে দাঁড়িয়ে আমার এখন মনে হয় আমি নিজে যদি একটা ল্যাব খুলি বা আমি যদি কোনো একটা পজিশনে যাই, সেখানে গিয়ে আমার পক্ষে ভাবাটা বা অন্য কিছু নিয়ে ভাবাটা নিজে পারবো টিচার, বাট আমি কাউকে ডিসকারেজ করছি না। বাট ফর মাই পজিশন, আমি আমার জায়গায় দাঁড়িয়ে অ্যাজ এ রিসার্চ স্কলার আমার মনে হয় রাইট নাও অবভিয়াসলি আমি নিজেও যেটা নিয়ে কাজ করি, ইট ইজ ভেরি মাচ রিলেটেড টু এনভায়রনমেন্ট। আমি ফুড নিউট্রিশন, আমি রাইস এন্ড নিউট্রিশন নিয়ে কাজ করি, এবং আমি সব জায়গায় বলি আই এম ডুইং and work on sustainable agriculture, and that is the practice that I am doing. So of course এটা করবো না, এটা করবো আমি আন্ডারে কাজ করি, প্ল্যান্ট ফিজিওলজিতে কাজ করি। তো এটাই আমার বক্তব্য যে of course I think research is very much necessary. Without that we cannot survive. We need good new techniques and tools so that এখন existing জিনিসগুলো we get better quality and also quantity of food. আমাদের এখন মনে হয় মেইন টার্গেট এটাই, so that we can improve the quality of our existing food and also increase the quantity of the food. But right now yeah, I think this is what I stand for right here.
যেটা আমাদের গর্ব যে আমরা এই ডিপার্টমেন্ট অফ বটানিতে লিগারশিপ ফোন করতে ফাইনালি ওয়ান অফ देम ও ফার্স্ট ক্লাস ফার্স্ট ইন সিএসআই এখনো যাচ্ছে যেটা জওহরলাল নেহরু ফুড রাইট ফেলোশিপের জন্য এখন দেশে চলেছে 3-4 মাসের মধ্যে আমেরিকা যাচ্ছে। তো সবে জার্মানি থেকে ঘুরে এলো, ফ্রান্স থেকে ঘুরে এলো। তো ও পড়াশোনায় ভালো, তারপরে ওর যেটা বক্তব্য সেটা আমার মনে হয় সবারই ঠিক টাইমে থাকে। আমরা সময়ের সাথে সাথে একটা দিক নিশ্চয়ই খুঁজে, আর যেটা অনিন্দ স্যার বললেন যে কষ্ট অত সহজ নয়, কিন্তু এতে তো মজা আছে, তো ভালোই মজাই আছে।
These are good examples of the department. আমাদের বন্ধুরা, আমাদেরই যারা আছে, তারা ইচ্ছা ফেলো, তারা ইনসা সায়েন্টিস্ট, তারা
আমি কিছুদিন আগে ফুলব্রাইটের একটা প্রিন্ট কালচার ওরিয়েন্টেশনে গেছিলাম, এবং আমার ফিরে এসে একটাই কথা মনে হয়েছে, আমাদের স্টুডেন্টরা, আমরা আজ স্টুডেন্ট হয়তো অনেক কিছু চেষ্টা করি না যেটা আমাদের করা উচিত। মানে আমাদের হয়তো মনে হয় যে আমরা পারবো না, কিন্তু অ্যাকচুয়ালি আমরা কোনোদিন অ্যাপ্লাই করার পাওয়ার নট পরের ব্যাপার, বাট এই যে জার্নিটা, এই যে একটা অ্যাপ্লিকেশনের প্রসিডিওর সেখান থেকেও কিন্তু আমার মনে হয় অনেক কিছু শেখার আছে, যেটা যতদিন আমরা সেটা করছি নিজেদেরকে হয়তো আমরা এমনি খুব ইনফেরিয়ার ভাবি, এরকম ব্যাপার না যে আমরা পারি না। মানে ICAR অনেক ইনস্টিটিউট, মানে অল ইন্ডিয়া অনেক ইনস্টিটিউট থেকে ছেলেমেয়েরা গেছে, ওয়াই নট ক্যালকাটা ইউনিভার্সিটি? কেন সবসময় কেন আমরা ভাবি সবাই আগে সবাই বলি যে কেন তোমরা এখানে
যেটা বলেছে সৌগত লাভে ও ফরেন স্টুডেন্টস চাইছে ও ভালো উচিত।
যে তোমরা তো মনে করো যে কোম্পানি থেকে আমি যোগ করবো, তাই না? কোম্পানিগুলো তো গিয়ে খোঁজ খবর রাখতে হবে কোথায় কোন কোম্পানিতে এই ধরনের লোক রিজার্ভ করে বড় লোক পাই না আমরা, করতে হবে। তাই তো এটা ব্যাপারটা।
আমার মনে হয় যে আমরা ওভারথিংকিং খুব বেশি করে ফেলি, এটা একটা প্রবলেম আমাদের ক্ষেত্রে। আমার ক্ষেত্রে হয়েছিল আমরা ফার্স্ট ব্যাচ অফ ইন্টারভ্যান্স ছিলেন একজন এমবিবিএস, উনি প্লান্টের কিছু নয় বা উনি কিছু জানেন না, তো আমি ভেবেছিলাম ওনার নামে ফেরত কেন প্লান্টকে দেওয়া হবে। তো আমি তো অ্যাপ্লাই করবো না বলে ঠিকই করে নিচ্ছি, স্যারের কাছে একদিন এসে আমি স্যারকে বললাম এমনি একটা বেরিয়েছে আর কদিন আছে, আমি কি অ্যাপ্লাই করবো? স্যার বললো, স্যার অ্যাকচুয়ালি এমকে ভানকে খুব ভালোভাবে চিনতেন, ওনার মনে হয়েছিল যে হয়তো ওনার লেগেসি প্লান্টেও দেবেন, কিন্তু ততদিনে বিজেপি বলেনি যে এটা প্লান্টের জন্য না অ্যানিমালের জন্য না কার জন্য। আমি জাস্ট অ্যাপ্লাই করে দিই, প্রজেক্ট লিখে অ্যাপ্লাই করি, আর ওটাই মানে আমরা অনেক বেশি ভেবে নিই হয়তো, যেগুলো আমরা না ভেবেই কাজটা করলেও হয়তো ভালো রেজাল্ট পাবো।
এমকে ভান সম্বন্ধে একটা কথা বলি। এমকে ভান একজন ডাক্তার এবং অ্যাডমিনিস্ট্রেটর ডেপুটি সেক্রেটারি ছিলেন। He often used to tell me he hates Bengali. He hates Bengali. Without a Bengali, nothing can be. Second sir.
My name is Sharma Kumar Chakraborty. I am an alumnus of this university. এখানে সবাই অনেকেই চেনেন, যারা চেনেন না তাদের জন্য বলছি। I begin with what Professor Krishna to say. Agriculture and culture একটু modify করছি ওনার কথাটাকে। Agriculture মানে ফসল, কৃষি, মানে agriculture is culture. No culture without agriculture. Agriculture is culture. It is synonymous. কৃষি ফসল থেকে কিন্তু সংস্কৃতিটা এসেছে, এটা সুনীতি কুমার চট্টোপাধ্যায় বলেছে। এক নম্বর কথা। দু নম্বর কথা হচ্ছে যে আজকে তো আমরা এই অ্যালামনি অ্যাসোসিয়েশন বটানি ডিপার্টমেন্ট এবং আজকে আমরা সারা বছর ব্যাপী প্রফেসর একে শর্মা বার্থ সেন্টেনারি সেলিব্রেট করছি। তো সেখানে একটা ওনার কথা আমি স্মরণ করছি। আমি কলেজে পড়াতাম, এখন রিটায়ার করেছি। তো মাঝে মাঝে জিজ্ঞেস করতেন স্যার, উনি সবাইকে অ্যাড্রেস করতেন বাবু, সবাই জানেন কিরম আছো বাবু, কি করছো বাবু, এরকম ভাবে জিজ্ঞেস করতেন। তাই এরকম মাঝে মাঝেই যেতাম, আমি এই ল্যাবেই কাজ করতাম। কি করছো, কোথায় আছো, কি সমস্যা হচ্ছে, তখন বলতাম যে এরকম কলেজে বটানি পড়াই ঠিকই, কিন্তু অনেকের মানে ওই ফার্স্ট ইয়ার সেকেন্ড ইয়ারে এতে উৎসাহ আনতে আমি জানি না কতটা পারছি না, আমার খুব কালঘাম ছুটে যাচ্ছে, মানে উৎসাহ জেনারেট করতে একটা কোথাও একটা পারসেপশনের গন্ডগোল হচ্ছে। তখন আমি যখন জিজ্ঞেস করি, আমি স্যারকে বলছিলাম যে স্কুলে কোনো সমস্যা হচ্ছে, উনি তখন বললেন যে তোমরা একটা কাজ করো, তোমরা ওই অ্যালামনি আছে তো তাদেরকে বলো যে একটু স্কুলের দিকে তাকাতে, কেননা স্কুলে স্যার জানতেন অনেক জায়গাতে তো স্যার ঘোরাঘুরি করতে কথাবার্তা বলতেন। আমারও নিজের অভিজ্ঞতা হয়েছে, তাই কথা বলে কথা বলে স্কুল লেভেল অন্তত আমাদের রাজ্যে আমাদের প্লান্ট সায়েন্স বা বটানি যাই বলুন না কেন, সেটা অত্যন্ত নেগলেক্টেড এবং এটার একটা পারসেপশনের ব্যাপার। আমি যতই বলি না কেন, হ্যাঁ, যে বড় বাবু, ওই একটা সাধারণ পারসেপশন যে কতগুলো ল্যাটিন না, কতগুলো শক্ত ডেসক্রিপশন, এই যে পারসেপশনটা রয়ে গেছে না, এটা পারসিস্ট করছে।
ছাত্রছাত্রীরা যখন কলেজে আসে, ইউনিভার্সিটিতে আসে, তখন তারা সাবজেক্টটা ভালোবাসতে শেখে, কিন্তু অনেকটা আমার দেরি হয়ে গেছে। এই এই মানে আমার মেসেজটা হচ্ছে ক্যাচ দেম ইয়াং, অর্থাৎ ছাত্রছাত্রীদের বলছি, তোমাকে ভাই বোন আত্মীয় স্বজন যারা স্কুলে পড়ে উঁচু ক্লাসে, বটানিটাকে তারা কিভাবে ভালোবাসতে শিখবে সেটা যদি একটু ভাবনা চিন্তা করো, তাহলে বোধহয় তোমাদের উপকার হবে, তাদের উপকার হবে। মানে এটুকু বলেই আজকে শেষ করছি।
অনেক হয়ে গেছে, শোনার ইচ্ছা নেই অনেকেরই। রাজীব বন্দ্যোপাধ্যায় এখন বর্ধমান বিশ্ববিদ্যালয়ে আছে, প্রফেসর হেড। আমাদের ওখানে প্রফেসর PM ভাদুরী ফার্ম আছে, রিসার্চ ফার্ম, আমাদের এগ্রিকালচার হয়। আমি সম্বন্ধে এক দু কথা বলবো। আগেকার দিনের মানুষ বা সংসদে যারা ছিলেন,
And I would like to express my most sincere thanks to Professor Sharma and Professor Rita Kundu for having this organization today and celebrating in a befitting way to our great grand teacher, Professor. I have come from Vishwanath. We are all colleagues. The professor and I read to the finalists. You have seen Chiriyakhana Zoo. He was director of National Library. Like a single Chobi. I was the director there. I got Kalam, Manmohan Singh, Sharma Chamuti, all the scholars of it. The botanists did in the role of the director of National Library. Prestigious. Government of your boss is very stuck. No one to ask anything. So all the botanists, be proud yourself. Do it yourself. Live it. Thank you very much.
তো সমাচারের কথা বলবো, বা আমরা কাজ করছি প্রফেসর PK গুপ্তা স্যারের সঙ্গে। আমরা এই বয়সেও একটা এজ অফ 90, 92, মানে এনারা কখনো শিখতে পিছপা হন না, এবং এর থেকে বেশি ইন্টারেস্ট দিয়ে ওনারা শেখে। এখন হয়তো আমাদের হাতে একটা স্মার্টফোন দিয়ে দিলে আমরা হেজিটেট করবো কিভাবে চালাবো, কি করবো, কিন্তু ওই বয়সে ওনারা এটাই আগ্রহ প্রকাশ করে। এই জন্য আমি নতুন স্টুডেন্টদের বলবো, ইয়াং স্টুডেন্টদের, তোমার প্যাশন থাকতে হবে, সেই বিষয়টায় আগ্রহ থাকতে হবে। এখানে আমি বড় বড় সায়েন্টিস্ট এবং প্রফেসরদের যথাযোগ্য সম্মান দিয়ে বলছি, কেউ কিছু জানে না, তুমি সব জানো। তুমি যদি ভেবে নাও এটা আমাকে করতে হবে, আমি যেমন ক্রিসপার টেকনোলজি জানি না, কিন্তু আমি একবার যদি ভেবে নিই এটা আমি করবো, আমি কিন্তু এটাকে হারানোর ক্ষমতা নেই, এটা আমি বলছি, এবং আমরা করে দেখাই। এরকম নতুন নতুন ফিল্ড, যেমন কম্পিউটার সায়েন্স, কম্পিউটারে বড় বড় এক্সপার্ট আছেন। আমি DIT লেকচার রাজনীতি চাকরি করতাম। আমি দ্য ফার্স্ট পারসন ইন ইন্ডিয়া যে ক্লাউড কম্পিউটিং এ কাজ করেছি লং ব্যাক, এবং ইন্ডিয়ায় আমাদের ফার্স্ট পেপার এবং অল ওয়ার্ল্ডে আমাদের পেপার গুলো হাইলি সাইটেড। আমাকে কম্পিউটার সায়েন্সের লোক এসে বলেছিল এইসব কাজ তো আমাদের করা উচিত, তোমরা কেন করছো? অনিবাবু বলেছেন যে কোনো সাবজেক্ট কারণ নয়, দ্য এন্ড অফ দ্য ডে সব সাবজেক্ট গিয়ে এক জায়গায় থেকে যাচ্ছে। এবং উনি এক্সাম্পল দিলেন, বটানি জুলজি করছে, জুলজি বটানি করছে, সেম থিং। তোমরা ভেবে নেবে স্যাররা কিছু জানে না, তোমরা নিজে পড়াশোনা করবে, মনে জোর আনবে, এবং যে কোনো সাবজেক্ট নতুন হোক যাই হোক তুমি পারবেই পারবে, সেইটা ভেবে নিয়ে এগোবে এবং পড়বে। আমাদের পাস করতে ছিল সুনিতা, ভয় পাচ্ছি ও চাকরি পাই না। স্বর্ণালী, আমি স্বর্ণালীকে সাহস দেওয়ার জন্য বলবো, আমি আমার পুরোনো ব্যাকগ্রাউন্ড ভুলে যাচ্ছি। আমি বর্তমানে 8 জন রিসার্চ স্কলার প্রডিউস করেছি 2015 সাল থেকে, বছর হচ্ছে। 8 জন ওয়েল এস্টাবলিশড ইন অ্যাব্রড অ্যান্ড ইন ইন্ডিয়া। আমার প্রথম যে স্কলার ডক্টর অপর্ণা ব্যানার্জি, right now she is full professor and science dean in University of Chile, এবং আলাদা অনেক স্টুডেন্ট তারা বিদেশে এস্টাবলিশ। আমি শুধু বর্ধমানের কথা বলছি, তার মানে এই নয় যে আমি খুব জানি কিভাবে খেলতে হবে, পুশ করতে হবে। ওই স্টুডেন্টরা কিন্তু প্রথম দিন এসেছিল, he or she is big zero। আমি আমার স্টুডেন্টদের বলি যে আমি তোমার টিচার ফর 2 months or 3 months, এরপর থেকে তুমি আমার টিচার হবে। তুমি তো করে এসে আমাকে শেখাবে, জানেন স্যার এটা এই হয়, জানেন স্যার ওটা ওই হয়, জানেন স্যার এটা এরকম করে করতে হবে। আমি তোমার কাছে শিখবো, তুমি এটা করতে পারো, তাহলে তুমি ঠিক স্টুডেন্ট। এই জিনিসটা শিখতে জানতে করতে হবে, তাহলেই হবে। থ্যাংক ইউ।
তারপরে কোনো বলবে, তারপর আরো বলবে 1 মিনিট করে বললো হবে।
সবাই খুব ক্লান্ত, বেশি সময় নেব না। ভাইস চ্যান্সেলর, ডিজি, হেড, ডিরেক্টর সবার কথা শুনলে একটা জিনিস আশা করি বুঝতে পেরেছো যে এগ্রিকালচারের লোকেরা বেশ ডেঞ্জারাস। সমস্ত কালচারের উপর আমরা ডমিনেট করে ফেললাম, একটা স্ট্রাকচারাল এন্ড ফাংশনাল জিনিসকে আমরা মোটামুটি এগ্রিকালচার বেরিয়ে দিলাম আজকে। তো ব্যাপারটা হচ্ছে যে একবার খালি ভাবো টাইম মেশিনে যদি যাকে আমরা আজকে রেসপেক্ট জানাচ্ছি প্রফেসর শর্মা যদি আজকে 1924 না হয়ে 2024 জন্মাতেন, কি ধরনের রিসার্চ উনি করতেন? তিনি সাইন্টিফিক করতেন। আর যদি আমরা 1924 জন্মাতাম, আমরা কি ধরনের রিসার্চ করতাম? তো সকাল থেকে যতগুলি প্রেজেন্টেশন হয়েছেন, এভরিবডি হ্যাজ শোন লটস অফ but he didn't do any of this. But still he is so famous. We all are paying respect to him. The thing is that he became self-confident, as it was being told, that whatever you do, use the latest technology of that time. Cytogenetics was the buzzword at that particular time. I think DNA was not even discovered. DNA came almost 40 years after he was born. So, yeah, 1964 is the paper, Nobel, and 53 is the paper. So, but he did not do far from the DNA, but then the whole biology research shifted towards your data generation, standard sequencing, then your linear sequencing. But today again we are coming back to that thing. Today I was discussing with my students. Now again, cytogenetics is taking the center stage for all the linear sequencing and other things. Without cytogenetics, you cannot do much of the work which you intend to do, whether it is sequencing or more supervision or whatever it is. You cannot assign any detailed group to any chromosome if you don't have the good cytogenetics expertise or if you don't have a physical map. So the one is again coming together. All the fields are converging. Only thing is that whatever you do, use the latest technology available. As Rajiv was telling, have full confidence and question. Tomorrow, I was discussing with him. We always tell about one pathogen that we say 500 pathogen, but only 3 days back a paper has come. They question no, it does not attack around 500 species. Only 43 paper we could find in the letter. But people are telling all are citing. So it was we used to work on helicobacter for one of the damaging pests. Everybody is saying 50% damage, but you go no configuration. So if your own configuration, you start. Mukdad is one example. He took up the challenge and he has started many new things which none of us cannot do. So whatever you do, you have full confidence, do the proper configuration and use the latest technology. Reach all these people, they all are experts. Until your mentors like Rita ma'am, that is there. I think we all, whoever living to work outside Bengal, we should look towards. And also we have worked together. I always used to tell her, why don't you send us some people from Calcutta University so that we can work on good transformation and cloning and other things. And now here I am hearing different types of stories that Calcutta University. But I am very sure you are the best of the best. You come forward. And any of you are interested to work with Jude, most welcome to visit Kaizen. I am very soon going to advertise their position or Muslim expertise. Anybody who want to work on the community or cloning or transformation, most welcome to join Kaizen Laboratory. Thank you for this.
সকল স্যার আমার প্রণাম। আমার সকল সায়েন্টিস্ট স্যার ম্যাডামদের কাছে একটাই জিনিস জানার, যে আমরা যখন আমি এখন ফোর্থ সেমিস্টার স্টুডেন্ট, আমি এখন এমএসসি করছি কলকাতা থেকে। তো হ্যাঁ, আমাদের বটনি ডিপার্টমেন্টে।
তো আমার প্রশ্ন হচ্ছে আমরা যখনই জেনোমেডিক্যাল ক্লাস করি বা ক্লাস করি আমরা প্রতিটা ক্লাসে মানে পড়ছি বিভিন্ন জিনিস তারপরে শুনি যে এটা এখনো আমাদের ভারতবর্ষে চালু হয়নি। তো আমি জানতে চাইছি ভারতবর্ষ কতদিন আটকে থাকবে, মানে এর পরের জিনিসগুলো যেগুলো আমরা পড়ছি সেই জিনিসগুলো আমাদের এখানে কবে আসবে?
Global in nature, neutral. It is up to you where, when, and how you can use them for your local purpose. Suppose your purpose, and you are a researcher, so there is no boundary. চালু এখানে তো আমাদের ক্যালকাটা ইউনিভার্সিটি নেই বা বর্ধমান ইউনিভার্সিটি নেই, এটা মিস্টার সায়েন্সে আছে বা অন্য কোথাও আছে, ইভেন বশিষ্ঠ টেক্সট পাওয়া যায় না, সরি, or even you can apply or ask some foreign institution. Even while we did the technology work, I got the BT protein, crystal protein genes from all over the world. Just writing a letter, I got Japan doesn't give anything to anybody. Japan, many Japanese scientists and professors still collaborating, and they gave us
a different question. When is these ethnic crops will be available in India for the consumption?
Consumption?
Yeah.
You mean GM for consumption? I thought some science.
No.
The GM crops, yes, BT cotton already in the field, BT cotton oil, we are consuming it with or without knowing. Absolutely fine. Canola oil, we are consuming it. Canola oil, you know that it is a GM crop. That oil we are using it. And others, Bangladesh, I have eaten golden banana. I have eaten brinjal, golden rice. These are all I have eaten. Bangladesh, you can have. They are consuming it. Indian regulatory system. There are a lot of people are sitting here. They are experts on regulatory aspect. And once this question was, I discussed with Dr. Van, and Van said, "Who are the regulators? You and me are the regulators. We are killing each other. We are the regulators sitting in the regulating body, and we are killing each other. When we sit in the regulatory body, we raise a lot of questions. Even Dr. Sonti, scientists like Sonti, raise so much questions while sitting in the regulator. When he as a scientist, he is working on Arabidopsis. And you can generate lots of genetic engineering. So they made a lot of genetic engineering in Arabidopsis. When I ask this question, he said, Arabidopsis doesn't go to the field. Yeah, that's the reason he is working. It's good for nothing. Why should he work on it if there is no purpose? Anyway, it's a regulatory matter. It will take time. India takes some time, extra time. It will come. But even the BT technology, BT working, they came to the government approved it. Government did not approve it. Farmers put it in the field. And enormous area covered by the farmers. Government wanted to destroy it. Then the farmers hesitated and said, you have to pay the price for the crops they grow. And that was an enormous amount of money. And that's the reason certain things which was not approved, the government had to approve. The day may come, while many of these GM crops and other important natural oil everywhere else in the world, people are consuming it, people are using it, they might come as such, and people will be using it. And that probably have to wait for that.
By 2020, we'll get the
This is
the Archana Government of India Mega Genome Editing Project has been launched, and 10 to 15 genome editing crops are in the trial. 10 to 15 genome editing crops are in the field trial. And no genome edited crops like the genome edited banana, genome edited rice are already in the field trial. So, and this is the mission of Government of India. By 2026, it will be released.
Okay. Thank you very much. And this part, actual official vote of thanks will be given by Professor Secretary of the our alumni association. But as my responsibility to thank all of the members who are sitting here is fantastic. Even we did not even think about it.
Hello. Actually, I'm involved with teaching for the last 20 years, more than 20 years. So my main concern is about the students' future. And Shonali has told that after doing her PhD, what she is going to do, is there any job recommended? So we are not very sure that what kind of job she is going to get here. And we cannot advise all of our students to go abroad because this is not a solution to the joblessness of Westerners vis-a-vis India. Sharma said that tomorrow is a vital day for the Indian politics because there will be the Lok Sabha parliamentary election results will be come out. That may not help or interfere with the belief of our students. But I believe that it is going to happen and that is going to interfere with policies of India. Because who will come to power for the next 5 years, that will determine the policy and that will actually determine the fate of Indian students, and especially the students of Sharma. So if there is constriction of funds for the universities, for the research, then what will happen to our students? I know that research is not everybody's cup of tea, but those who are able, those who have potential are not getting properly funded for the project. But especially the universities are not getting funds for the project. If they think that the university teachers are not capable of doing research, then they should shut it down. But they are not funding, and that is causing a great problem for our students. Not only for the research work, but for the day-to-day practical also. If you don't get enough funds, we cannot do experiments, which is state of the art. We are still lagging behind with our routine experiments. And if we are provided with much more funds, then we can do better. And we know that our students are far better. And they don't know their potential because they are not exported to the maths. That's why they are far behind. They don't know even what they can do. So it is our responsibility to help them, to show them the path. First, we are all here to celebrate the birth centenary of our star. We could do this because we are now in a position. But in the next 100 years, we don't know what these students will be. And if we are not in a position to select the teachers, we will go to oblivion. To save us from oblivion, we need to place our students in better position. Thank you very much.
Before still asking any student would like to say any word or any comment. Any student?
আমি বলছি সরকারি যে ঘটনা আমাকে আমার ভালো লেগেছে সেটা হচ্ছে ওর টিচার হতে চায়। আমরা আমাদের ছাত্র জীবনে বহু আমরা যখন ছাত্র ছিলাম তখন প্রথম জিনিস যেটা ভেবেছি সেটা হচ্ছে টিচার হবো। জীবনে টিচার হওয়া ছাড়া অন্য কোনো স্বপ্ন রেখে কাজ করিনি এবং সেটাই করতে চেয়েছি। টিচার হতে চেয়ে চাইছি, স্বর্ণালীকে আমি টিচিং কমিউনিটিতে ওয়েলকাম করছি, চাই যে সে টিচার হোক। কিন্তু এখনকার পরিস্থিতি যা কিছুই পাবো না টিচিংটা টিচার হই, এরকম এই জিনিসটা খুবই আন্ডারগ্রাউন্ড টিচিং
ওদের সঙ্গে গেছে, আমরা আমাদের সঙ্গে আমাদের জীবনে বহু টিচার পেয়েছি যারা দয়া করে টিচিং এসেছে, মানে না এলেই ভালো হতো, কিন্তু টিচিং এসে পড়েছে, তাদের কাছে ছাত্ররাও কিছু পাইনি, আমরাও কোডিং হিসেবে কিছু পাইনি, হয়তো অন্য ফিল্ডে গেলে ভালো করতেন, কিছুই পারনি তাই টিচিং এসে। সম্মানের এই স্পিরিটটা আমি খুবই সম্মান করি, আমি ওয়েলকাম করি। এর পরবর্তী
Still, I'm asking again the students, if you have something left out that you want to listen or you want to say anything, any of the students? Krajap, who are the people came from Krajap? Can you please stand? People came from Krajap. It is important that if one of you should say something, who will speak? Who will speak? You have to say. Anyone?
স্যার, আমার নাম সুদীপ্তা কুমার, বিডিং ডিপার্টমেন্টে। স্যার, আমার যা কোয়েশ্চেন ছিল জিজ্ঞাসা অলরেডি করে নিয়েছে, অ্যান্সার মোটামুটি আমি পেয়ে গেছি। আর স্যার খুব ভালো এক্সপোজার, অনেক কিছু শিখছে আমরা সবাই। আর স্যার একটা রিকোয়েস্ট ছিল, আমার এখানে ফ্রেন্ডস আছে যারা বাংলা বাইরে থেকে আসে, তারা সেশন দেবো, তো ওদের জন্য কিছু মানে আপনারা কিছু বলে না ইংলিশে বাংলাতে কিছু ডিসকাশন যেগুলো ওরা বুঝতে পারেনি ওই জন্য that would be great. That would be very appreciated. I would definitely do that. Sir, I request you to come to and give them feedback. No,
Okay.
Thank you.
Thank you.
Thank you, sir, for the opportunity.
So again, my last word to thank you all of you for your brilliant presentations and discussion and interactions.
ঠিক আছে, আপনি এটা অন আছে।
Good evening, friends. After day-long deliberation, this is time to propose vote of thanks. On behalf of the Alumni Association, I am honored to propose vote of thanks to the speakers, other dignitaries, research scholars, and students participated in the symposium entitled Structural and Functional Genomics in Plants, jointly organized by the Alumni Association and Department of Botany, University of Calcutta. In scientific session, eminent scientists like Professor Himanshu Pathak, Director General ICAR, Professor Rajiv Bharsne, Director, Center for Crop and Food Innovation, Australia, Dr. Anindo Bandyopadhyay, Principal Scientist, CIMIT, Mexico, Dr. Sanjay Rokshit, Director, Indian Institute of Agricultural Biotechnology, Ranchi, Dr. Gourango Kaur, Director, CRIJAP, ICAR, Barrackpur, all delivered valuable, illuminating and pathfinding lectures, starting from climate resilient Indian agriculture and future vision to genome for food security and environmental sustainability, crop genome editing for trade development, silent revolution in maize, nutrition and energy improvement, and also genome improvement in jute, allied fibers, and its possible impact. All eminent speakers have contributed to the glory of the event dedicated to the memory of Professor Arun Kumar Sharma. Honorable speakers, we had an opportunity to bear your thoughts, and this will encourage us to organize our future events. We extend our heartfelt gratitude to all the speakers. During panel discussion on the topic prospects and progress in plant sciences, many senior scientists, teachers, I'm not going to read out all the names, they participated, including one young scientist, of course, our student, scholar, that is Dr. Shubhobroto Majumdar also, they all participated in this session, and they discussed, they interacted with the students and scholars, specifically on 4 areas. Post-MSc prospects of botany, industry research interface, research areas at national and international level, and so on. So we also extend our gratitude to all the panel members. Thanks are due to all the participating students and scholars in the said discussion. We are thankful to the head of the Department of Botany, Professor Rita Kundu, for allowing us to conduct this symposium proceedings in the Archana Sharma Memorial Hall, and her support to make this occasion a grand success. We also extend our gratitude to all the participating faculty members of the Department of Botany. Several other dignitaries participated from different institutions and universities to make this event a grand success. I myself acknowledge the contribution of all our EC members work under the guidance of our President, Professor S.K. Doctor, to make this event a grand success. I also thank to all the staff members of Botany Department for their help in arranging this event. Once again, thank you all.
মমিতা সমস্ত সুদীপ্ত এখানে এসো এসো দাঁড়াও দাঁড়াও সুদীপ্ত what I like to say মমিতা বন্দ্যোপাধ্যায় কেউ বন্দ্যোপাধ্যায় and সৌমিত্র পাল, of course you know Rita Kundu person, মমিতা সৌমিত্র রিতা তিনজন ডিপার্টমেন্টে অসাধারণ কাজ করেছে এই পেজিংটাকে অর্গানাইজ করার জন্য। আমি সুদীপ্ত, so give a big hands to thank you. ছবিটা কি অর্গানাইজ করো? আমরা বসে আছি এখানে, তোমরা একদম দাঁড়িয়ে যাও পেছনে, আর বাকিরা ঘুরে বসো। আসবে আমরা উপরে যাই, আমরা উপরে যাই। এখানে
ক্যামেরাম্যান উপরে যান।
Very good idea. It's a wonder.